We now proceed to the third session of the conference, Building Collaborative Networks to Scale Conservation Efforts, focusing on Theme 10, Strengthening Collaboration to Conserve Tree Genetic Resources. The session will be chaired by Dr. Hisashi Abe, Japan International Research Center for Agricultural Sciences, Japan. The session will be co-chaired by Dr. Rekha R. Warrier, ICFR AFGTB. The moderator of this session is Dr. R. Anandalakshmi, ICFR AFGTB. To say a few words about the moderator, Dr. R. Anandalakshmi is a scientist G in the Forest Genetic Resource Management Division at ICFR AFGTB. She has made significant contributions to seed technology, forest genetic resource conservation, and development of improved planting material for forestry species. I now invite her to kindly take over the proceedings. Over to you, madam.
A very good morning to one and all present here. Thank you, Naushad, for that introduction. Now we are into the session 3 of the third day of this conference. The topic is building collaborative networks to scale conservation efforts. I take great privilege in welcoming the chair, Dr. Abe Hisashi. Dr. Abe Hisashi is the Director of Forestry Division of JIRCAS. JIRCAS, that is Japan International Research Center for Agricultural Sciences. Sir has secured his PhD in 1995 from Hokkaido University, Japan. From 2023 to 2025, he has been associated with the Forestry and Forest Products Research Institute, Japan. Recently, from 2023 to 2025, he has been holding the position of director of this department. His area of specialization is wood science, wood identification, non-destructive wood anatomy, and wood formation and its properties. He has been functioning as visiting researcher at Swedish University of Agricultural Sciences and also a visiting professor at JIRCAS Center of Malaysia and Indonesia. Presently, he is also the Deputy Secretary of International Association of Wood Anatomists. Welcome, sir. Now I would like to introduce the co-chair of this session, Dr. Rekha R. Warrier. She is the principal scientist and head of the Division of Bioprospecting, IFGTB. She has more than 25 years of research experience in the field of forest biotechnology. She has secured her PhD in biochemistry from the prestigious institute, Avinashalingam University, Coimbatore. Some of her areas of research interest include application of biotechnology for large-scale planting, assessment and conservation of forest genetic resources, seed and stress physiology. Especially, she has concentrated on medicinal plants. And the highlight of her work is tree improvement of Melia dubia, which is a very important pulpwood and veneer species. She has been working in that species for more than 10 years and also developed dust testing guidelines for Melia dubia. Presently, she also manages a tissue culture facility at IFGTB and is involved in commercial production of improved teak varieties. And she has extended the superior clones to various private labs and state forest departments. And there are many more kaleidoscopic areas to tell about her. Due to shortage of time, I confine to this. And I welcome everybody to attend to this session and participate in very fruitful discussion. Thank you.
Thank you for your kind introduction, Dr. Anandalakshmi. Today, I'm chairing this theme number 10, strengthening collaboration to conserve tree genetic resources, on behalf of Dr. Lee, NIFOS, South Korea, because of his urgent mission in his research institute. This theme number 10 session, we have one keynote speech, 3 oral presentations, and 6 short oral presentations. First of all, I'd like to invite Dr. Jarkko Koskela for keynote speech. Dr. Koskela, floor is yours.
Thank you, Chair, and good morning to everybody. And I would like to also thank the conference organizers for giving me this opportunity to say you a few words about the international regional collaboration on forest-centric resources. As you have already heard during this conference, the distribution of many tree species extend over many countries, and also the threat they are facing, like climate change and pests and diseases, they do not respect political borders. So this makes the international regional cooperation important. Well, you have seen this is the definition for forest-centric resources, and you have seen it already in a few presentations during this conference. But I just wanted to put it here and just to remind that in our work, when we refer to forest-centric resources, it also covers the other woody plant species, shrubs, bamboos, rattans, and tree-like palms managed in the forestry context. Much of what I'm going to talk is based on the results of the second global assessment on forest-centric resources FAO published last year. The full report is available in English only, but we have prepared in brief version, and that's available in all 6 languages. I have brought some hard copies of the in brief, so I will distribute it after my talk. And also FAO prepared sort of an interactive story, which is available on the FAO website based on the report findings. So we received data and information from 77 countries. This was a bit less than we were hoped, but it's still quite good snapshot because it covers the three-fourths of the global forest area. And we also got reports from 3 regional networks and our international partners. And together with the report, we also launched a new information system, basically making available the data the countries reported for the global assessment. And we are continuing to keep the data up to date, working with countries. And when this was launched, we have got some more data. So currently we have data from 80 countries, and we hope to further increase this and also keep the data updated. So as many of you know, of course, the collaboration on forest-centric resources has a very long history. In the first conference day, you heard about INFRO, and of course INFRO is perhaps the oldest collaborative platform, which has been operating now more than 100 years, as we heard from Phil. And then also the regional network here in Asia, the Asia Pacific Forest-centric Resources Program. That's, let's say, more recent collaborative platform. That's also turning 20 years. And of course then, if you look back, I think in the 1960s and 70s, there were quite a few international regional efforts. So, and many of those are still active even today. But perhaps I start first based on the report what we know the state of the collaboration. This is showing the number of countries participating in international research collaboration on forest-centric resources. This is nearly 70% of those countries which have reported to us. So we can see that majority of the countries are participating in the international cooperation. And also if you look at the regional networks, that's also close to 70% globally. And we have some countries also which have started or indicated they are seeking opportunities to participate the regional networks in different parts of the world. Well, this is not an exhaustive list, but just to give you an example what the countries reported and mentioned in their reports. So, of course, globally, we have, like most countries, refer to the UFRO, and then also Botanic Gardens Conservation International, and also the TICNet was mentioned, of course, in those countries which are planting teak. And then in regional networks, I already mentioned AFOGEN. Of course, we have also in IFOGEN in Europe. And then as an example, under the FAO Regional Forestry Commission for North America, they also have a working group on forestry resources. And of course, many organizations, as you have also heard during this meeting, are working on forest resources, Biodiversity International, World Agroforestry, and also initiative what is called CAMCOR, coordinated by the North Carolina State University, the Kew Gardens in UK, and also OECD in the sense that they are running the scheme on certifying forest reproductive material. And also, well, FAO is also trying to support the international cooperation. So first of all, we are now, since 2009, the Intergovernmental Technical Working Group on Forest and Resources, which is sort of a continuation of what used to be the panel of experts on forestry resources, which was already established late 1960s. Then also, soon after the Second World War, the International Commission on Poplars was established. And a couple of years ago, they, of course, broadened their mandate away from poplars and willows, covering the other fast-growing tree species. And also they have a working party dealing with forest and resources. And then FAO has a body called Sila Mediterranean, basically focusing on the forestry issues on the Mediterranean countries. And they also have a working group on forest and resources. So these were also some examples what the countries mentioned in their reports. So of course, I think the reason why countries are participating in international regional cooperation so that they find it useful. So these are some benefits emerging from the reports. So of course, the first one is the sharing of information and knowledge. I think all countries appreciate this. The collaboration has also helped advance the science, but also applying the research results in the practical management of forest and resources. And also what was mentioned was developing the conservation strategies and methodologies. And also it has been helping or increasing the efficiency of managing forest and resources across the borders. And also many countries which contributed, if they have a regional strategy, they mentioned that the collaboration helps them to avoid duplication efforts. And also many countries emphasized the capacity building through the collaboration. But of course, there are also many challenges, and this is just a short list what also came from the country reports. So of course, some of these platforms have ceased to exist or become dysfunctional. One example, we had good collaboration with Denmark in the late 60s. It was the Danida Forest Research Center, what was established based on the collaborative project with FAO and Denmark. And that, of course, the Danida Research Center was very active in different parts of the world for several decades. But of course, then the donor priorities sort of changed in Denmark. So what was at the time the Danida Forest Research Center became part of the, well, first the forest and landscape, and now the activities or all the documents and so forth are still maintained by University of Copenhagen. The other point, countries mentioned that the transfer and or exchange of germ plants for research and development. Of course, now when the Nagoya Protocol came into force, countries mentioned that it hasn't stopped this exchange, but it made it a bit more complicated, like this requiring a bit more paperwork if you are doing it. So in the current day, international regulation might be rather, it would create lots of additional paperwork if we want to establish those international provenance trials, for example, which were largely established in the 60s and 70s. Also, what came from the reports was the practical application of technical or scientific concepts in the context of regional cooperation. I think one example here is the in situ conservation. So many countries still globally, they look or they consider what accounts as in situ conservation of forested resources. I know the AFOGEN network has recently also discussed this and tried to sort of increase the common understanding how to implement in situ conservation in practice. And of course, similar discussion has already taken place also in Europe. Many countries also, of course, there's still lots of shortage of financial and human resources, and also the research infrastructure. So many countries mentioned that they have a limitation in this regard, and that's sort of hindering their contribution to the regional and international cooperation. Then also seems that the scientific and technical capacity is eroding throughout the world. We also see this in basically all regions mention that they have capacity what they have built. They have scientists or even the technicians, managers dealing with forested resources. In many countries, these colleagues are retiring. So gradually, when they are retiring, there's no new generation coming which will be trained related science and also the technical capacities. And this relates to the second point that many countries mentioned the poor coverage of forested resources in a technical and vocational education, and that's also, I think, relates or covers also the academic education. We have some bright spots in few countries across the world, but the quite common picture is that forested resources as a topic is not that well represented or covered by technical and vocational as well as the academic education. And also, while we have made some progress in increasing the availability of information on forested resources, but many countries also consider that they may have gathered much specific information on their work and what they have been doing for their forested resources. And this sort of hinders their contribution and participation in the international cooperation. Well, if you then look at many of these challenges relates to what kind of capacities countries have within their own places or national level. So this makes useful to look at the institutional framework countries have in place. So firstly, what you call the national coordination mechanism. And this is, for example, we have heard yesterday in India, the national program on forested resources. So close to 70% of the countries globally, they have such a coordination mechanism in place. And many countries have sort of initiated the process to set up such a coordination mechanism. So of course this kind of mechanism also helps countries then to contribute and participate in the international regional cooperation. And we have basically seen that if you look back, and you see it's starting from the 1990, so we have seen that these mechanisms sort of are growing gradually, and of course what we have now is quite good, but there's still room to have many of these or more of these coordination mechanism in place in different countries. If we then look at the sort of the national strategies on forest-centric resources, the picture is quite similar there. Large majority of the reporting countries, they have a national strategy in place. And of course, this can be implemented in practice in many ways. Some countries have a standalone strategy. Some have it somehow linked or is part of the national forest program. Or then some countries may have a national strategy for all genetic resources for food and agriculture. So it's covering then the agricultural crops, livestock and fisheries. And then finally, it's also what we call the inventories on forest-centric resources. So this is basically documenting all the work, research collections, breeding populations, and so forth. So this is, something different from the national forest inventories, which FAO is also supporting many countries. But of course, the building block is for the inventories of forest-centric resources. You need to know, first of all, the distribution of tree species and so forth. But then what the national forest inventories typically don't, document what are the conservation units and the breeding populations and so forth, what are the seed stands. So we need a specific inventory on forest-centric resources to capture those. Well, of course, it's not all so dark, so there are some opportunities also for strengthening the international regional cooperation. Well, first, as this second global assessment tried to make the point that forest-centric resources makes many contributions to sustainable development in a broad sense, and of course including the sustainable forest management. So this is of course something it's good to keep on highlighting, especially for the policymakers. And well, the global efforts to conserve biodiversity, mitigate climate change, and combat desertification, they also still continuing. And well, Phil mentioned the presentation, of course, now in Convention on Biological Diversity. There's more sort of a clear reference to the genetic diversity and the need to countries to conserve it. Of course, it has always been in the CBD agenda, but I think now the formulation is at least a bit more clear. As I mentioned also, of course, we need to still collect more information on forest-centric resources. But of course now I would say that the situation is better than 10 years ago, that this helps us to demonstrate also the contribution of forest-centric resources in the sustainable development. Then also, we have also heard many examples during this conference that the research is advancing and the technologies available are evolving rapidly. So of course this presents an opportunity. Then of course there are quite many actually opportunities for mobilizing funding from the multilateral financing mechanism. But of course this would not support standalone projects just focusing on forest-centric resources. Typically these are more cross-sectoral and focusing on the larger topics. For example, what project like what the Global Environment Facility is promoting or financing. Well, we could also perhaps consider as an opportunity the fact that last year in July, the FAO conference welcomed the second global assessment, and it also adopted the revised global plan of action on forest-centric resources, and it invited members to strengthen their efforts to manage these resources, and also called all partners and stakeholders, including donors, to support and collaborate in the implementation of the global plan of action. So this is just to say that, of course, this is not the sort of strong political commitment. It's non-binding, but it's good reference. For example, if you are preparing research proposals, just to mention that basically all FAO member countries, which are close to 200, they have signed up for this one. So it's good to remind them of the importance of forest-centric resources. So basically the global plan of action, actually the text changed rather little. It's basically just updating the language, and we have the same 4 priorities and then 27 strategic priorities, and under them quite a few specific actions which are encouraged for the countries, but also at the regional and national level. So this, well, first the first priority is priority area is focusing on information. The second one on conservation, both in situ and ex situ. The third one, sustainable use, development and management. And then the fourth one on policies, institutions, and capacity building. But what I also want to highlight here that 1/3 of the strategic priorities, they focus on international and regional priorities. So of course the role of this collaboration is also reflected in many ways in the global plan of action. Well, I'm just to, of course, the global plan of action and the report was prepared under the auspices of the Commission on Genetic Resources for Food and Agriculture, which is one of the FAO body originally established in 1983. Well, at that time only focusing on plant genetic resources, or basically the agricultural crops. But then in 1995, the FAO conference broadened the mandate of this commission to basically cover all components of biodiversity relevant to food and agriculture. And I mentioned earlier then in 2009, the Commission established the Intergovernmental Technical Working Group on forest-centric resources when FAO was asked to prepare the first global assessment. Well, I mean that, just to highlight that, of course, now global action, we continue monitoring how it's been implemented. So the Commission work in all different sectors basically follows this cycle of first countries reporting, then FAO is preparing the assessment, then there will be a policy response discussed by the Commission, which is in this case the global plan of action. Then countries are expected and other partners to implement, and FAO is tasked to monitor and report back to the Commission the progress made. So of course in this case we are continuing the work. We have a global network of national focal points on forest-centric resources. I think we are just over more than 100 countries have nominated them. So of course we have expectation now we can continue gathering data and information from those countries which have not yet provided information. And also FAO is expected to present what we call the implementation report to the Commission basically every 5 years. It's always a bit plus minus, but more or less the 5-year cycle. And this global assessment, the Commission has applied the 10-year cycle. That's also plus minus, but as we have now published last year, so I think the Commission is going to meet next year and update its program of work. So we can expect that in about 10 years' time, they expect that countries report again, and the FAO prepares the third report for the Commission. And of course, we are continuing providing technical support to countries and continuing our collaboration also with the international partners and regional networks to support these in implementation activities. Well, just to mention an example of FAO project and programs through which we are trying to also support countries. And many of these, of course, are not directly which are focusing only on forest-centric resources, but there are components or they are very relevant to the activities these programs or projects are doing. So we have the Technical Cooperation Program, as TCP, as we call it. So basically each FAO country office, they have sort of allocation of funds in which the responsible ministry usually presents what are the country's priorities for, maybe having a small project at the country level in collaboration with the FAO. Of course, we see that, I think, 2004, all the funds FAO made available through this mechanism, it only 4% went to the forest sector, and even less to activities related to forest-centric resources. Just to say that it's often then the ministries putting forward the priorities to the FAO country office. They tend to emphasize agriculture and livestock, and often the forest is a bit neglected. So of course, I think if you have a discussion with the relevant colleagues in the ministry, maybe it's good to remind that also forests are important when countries consider this approach. But then FAO has also programs on what's called action against desertification. That's mainly in Africa, and also the forest and landscape and restoration mechanism. So of course these programs have several angles which are relevant to the forest-centric resources. Our colleagues also run what is funded by Global Environment Facility, is the Sustainable Forest Management Impact Program on drylands. That's also something we are now discussing, maybe how to embed some activities on forest-centric resources. Then, if you go to the FAO website, and there's separate websites listing all the projects FAO is helping countries to run with the funding from Global Environment Facility and then the Green Climate Fund. Of course, these are also very large projects and covering many topics, but there are a few which sort of have the angle of, especially the CHEF project, maybe conserving forest-centric resources, and in the Green Climate Fund, some projects involve, of course, tree planting, and there comes the question of how they can source the seed and seedlings. And finally, of course, we are trying to mobilize some global level projects to support the implementation of the Global Plan of Action. Okay, well, to conclude with just a few points, I think what I already mentioned, the Global Plan of Action is a useful policy document, still reflecting the needs and priorities countries have reported, and I think that's, well, various purposes, I think, useful for maybe for your work also, just to make a reference to this document. Well, overall, we see that the global awareness on the importance of forest-centric resources still needs to be improved. There are many countries mentioning that in their reports that policymakers in their countries sometimes do not realize that basically everything we do in forests, how forests contribute to the livelihoods, there always this genetic aspect there. So I think this needs to be enforced. And also finally, the international and regional cooperation, that's very crucial for the implementation of the Global Plan of Action. And of course, this is something that also needs to be reinforced. I stop here, and you can find all the publication and material from our website. We are now building a new one. It should be available maybe in 1 month's time or so, but the old one should still work, and you can find the material there. Thank you.
Thank you very much, Dr. Koshira, and for your comprehensive international collaboration and activities of FAO. But I'm very sorry to not introduce your position. Dr. Koshira is a forestry officer in Food and Agriculture Organization, FAO. I'd like to invite some questions from the floor on number 2. Okay. Oh, yes.
My question is In terms of conservation, when you put the whole process of conservation, the original thought process of a Noah's Ark, the ex situ conservation, seems that it has perfectly worked. Congratulations to FAO. Since 70s, wonderful programs like arid acacias starting from the 1960s. My question is, there are certain systems like mangroves. The very concept of ex situ conservation doesn't work at all. It is everything is in situ conservation. What is the view of FAO's conservation policy on critical ecosystems like mangroves, where ex situ conservation, like storing seeds, pollen, nothing is possible? Because
They're floating away.
so, do you have anything like, have you, you have some philosophies for mangrove in situ conservation into this group?
Well, that's a good question. Mangroves. Well, we do have a colleague which is also coordinating FAO's global work on mangroves. There's no specific genetic element there, but there has been studies done as part of the FAO's global forest resources assessment, and I think it was 2 or 3 years ago. I think FAO published a new global assessment of the mangroves. I mean, mangroves, of course. I mean, their importance is fully understood and promoted in FAO's work, both on forestry as well as the fisheries. So we have some examples of these. But specifically for in situ conservation of mangroves, we don't have any specific activities there at the moment. But I think the same principles apply. Of course, mangroves are a bit more complex in that sense, and you were right. It's a bit difficult. Well, you could have ex situ collections of mangroves, but indeed you cannot, you cannot store the seeds, or seedlings actually, because they start germinating right away. But I think the overall principles you could apply that have something specific on in situ conservation. We don't have any specific guidelines how to make in situ conservation of mangroves, but I think the mangrove publication, I think that, if I recall well, there are some overall management guidelines for improving the management and restoration of mangrove forest. I think you might find something there.
Thank you very much. On this, I just want to tag upon, we have a working group right now on Bureau of Indian Standards. We are setting up for how do we go about standards of conservations in mangroves. And shortly we are releasing that document from BIS, Bureau of Indian Standards. Coincidentally, the SSD group, I'm coordinating it. I'll be glad to share it up so that we can share it with all our regional colleagues.
Yeah.
Thank you very much.
Thank you very much. Sorry, almost time, already 10 minutes past the schedule. Thank you very much, Dr. Kochera. Please give him the hands. Next speaker is Dr. Geeta Joshi from ICFRE. Yeah, floor is yours.
Good morning to all. With the permission of Chair and Co-Chair, I would be presenting on the role of forestry extension in popularizing improved germplasm for enhancing productivity. As we all know that any tree improvement or a genetic program ultimately aims at enhancing the productivity of a population. And in India, we are to aim at enhancing the productivity. We are going to large-scale plantations, and India tops in the extent of plantations like eucalyptus 3 million, teak 2.6 million, casuarina almost 0.8 million, and other species also 1 to 2 million. However, in spite of that, the global productivity of our wood productivity in India remains very low. And it is to the extent of only 5 to 1 meter cube per hectare per year, where the average global is around 2 to 3 meter cube per hectare per year. And enhancing productivity through deployment of genetically improved and locally adapted germplasm is therefore essential for ecological and economic sustainability. And we in ICFRA, as a council, has been working on these aspects of enhancing the productivity through our various tree improvement and genetic-related programs. Before I go ahead with what I do, let me introduce you, because this is not the work, my own work. This is the work as a whole of a bunch of our scientists from the council, from different 9 institutes. So let me first introduce to ICFRA. ICFRA, the vision of ICFRA is to achieve long-term ecological stability, sustainable development, and economic security through conservation and scientific management of forest ecosystem. We have, if you can see a yellow dot on the top of India map, in between the 2 reds, that is our headquarters in Dehradun. That is near to Delhi. And right now, the bottommost, we are here at IFGTB Coimbatore. In Coimbatore we are now, which is at the bottom red dot, what you are seeing there we are. So we are 9 institutes all over India, spread across north, south, east and west, and we have 5 centers also. So this is the work what I am going to present is related to all the institutes which have, where we are doing tree improvement related work. And so tree improvement, basically, as we all know, we have been discussing for past 2 days, this is what is going on, so I will not dwell much details on it. We do the assessment of diversity, selection of superior genotypes, establishment of trials, then development of varieties and clones, production of quality planting material, and assessment of demonstration plots. This is basically in nutshell we do. And we are working on a number of species like Casuarina, Eucalyptus, Seasham, Melia. Initially we started with exotic species with Casuarina and Eucalyptus, but now we have taken on a large number of our indigenous species we have taken on. And since in last 3 decades work, we have released, so far we have released 74 clones and varieties. And these clones and varieties are based on their end product, that is the useful end product. So wood-based clones we have of Casuarina, Poplar, Eucalyptus, Seasham, and variety of Melia. This we have done. And oil-based, seed oil-based is of Calophyllum and Neem. Calophyllum clone and Neem variety we have done. And we have also been working on medicinal plants. Medicinal plants, basically we have worked for the our northern area. Our HFRA Shimla Institute has been working on it. Now, research is one part that we do, and now taking it to the end, stakeholders, to the end users is very much important. And forestry extension plays a very important role in translating these genetic gains to landscapes and communities. So that becomes very essential, because still basically when we are researchers, we are happy with publishing papers and like that, and attending conferences and publishing papers. But ultimate gain is when we take it to stakeholders and it is implemented in the field. So here I am trying to show that how ICFRA is trying to do, take it to the people, implement it in the field, and get the real benefits that are required, but that are ultimately aimed from these tree improvement programs. So what we do is we are involved in disseminating research outcomes through a combination of technologies and transfers and mechanism that we do. And in that we do go for supply of quality planting material, license agreements we do, establishment of demonstration plots, Van Vigyan Kendras and demonstration villages we have through which we do it. We conduct tree grower melas, trainings and workshops, publications, documentaries and videos, because it is a digital world, we do for that. And also we use now these days using social media for disseminating the information. So we ultimately ensure that the quality planting material reaches forest department, industries, farmers and civil society as such. And if you look at it, our stakeholders are very diverse. It is not simply forest departments, it is farmers also, industries also, then plantation companies, even tribals, they are, because they are directly dependent on forest for their livelihood, so they are also. Then self-help groups, companies are coming up with plantation, basically paper and pulp industry. Then NGOs, university, colleges and research institutes. This for the popularization of clones and varieties, first I will be telling what we do, and then I will be giving few examples of how we have done it. So what we do is clones for popularizing these things. What we do is in-house in our institutes we produce this quality planting material. It is in the form of seedling of plantlets, that is through the tissue culture or through vegetative propagation, and then we supply it to our stakeholders. Whoever requires it, we supply them, and also we demonstrate it to them, so that whatever we have achieved ultimately when we say the quality is better, the growth is better, the wood quality is better, the seed oil content is better, we have to show it to them. So we supply it to them, make it available to them. And since we are the research institutes, we don't have that much of capability of producing the material in the mass production, large scale production. So what we do is we do the license agreement for production of quality planting material with wood-based industries, with nurseries, and also with private tissue culture lab. To our private tissue culture lab, we transfer of technology of propagation, we transfer it to them. And then we also provide technical support to state forest departments for establishing and operationalization of their tissue culture labs. And we also help the forest departments in upgrading their nurseries. Recently, we have been doing it for Himachal Pradesh Forest Department, where 32 nurseries we are upgrading. So this is the regular work that we help all the state forest departments in doing it. And since seeing is believing, so what we do is whatever we say that this much gain we have received or this is the quality planting material, we put it as demonstration plots also at different places for people to see and then take it forward. And to sensitize people about the work that the outcomes we have received, we regularly conduct trainings and workshops, not only for forest departments, but for farmers also, and even for industries involving paper and pulp industries, wood-based industries and all, so that they are able to know that this quality wood is now available in the market. So these are available, and we try to take their help also in production of, large-scale production of these materials. Next, what we do is, another mechanism that we take is we have Van Vigyan Kendras and demonstration villages we have established. So around 48 Van Vigyan Kendras we have and 10 demonstration villages we have. And there we regularly conduct training program, awareness program, and there we go for production of quality planting material, demonstration plots we establish there, and we also provide advisories to our state forest departments that how to lay out plantation, what material to use, how for maintenance and management at later stages, this we provide. And also on the insect pest management, which usually plantations face once we lay down the plantations. And on a large scale, what we try to do is, as Dr. Nagaraj was showing yesterday, we conduct tree grower melas. There, in one platform, we bring all our stakeholders and then and showcase all our technologies to them. So this is how we go ahead with our extension programs. And now I'll be talking about the impact of this research and extension activities, showing the examples of few species. Like in case of Casuarina, we have released 19 clones so far. And the wood production has increased from 40 to 50 tons to 60 to 80 tons per acre. And the rotation period has reduced from 5 to 6 years to 3 to 4 years. It has increased. And this has provided stability to farmers from fluctuating income from agriculture crops. And what we do is we do major collaborations with farmers and paper and pulp industry. And in this case, we have done 10 license agreements have been signed, where mass scale production is being done. And so far, around 65 crore plants have been supplied to stakeholders with approximate 1 crore 85,000 acres of plantation raised in different states. And this work has been done by our Institute of Forest Genetics and Tree Breeding, Coimbatore. And the cumulative gain from these clones is around more than 2 lakh crores. That is it. And yesterday, as Dr. Nagaraj was explaining, we have also produced windbreak clones in case of Casuarina. Next species, this is also Casuarina being as exotic. This is also exotic because these are the two species on which we initially started the work with. And in case of IFGTB, we have released 13 clones so far, and 3 clones are licensed. And we have done around 3 license agreements. As you can see, year-wise, we have given with different TNPL, SPB, JK Paper Mills. These are the private paper and pulp industries. So we have been supplying plants to them or providing the technology for production of quality planting material to them regularly. This has resulted in an increase of almost 15%, and benefit to farmers is around 12,000 per acre they are getting the benefit, and total benefit is around 46.95 crores that has been estimated so far. Next species that we have been working is, this species has been worked from FRI Dehradun Institute there. And in this case, this is an indigenous species. In this, we have released 10 varieties, and the productivity, which was 10 to 12 meter cube per hectare per year in unimproved population. With these, in these improved varieties, it is now 34 to 55 almost meter cube per hectare per year. The productivity has enhanced to that extent. We have supplied around 56 quintal of seeds so far, and around 1.7 crore seedlings have been established in the field. And it is expected that by the end of 7 years, we'll be getting around 70 lakh of industrial timber is expected from this. And these plantations have been established, and majorly this work has, these varieties are suitable for the northern part of India, northern and central part of India. And in those states, these have been established. And we have done 3 license agreement with nurseries for production of quality planting material, so that more plantations can come up with this. Next species that we have worked is, it is Popular. Popular is a species which is basically in North India, clones are available for this, but this is for central part of India. Bihar, we have our Institute of Forest Genetics and Productivity. Forest Institute, Institute of Forest Productivity has released 5 clones for Bihar specifically, which is suitable for Bihar region. And in the Vaishali district alone of Bihar, around 68 lakh popular plants has been planted under agroforestry models. So these species, what we are doing, we are not going only for block plantation, all the species what we are discussing, but we encourage agroforestry also. And for that, simultaneously, we develop for all these species agroforestry models also. So agroforestry models have been developed, and under this, it has been 68.4 lakh plants have been established, and which is expected to produce industrial wood of around 410 crores rupees, 410 crores. And we have been regularly providing training under this, and it is estimated that by the end of 6 to 7 years, we will have a return of around rupees 1,200 approximately that much crore return will the farmers will get from this. And we have trained the farmers in growing these plants with a buyback guarantee from the in industries, from the buyback guarantee from them. And they have also, nurserymen and farmers have gained by selling the plants also. And in this case, 7 license agreement have been signed so far with the industries. Teak tissue culture has been discussed yesterday also in detail. And in this, we have, this work has been done by IFGTB. And we have produced standardized technology for mass production of through tissue culture clones. And this has been supplied to private industries as SFDs and in different states. And more than 100 hectare of teak has come across pan-India plantations have come up. We have done 9 license agreement with different private commercial tissue culture labs. This has been done. So now private commercial tissue culture labs are producing tissue culture plants of teak. And I'm happy to share that FAO has selected teak tissue culture to encourage farm forestry as one of the success stories of, one of its success stories. And we have also supported certain SFDs like Chhattisgarh, Karnataka, Tamil Nadu, and Maharashtra in establishing tissue culture labs. There's one species which was mentioned by Dr. Om yesterday in a fleeting response about sandalwood. So we have been working on sandalwood also. Our Institute of Wood Science and Technology is working on that. And other institutes are also now collaborating with, for these species. We have standardized a protocol for a nursery protocol for mass production of quality planting material of sandalwood. We have developed package and practices for sandalwood cultivation and also standardized models for agroforestry. And we have conducted more than 70 trainings have been conducted recent past few years, and where we give information regarding seedling production, cultivation management to our various across, this has been done across India. This is a species which was confined to southern part of India initially, but now with our efforts, now it has gone to the non-traditional states like Gujarat, Maharashtra, Telangana, Madhya Pradesh, Rajasthan. This also has come up in different states. And it is estimated that a net income of 80 lakh can be obtained in 1 hectare at the end of 15 to 20 years. This is expected from, Then another species that we have been working on is neem. Neem oil is a very important part of, seed oil is important part of it. And we have developed package of, we have, along with one company, IFCO, we have released 6 genetically improved varieties of neem, which is suitable for North and Central India. These are early flowering, which flowers in 3 years as compared to 6 to 10 years in the normal unimproved populations. And the average oil content has increased, is more in this from 25 to 39%. It is more in this, in the improved population. And as a content is also more, which was 2,500 in unimproved population. In the improved population, it is up to 900 ppm it has been found. And we have also helped the states in establishing tissue culture labs for producing two type of plants. And also we have established seed multiplication areas for these varieties that we have developed in states of Gujarat, Andhra Pradesh, and Uttar Pradesh. Now, coming over to bamboo, other than the tree
Geeta,
species, we are also...
One more minute
Yeah,
to wrap
I'll
up.
finish. We are also working on bamboo. We have identified 548 superior culms of 10 species of bamboo, established 7 bamboo systems. Then we produce through our nurseries and tissue culture lab quality planting material. Through material transfer agreement, we supply the quality planting material to State Bamboo Mission and other stakeholders. Even to private commercial labs, we have been supplying technology for tissue culture in vitro propagation, and through state forest departments also we have been doing that. Demonstration plants we established, and value chain projects we are doing, where the research institute, farmer, and the paper mills are involved in, so that the farmers at the end of the growth period, they get the buyback guarantee. Then we have been giving skill development training on public propagation, cultivation, preservation, biochar, briquetting, insect, like handicraft manufacturing, so that full value chain is there. And it is, we not only provide the improved germplasm, but we also give that how to utilize it at the end. In the end, I would like to conclude with that extension activities strengthen tree seed systems, promotes participatory adoption, and accelerates the deployment of unimproved germplasm across ecological regions. And widespread uptake of improved clones and varieties has the potential to significantly enhance the productivity of India's plantation, reduce pressure on natural forests, and secure long-term raw material availability of industry. And it also contributes to ecological restoration, climate adaptation, and income enhancement of farmers, aligning strongly with the sustainable utilization and resilience of tree genetic resources globally. Thank you.
Thank you, Dr. Geeta. We understand that putting the entire work of a council into a capsule requires much more time, but you have done it very beautifully. Thank you very much. We may not be taking questions here due to paucity of time.
Thank you, Dr. Joshi. Next speaker is Dr. Rajkumar from TNFD. Dr. Rajkumar, the floor is yours.
Good morning, Chair, Co-Chair, Moderator, and distinguished scientists and participants of this work meeting. Actually, I'll be presenting a field-level applications, and my presentation may not be in the structure what the researchers are presenting, but what we are, as a park manager of a tiger reserve, very important landscape area, what we are managing the forest, I will make a brief presentation. Actually, I am representing from Satyamangalam Tiger Reserve. It is located, it is one of the important corridor, elephant and other wildlife corridor connecting Western Ghats and Eastern Ghats. If you see the map, Satyamangalam is in that connecting between the Western Ghats and the Eastern Ghats, and this is surrounded by all protected areas, wildlife sanctuaries. And this area, if you see, there is a gorge and a valley. It is called Moyar Valley, which is connecting these two important landscape. There is a plateau area, it is Talamalai Plateau and the Eastern Ghats. This area actually a wildlife area where our elephant population is about 750 around. So out of 3,000 elephants estimated in Tamil Nadu, 750 elephants are using this landscape. So this habitat has more importance. So 1 in 4 of elephant and tiger landscape also. As per All India Tiger estimation, the tiger population is in this landscape is very high. And this corridor is very important because any disturbance in this valley that will affect the gene flow of wild animal from the Western Ghats to the Karnataka side and other landscape. So this has a unique problem of invasive species, mostly Prosopis juliflora. This is the panoramic view of that area. That is a Moyar Valley. And this invasive species is posing a major threat. And our recent estimate, we have made a block, and it is about a 4,000 hectare area is invaded with approximately Prosopis juliflora, and it is suppressing the local vegetation and affects the habitat quality of that. So what we did on a pilot basis, we are planning for a removal and how we are documenting that removal and monitoring that area for subsequent regeneration status using the drone, UAV-based technology, that with AI-based UAV. And after that, how we can replicate to the rest of the area. Actually, this same methodology we use for other invasive species removal like Lantana prosopis and Senna spectabilis. And for this presentation purpose, I have taken a one small area. This is a bird view, how the important landscape is full of prosopis. This is a bird, that aerial view. If you see there, the entire ground cover is fully covered with full of prosopis. So this is the area, then we have mechanically we removed it. So after removal, we can see the maximum area is full of invasives left with very local vegetation. And we do the small sample plot and do biodiversity assessment also in that removal area. What is the regeneration of that area after removal, and what is the first year and second year removal, and for this drone we have taken an aerial view of before removal and after removal, because this is very important documentation, helpful as in planning purpose and also for ensuring the quality of the work. And this different indices, actually there is a server I cannot access from here. From there we can do a lot of assessment in that area. We can get the terrain model. In one aerial documentation, it is about at least 4,000 images are taken, and all the images are stitched, and it is creating a 3-dimension model and a 3D map, contour, similarly different indices, everything will get it. So digital surface model also you are getting it. So this is showing the after removal, and all the small one as removal material is heaped. Actually, that if I'm able to access the server, I can see the difference, but paucity of time, I'm unable to explain that one. This is a after removal image. So we can, this one small area, every month we are taking a subsequent, that area contiguous to this removal area. So like that we are taking some 25 hectare, practically we are able to do it in a month, and we are covering that area. So all this documented, and we are able to, I think, and click. So we are, So there is a tool, this is a very open source software, WebODM, which is useful for processing of all the drone images. There are different applications in it. It is very useful in our forest mapping purposes. You can go to presentation. So same way, this is for post removal. I think we can show all other the rest of the area, everything, but due to time and this technical thing, I am unable to show it. Next, can go back. So our observation after that, the removal area is subsequently monitored using drone, and we can ensure that the entire area is completely covered. So what is our next plan? That is more important, because what we did is only for small area, and in future we are in the process of finalizing a tender in which a project developer is chosen, and we will convert all this invasive biomass into a biochar and register as a carbon project in the voluntary carbon market. And the revenue realized from the carbon credit sale, as well as in the sale of biochar and the other wood vinegar, that will be plowed back to a separate account, and that money will be again, we will use it for restoration and subsequent removal of that. So there will be a sustainability in the project. So definitely that will change the way we manage that largest landscape, which is about 220,000 hectare area, and habitat for a tiger, leopard, and elephants. So this will give you a local livelihood for the tribal community and the local people also. So this is our experience and our plan for the entire area, and we have started it, and maybe within a month we will be able to launch that biochar project that will helpful in continuous sustainable way of managing this landscape. With this, I want to conclude my presentations. Any doubt? Thank you.
Thank you, Dr. Rajkumar. That was good effort from the Forest Department for eradication of the invasives as well as for their utilization.
Yes.
And I'm sure you have also developed some plans for reforesting whatever you have taken out, trying
Yes.
to bring in some other species suitable to the site,
Yes.
so that those sites are not left alone, especially since you mentioned that's an elephant corridor.
Yes. Actually, this removal area we are monitoring for the wild animal movement, and the plantation raising is always a challenge in wildlife area. So we mostly monitor for the natural regeneration, which are local plant species which are coming, and we also involve our research wing, and they do a small plot in which they are conducting study on the natural plant species. And we are planning for a in-situ nurseries in that area, so that we will take up planting after that. But this area, because of high elephant population, our that artificial regeneration or our plantation is a big challenge. The survival of the plantation is a big challenge.
Sir, I have a question. Regarding this soil seed bank. How are you going to control this? Prosopis is a species which comes up very profusely.
Yes, and that actually we have taken in that soil, we are able to see the one problem is the root, which goes up to 1 meter, then the seed bank. So our observation, once we remove the Prosopis in the next monsoon, there is a profuse regeneration. So we have to immediately take that, remove that growth, secondary growth. Then after another 3 weeks, there will be another growth. So subsequently, we have to take up for 3 removal in one season. So we have to follow it for the second year and third year. So that is the most important part to prevent further regeneration, further invasion of the species.
Thank you.
Any other doubt on this?
Yeah. Any other question or comments? Okay. Thank you very much, Dr.
Thank you. Thank you.
Okay, Mayor. I'm going to the next. We are going to the. You are going to the next speaker. Okay. Dr. Saumya Singh. from IIFM, Bhopal, India. Floor is yours.
A very good morning, everyone, respected delegates and participants. Today, I am going to present our institute. So myself, Dr. Soumya Singh, and my colleague, Dr. Gajen Singh Vishwakarma, and we both are going to present our institute, which is located in central India, Madhya Pradesh state, in Bhopal. So it's the Indian Institute of Forest Management. Next, please. Okay, sorry. Yeah, so this is our beautiful campus, and so what we thought that we were given this presentation to talk about what we do. So we thought we'll just put together the different activities we are doing, what research focus we have, and teaching and everything, and then also we can talk later about the collaboration opportunities. Yeah, so something about our institute. Our IFM is an autonomous institution under the Ministry of Environment and Forest and Climate Change, MOEFCC, which was established in 1982. IFM also, we are deemed to be university since November 2023. And we have our main campus is in Bhopal, and but we also have one campus in Kurseong, West Bengal, and we are expanding our campus. So we just I think we are starting this year in Mayurbhanj in Odisha also. So our third campus will be soon developed in Odisha as well. IFM, so we are a management school, but we have 4 verticals. So we have a research, we have a teaching, which is education here listed, we have a consultancy, and we have a training. So starting with the education teaching part, we have 4 MBA programs. So the flagship program is the forestry management, forest management. Then the second is the sustainability management. So we have MBA in sustainability management. We are just started last year, sustainable development, and also the development and sustainable finance. So we are a management school, but in a very unique way. So we have this sector-specific management, focus. And then also we have a PhD program. Our PhD student is here. She presented yesterday, I think on first day only. Then we have a training program, which is a very large focus of our institution. So the training, we are training as a management development program. We have this starting from the IFS officers to the other stakeholders. So we have in last 2 years, we have actually conducted more than 50 trainings programs, ranging from the IFS officers to the academicians to the industry folks. And I think the topic we cover actually, it covers our research focus. So in research, we have a different kind of externally funded project, also internally funded project. In externally funded project, we are currently collaborating with the World Bank, UNEP, and different, more than 15 states and UTs we are collaborating with and working with within India and outside India. About consultancy, we have several projects on that also. This is what I would take more time to talk about. So we have, in general, I mean, you're welcome to visit our website, but we just, it's a great opportunity to talk more about it. So we have 10 research area, starting from ecosystem and environment management area, which we both are coming from. Since it's a management school, we also have, the list is here. I think the most relevant would be the environment and development economics and others. So we both are coming from the environment, ecosystem and environment management area, which actually range different topics. So for example, I work on air pollution and atmospheric science. He's working on the water and water resource management, and he can talk more about that. We have forestry management. We have ecotourism. We have waste management. We have various sector, various topic ranging in environment and forest management. And then we also have a different center for excellence. starting from ecological service management, corporate social responsibility, climate change studies, which is a very focused area in our ecosystem environment management research theme. So we work on different sectors. So for example, I work on air pollution, climate change and health. There are ecology, health and climate change are also connections. So we try to cover different aspects of the environment and forestry in this research theme. And this is some of the pictures from our training programs. We have our director, Dr. K. Ravichandran, here. And then I would actually give the mic to Dr. Gajendra.
So, good morning all. So, in case of academics and research, we have now recognition of different internationals and national bodies, and we have already initiated some forms, so that we'll get accredited by ICWIS and other organizations. So, apart from this, we have a very specific scheme that is especially for the certification of our natural forest and tree outside forest. That is, brand name is Praman. So the vision and mission of that particular scheme to sustainably manage the forest and tree outside forest area, and that should be comply with all the international regulations that will be developed by India Bhopal process. So that in that particular case, we have our pilot program that is done between 1998 to 2016, in which we have worked on the 8 criteria and 37 indicators. So that is a first prototype program, and then it is incorporated in the National Working Plan code. And on the basis of that, we have working as a scheme operating agency that will provide a certification for these 3 things. Let's forest management, tree outside forest, and change of custodies. So in that way, we are now working in so that we can certified about natural forest and other resources. So in this scheme, our focus on so that the forest will get market-based incentives, we can responsibly secure the material and seamless trade, or it will also enhance the transparency and traceability of all the material that we are procuring. It will also promote to earn green credit, and we also try to align with the international standards. So for this, we have a dedicated center in our organization. We have a dedicated team that will go the field and check all the criteria indicators. So if you want to know more information, you can visit our website. We have a dedicated page for this. So all the information regarding this, it is mentioned in this particular page. So these are the training program, especially for this certification scheme. We oftenly do, so you can visit our website. So in that we have a schedule, so you can get the information. So thanks with this. Thank you very much. So
Thank
I
you.
would like to say thanks to all the organizers that give this particular opportunity to represent our institute. Thank you.
Thank you, Drs. Soumya and Gajendra Singh. That was quite an insight into IIFM Bhopal. And yeah, we are very happy that you've shared the Praman details here, because that is not known to most people. So this is a good opportunity where people would come to know about it and see where they can adopt that. And thank you for sticking on to time. Thank you very much. So in case of any questions, you may please contact them during the tea break. Right? So we now move to the short oral presentations theme. Over to the chair.
Okay, thank you very much. We are moving to the short oral presentation. There are 6 presentations here. 6 minutes? 5 minutes? 5 minutes, okay. Can I ask the presenter for 5 minutes talk? First of all, I'd like to invite Dr. Maya Bell from CFRE IFGTB Floor is yours.
Respected chair, co-chair, moderator, and distinguished delegates from different parts of the globe, scientists and student fraternity. Good morning, honored person here. Today I am going to talk about the genetic variability on the fiber-related traits of Malina arborea. Malina is one of the most important multi-purpose, fast-growing tree species grown by the farmer, forest department, and wood-based industries. This is an excellent tree species suited for the various agroforestry systems practiced in different parts of the globe, particularly in India and southern parts of the country. The banana is a farmer-friendly crop. It is very fast-growing. Due to the multi-purpose nature and copying ability, the farmers are growing the trees in the farm forestry. The wood is mainly used for paper, pulp, plywood, MDF, and light constructions. It has an excellent wood quality for construction purpose also. Despite of the importance on the multi-purpose utility, the fiber property on the wood is very, very limited. Hence, the present study aimed to evaluate the fiber properties and its derived indices of different clones selected in ICFR IFGTP. The Institute of Forest Genetics and Tree Breeding is implementing systematic tree improvement program of Malina arborea past 15 years. We have conducted extensive survey in different parts of the country, assembled the wide variable genetic resources. We have established the progeny trials. The progeny trials were evaluated based on the growth traits. The superior progenies, again, we assembled in the clone banks. So these studies were conducted in the clonal trial of Malina arborea, established by Institute of Forest Genetics and Tree Breeding. These clonal trials was 4 years age. The Malina arborea plants are planted in 3 into 3 meter spacings. Around 21 clones were planted. The wood samples were collected from the breast height. The core wood samples were collected. The collected samples were processed with the hydrogen peroxide and glacial acetic acids. As per the standard protocols, we have extracted the fiber, and the fiber analysis we did. We have studied the variation in the fiber length, fiber width, lobe and diameter, cell wall thickness, and we have also, based on the data generated from the fiber properties, we calculated the Rungal ratio and slenderness ratio and flexibility coefficient ratio. There is a wide variations on the fiber length. The fiber length is varied from 933 micrometer to 1045 micrometer with a mean of 992 micrometer. Whereas the fiber length and fiber length it's comparable with Louisiana and Casuarina. It's higher than with the eucalyptus, where commercially the pulpwood industries are using. The fiber width is varied from 14.7 micrometer to 24.38 micrometer. The fiber width also it's comparable with other pulp properties. All the parameters like lumen diameter, cell wall thickness, Rungal ratio, flexibility coefficients have the wide variation between the clones we studied, and it is also comparable. Sometimes it's higher than some of the clones are expressed the higher value of the fiber properties compared to eucalyptus. So these data we have also analyzed with the correlations with the different fiber properties. The fiber length showed the positive correlation with the fiber width and the lumen diameter. Fiber width was strongly positively correlated with the lumen diameter and cell wall thickness, but it is negatively correlated with the slenderness ratio. The lumen diameter was exhibited the positive correlation with the flexibility coefficient and negative relationship with the Rungal ratio. We have also analyzed with the fiber data with the multi-trait genotype and daguerreotype indexes. These indices grouped all the 20 clones into 2 different factors. The first factor, FA1, has the most desirable factor, fiber length, fiber width, lumen diameter, and cell wall thickness. These characters has potential to improve those genetically. Among this analysis also considers the different characters pulled together and identify the superior clones for, particularly for the pulpwood industries. Among different clones, IFGTP Malina 13, GA3, GA7, GA10 shows the superior properties will comparing all the fiber properties. So these studies also shows a significant variability on the desirable properties. It has a positive relationship with the lower Runge ratio and higher flexible coefficients. Among different 21 clones selected, IFGTP GA1, GA7 show the most promising fiber properties. It can be promoted by the wood-based industries, particularly in the pulpwood industries for the pulpwood productions. Since it's a Melena wood is used for excellent paper making, it can be considered for the farm forestry program. Thank you for the opportunities.
Thank you very much. Any questions? I have one question.
Yes.
You studied Gumerina arborea.
Yes.
Are there any relationship between wood density and fiber properties?
Yes, it is a negative correlation, sir.
Oh, negatively correlation. Even cell wall thickness.
Yes, sir. It's negative only, sir.
Okay.
Wherever the higher density is there, fiber length and fiber properties
Okay.
has a negative correlation.
Thank you very much. Any other question from the floor? Okay, thank you very much.
Thank you for the opportunities.
Please hand.
I now invite Ms. Komalari from Institute of Forest Genetics and Tree Breeding to present her paper.
Good morning, everyone. I'm Komal from IFGTB ICFRE. I'll be presenting my work on evaluation of rooting response in teak genotypes. Teak, Tectona grandis, commonly known as teak, is a valuable tropical hardwood species due to its superior properties such as high tensile strength, wood durability, and natural decay resistance against fungal and pathogens. Owing to these characteristics, the teak demand far exceeds its supply. So, as an alternative, this highlights the need for efficient clonal propagation strategies, such as tissue culture, which helps to attain large-scale multiplication of superior genotypes. However, the success of this method depends on successful rooting, on its ability to form roots. So, as studies have shown, auxin hormone helps to promote rooting. In the study, we have evaluated for 10 teak clones for rooting response under different auxin hormone and varying concentration, with an aim to identify effective auxin treatment for large-scale and uniform clonal propagation, which will help for commercial supply. So in the study, 10 teak clones, namely TG1, TG2, TG3, TG4, TG5, 8, 15, 19, and 20, were selected and treated with 3 auxin hormones, that is IA, IB, and NA at varying concentrations, 0, 250, 500, 750, and 1000 ppm concentration, and evaluated for their rooting response. After 60 days, that is after the primary hardening period, variables such as root number, root length, root width, node width, and survival data were collected, and they were subjected to statistical analysis such as ANOVA, Duncan, and Gray relational analysis. To test the effect of treatment, ANOVA was carried out, which showed that all rooting traits showed significant clonal variation, and the hormone treatment, that is auxin, influenced average root number, survival, and root effect index, while concentration influenced survival only. The interaction effect between clone and treatment showed that it was significant for average root number and survival, which shows that the treatment had effect on rooting efficiency. Further, to test the difference in the treatment mean score, post-hoc test was carried out, which showed that IBA treatment had increased average root number, whereas low concentration improved survival, and on the other rooting traits, similar response were observed. However, success of rooting depends on multiple rooting traits. Selecting an optimal treatment based on one particular trait could be misleading. So, Gray relational analysis, a multi-trait optimization approach, was carried out, which helps to integrate all these multi traits observed into a single performance-based score, and based on the ranking, the highest ranked score showed the optimal treatment for the clone, for its respective clone. So the Gray relational analysis results revealed clone-specific optimal treatments. However, auxin hormone, particularly IBA, was associated with superior performance across most of the clones, particularly in the range of 250 to 750 ppm concentration. To conclude, statistical analysis supported advantageous routing in teak is genotype dependent, and the integrated analysis using ANOVA, Duncan, and Gray relation analysis showed auxin response varies among clones. However, IBA-based treatment considered delivers superior routing performance across most of the clones. However, significant clonal variation in optimal concentration indicates that a single universal treatment is not optimal. So, clone-specific treatment optimization is required to attain synchronous routing for commercial supply. Thank you.
Thank you very much. Are there any question or comment from the floor? Okay.
Have you seen any correlation between the clone performance in the field? I mean, if some of the clones that grow best, so they are more useful, and the capacity for routing, or is just every clone is unique and do whatever? So there is not any trade-off. Is there any trade-off?
Did you see any difference in the performance because of the difference in the routing in the field? Did you see any?
Yes, sir. Yes, sir.
Yeah. Okay. Thank you.
Depending on the clone,
Yeah.
the routing performance varies.
Yeah, I have understand that, but I wonder, you are propagating these clones because they are important for forestry, and I was wondering if there is any trade-off with the properties of the clone in the field, in the productivity, thinking that maybe better clones, they may root worse because there is some kind of trade-off, or maybe not. That's my question. It's okay, we can talk later.
Sorry, I didn't hear you. Can I come in?
Yeah, we can talk later. Yeah, thank you.
Thank you.
Yeah, thank you very much. Thank you very much. Our next speaker is Dr. Agira Saj from IFGTB. Yeah, floor is yours.
Good morning, everyone. My name is Akhila, and here to present the topic, Distinctness, Uniformity, and Stability Evaluation of GK10 for registering as an extant variety of Mealyardubia under PPV FRA. So in India, many plant varieties was poorly documented, which limited its legal protection. And this gap was actually filled by Protection of Plant Variety and Farmers' Right Act that enacted in India in 2001, which provides some kind of legal protection as well as intellectual property rights to farmers, breeders, and researchers for a new or existing variety. But for registering a new or existing variety under PPV FRA, the particular variety should be passed through DEST testing. DEST testing is a prerequisite for providing or granting plant breeders' right. Test testing is nothing but distinctness, uniformity, as well as stability test, where each term stands for intervarietal variation, intravarietal similarity, as well as the stability of the character even after subsequent propagation. So here we have submitted a candidate variety called GK10 of Melia dubia, which is an ecologically and economically important tree species because it is widely used in plywood, pulpwood, as well as various agroforestry programs as an extant variety, VCK, that is variety of common knowledge, since GK10 is a pre-existing, already known, commonly available in public domain variety among the Melia dubia under PPV-FR authority. So the objectives of my study was framed accordingly, that to characterize GK10 of Melia dubia using the DESC guidelines, which is already published by this PPV-FR authority, then identify the unique characters of this GK10 that can even distinguish this particular variety from the other reference varieties of the database, and register GK10 as an extant variety VCK under PPV-FRA. The methodologies here I followed are completely based upon a standard descriptor guidelines that has published by PPV-FR authority for Melia. So the on-site dust testing was conducted for GK10 at 2 locations of Tamil Nadu, Salem, as well as Namakkal, and both the plantation were around 5 years old. Healthy 80 trees that were planted in a block with a spacing of 4x4, especially free from disease as well as pests, were taken in consideration for conducting this dust testing. And all the observation was noted only on the mature parts of the plant or the tree, as well as the samples were taken from the mid portion of the crown. And in case of these flowers as well as inflorescence, the data were taken from a fully developed panicle. Once the data were recorded, it was followed by the trait evaluation. As I already mentioned, the trait evaluation was completely based upon the DESC guidelines. The DESC guidelines of Melia is already published by PPFRA in 2018, and as per this DESC guideline, there are 40 quantitative as well as qualitative characters that represents leaf, flowers, drupes, endocarp, as well as stem branches. So each characters or each descriptor have different state of expressions that has observed during the time of development of the descriptors from this reference variety, and each state of expression is assigned with a numerical node. So this numerical node was actually recorded for the GK10. And the qualitative traits were visually scored using VS method, where a single observation was made in an individual tree, as well as the quantitative method. Quantitative traits were taken from the multiple parts of the tree, of single tree, and measurements were done using a software called ImageJ software. Once the trait evaluation is completed, the traits that were scored using this method was taken for generating the descriptor matrix. So descriptor matrix was generated using the node values of GK10 along with the node values that are recorded for 52 other reference varieties of Melia dubia in database. Then this was followed by the comparative descriptor analysis, where descriptor data were analyzed to trait group wise by pooling stem, leaf, branch, flowers, as well as drupes data separately. Its frequency and percentage of each descriptor states of the reference clones as well as the GK10 was noted, and traits were arranged in a low discriminatory power, and finally a descriptor-driven clustering was made. Then from that we identified the candidate variety with a distinct character as well as a closest reference variety for our candidate variety. Once we identified the candidate variety, we submitted the application under PPVFRA and to register it under extant variety VCK. The authority will review the distinctness, uniformity, stability of this particular variety, and if the authority find it the particular candidate is distinct from the rest else reference variety, they will provide the registration. So over to the result part, as I already mentioned, we did a descriptor-driven clustering using the data analyzed in trait group wise. Across all the clusters, GK10 actually didn't show any overlap with the rest of the other reference varieties. So it shows that the particular variety GK10 is different or distinct from the other 52 reference varieties which we used for this clustering. And during the analysis, we also identified that the MD11 from this reference database as the closest reference variety of this GK10. But GK10 as well as the MD11 variety were different in 13 characters, and out of which The fruit length, fruit width, as well as the endocarp length, endocarp width were 4 essential characters that have been denoted by the PPFRA guidelines of Melia for dust testing. So finally, we identified the GK10 have a unique combination of descriptors, therefore identified it's morphologically different from the rest of the reference varieties, and submitted the application under PPFRA, and GK10 of Melia dubia has been officially registered as an extended variety under the Protection of Plant Varieties and Farmers' Rights Authority, Government of India. So overall, about this study, like the morphological descriptor analysis or dust testing of Melia dubia's candidate variety, GK10 shows that the variety is different from the rest of the reference variety, and the registration number is as shown on the screen. And under PPFR authority, we were able to successfully register this particular variety as an extant variety and granting the legal protection as an extant variety, documenting this to the Melia dubia diversity. Thank you.
Thank you very much. Yeah, some question? Okay. Thank you very much.
Thank you.
Next speaker is Fatima Sirin from ICFRE TFRI, Jabalpur. The floor is yours.
Good morning, everyone. Good morning, Chair, Co-Chair, and Moderator. With the Chair's permission, I will start my presentation. It is unfolding the diversity of Oroxylum indicum for conservation. I will be giving a very short presentation for some preliminary results on Oroxylum indicum. Oroxylum indicum is commonly known as trumpet tree or Sheonak or Tetu. And it is a very highly medicinal value important tree, which is used in a number of medicinal preparations in the traditional systems of Indian medicines like Ayurveda, in some formulations like Dashmularishta and Chavanprash. The parts which are commonly used are the roots, leaves, as well as the seeds and fruits. So, I have to look here, I think. So the goal for this study was, and it is becoming vulnerable in many areas because the habitat is getting disturbed and it is getting degraded. So it is being threatened in the natural populations because of poor regeneration also. So the goal of this study was to study the regeneration as well as the phytochemical biodiversity as well as the genetic diversity across the different populations. So the methodology was, we carried out this study in the three central Indian states of Madhya Pradesh, Chhattisgarh, and Maharashtra. And we were able to locate 10 naturally occurring populations. And we sampled around 100 trees in these 10 naturally occurring populations. And they were geotagged and all the morphological characters were measured. And then we have collected the leaves from these trees, and we have carried out phytochemical estimation as well as the genetic diversity estimations. So the DNA was extracted through the CTAB method, and biochemical estimations, we have concentrated on 4 major biochemicals like the alkaloids, flavonoids, and sugar and the phenols. And for the genetic diversity assays, genetic diversity assessment studies, we have done the normal ones, like we have used screened 18 SSR primers, and when we screened out 11 SSR primers, which are very polymorphic in nature. And then we have done the genetic diversity parameter studies like observed heterozygosity, expected heterozygosity, AMOVA, etc. Then the results were that we found that the natural regeneration of this species was very poor across all the sites. And the major limitations we observed were that the fruits and seeds were being predated by many birds and as well as animals like monkeys. And then the seedling recruitment was very sparse. And the survival of the seedling in the nature was also very poor. And we could find seedlings only near the mother trees in the open, because this tree prefers open to grow in the open areas, not under any canopy. Then when we did the phytochemical analysis, we found that among the population, significant variation was there, and the sugars were ranging from 1.32 to 12.74%, whereas the alkaloid was ranging from 9.40 to 28.57%, and the flavonoids were ranging from 2 to 31.25%, and the phenolics were ranging from 1 to 7.21%. And we found that the observed heterozygosity was less than the expected heterozygosity, and the observed heterozygosity was ranging from 0.336 to 0.744 with an average of 0.65, whereas the expected heterozygosity was ranging from 0.849 to 0.917 with an average of 0.891. And when we did the cluster analysis, the populations fell into 3 clusters, and AMOA revealed that the within population variation was, genetic variation was to the tune of 72%, and the remaining was among the population to the tune of 28%. So we find that despite the population decline, the intra-population variation is quite high. So this variation among the phytochemical genetics and morphological characters can be later on used for the tree improvement programs and conservation. So we can conclude that this tree needs immediate in situ protection. Should be protected in the areas where it is growing in situ conditions, as well as it should be conserved under ex situ conservation in maybe in germplasm banks and all. Thank you so much.
Thank you very much. Are there any question or comment from the floor? Okay, I have one. Is this a medicinal plant?
Yeah, it is a medicinal.
Okay, the natural growing individuals are harvested by illegally by the people?
Yes, they are growing here and there. Sporadic population is there, not very concentrated population. We can find isolated trees also, and the local people collect the fruits and seeds and the leaves also, and sometimes they will cut the tree and dig the roots also for preparing the medicines.
Okay, the fruits are used for medicines.
Yeah.
Okay,
Most
root. Okay, then they have to cut the tree.
Yeah.
Yeah. Illegally harvestation.
Yeah.
Okay, harvest. Okay, please.
Many thanks for your presentation. It's a very complete study. So I have just a question about what are the plans for ex situ conservation, if you have already some plans for that.
Yes, we have some plans for that. We have looked at the populations which were having good trees in them with good morphological characters, as well as the flavonoids was more. And then we have grown them through seeds. We have grown them through seeds, and they are in our nursery, and we are planning to put them into a germplasm bank later on. Okay, thank you.
Thank you very much, Doctor.
Thank you so much.
Next speaker is Dr. The next speaker is Dr. Raja Mohan from TNFD. Yeah, floor is yours.
Respected chairs, co-chairs, respected PCCF researcher, CCF ma'am, scientists and participants. Very good morning to all. Last over 3 days in the conference discussing regarding the variety species, conservation genetics and the vegetation changes. In the vegetation changes, the impact is severe in the field. As a forest officer, we are continuously facing the problem in the form of conflict. Last 20 years, 15 years back, there was no conflict issues reported. I am a district forest officer at Tenkasi. So last month itself, I paid around 15 lakhs rupees compensation. Some of the divisions like Coimbatore and Hosur, they are paying more than crores compensation. Because the reason behind this, the a scarcity of suitable foders inside the reserve forest, and anthropogenic factors and agriculture practices in the fringe villages. In 1970, there is one British Gazette published by British in Trinivali. They mentioned about seeded banana present in the Kuttalam Reserve Forest. Now there is no seeded banana in this area. Like that, around 716 species of variety species reported as extinct, about to extinct in our Western Ghats. Our respected species of initiative, we prioritized 25 species for retrieving these variety species. From the 25 species, Forest Genetic Division, the research wing, we tried 15 species in our research lab. Around out of 15, we retrieved 7 species. In that 7 species, we are facing a lot of difficulties. We are utilizing seed as well as the vegetative propagation materials. For example, the seed itself, we are facing difficulties. Some seeds like Deuterocarpus bodiloni, we are collecting around 300 seeds, but the most of the seeds are sterile, and some of the seeds are not infilled seeds we received. Only 6% seeds only germinated. Some another species like Hypnocarpus macrocarpa, we found 3 locations, 2 populations. One is in ATR, another one is in MTR. We collected seeds from both places. We tried different seed treatment. Apart from the 2 places, the ATR population germinated well, but in MTR population, all the seeds have infected with fungus. So even the immature seeds we dissected and found that have a fungus. So detailed studies required in RET species. Like Antistropha ceratifolia species, initially last year we got a limited number of seeds and we tried in our lab, but we failed to germinate. This year we got around 40 numbers of seeds, and we utilized the soil from where we taken the RET species. We utilized the same soil. We successfully established it. Around 40 seeds, 25 seeds germinated. So the impact of the soil, then relations between the soil and the environmental is most important to establish RET species. In compared to another thing, vegetative propagation, we did all the hormonal composition as well as we did different methods for vegetative propagation. But even though we are failed, we are getting around 20% germination only we got in the vegetative propagation. Some of the species we are unable to get the rooting. Only the shooting level we have succeeded. So in this thing, in RET species, there is another, I forgot to say about wild banana. Previously, there was a lot of wild banana in the forest area, so there was no conflict issues. Now, almost in extinct conditions. Very few species only, few numbers of species only available in ATR and some part of KMTR. Musa acuminata and Musa bulbiciana. We collected a seed from ATR and retrieved. We tried a lot of treatment, and then finally we succeeded. We standardized the protocol for establishing these seedlings. Now we planted all our research centers. Last month I took the seeds and we planted in wherever we removed invasive species. So we planted there. We are introducing these things throughout our forest areas. Then in this RET retrieval, we need detailed studies in micropropagation as well as the biology, seed biology. So I requested scientific community join hand with us to establishing the RET species and mitigating the conflict issues as well as addressing the climate change. Thank you.
Thank you very much, Dr. Rajamohan. Are there any question or comment from the floor? Yes, please.
Yes, ma'am.
Thanks, Dr. Rajmohan, for a nice study on seeds of RET species. I just wanted to know about the species Dipterocarpus bodilonii. That is just 6% germination is there. So did you collect the seeds from the forest floor or
Yes,
they were
forest. We collected from forest, ma'am.
Forest floor?
Yes, ma'am.
So any predation reported in the seeds of the species? Predation, like were they complete or they were damaged?
Ma'am, actually, we collected the seeds, ma'am. The embryo itself, there is the ill-filled embryos, some of the embryos not germinated, sterile natures, ma'am. We dissected the seeds, ma'am. We found there is no endosperm. Some seeds, there is no embryos. So only few seeds only having the fertile embryos and endosperm, ma'am.
Okay. And so what is the extent of the population of the species in the Western Ghats? Any idea?
In 8-year Anamala tiger reserve, ma'am, we have around 6 trees we found, ma'am. Very limited numbers, ma'am.
So it is it's of high conservation concern.
Yes, ma'am.
So
Yes,
maybe you'll have to go for the vegetative propagation of the species.
Yes, ma'am. We are doing vegetative propagation, ma'am. But
Right.
we are working on the standardization of protocol, ma'am.
Right. Thank you.
Thank you, ma'am.
Thank you very much. Thank you very much, Dr. Jamohan. I'd like to go to the last speaker, Dr. Ganesh Kumar. Yeah, floor is yours.
Hello. Through this poster, I wish to show how the population genetic parameters, if analyzed effectively using the available tools, can maximize the genetic gain of a particular population, particularly captive bred population. Good morning to all. My poster topic is Genetic and Demographic Management of Captive Asiatic Lion Population, Panthera leopessica. We all know this is listed under red list of IUCN, and Apatix is one of CITES, which needs highest protection. The problem statement here is this animal, this Asiatic lion is restricted to a particular forest area, that is Gir of Gujarat state of India. It's a Junagadh, erstwhile Junagadh area. Here only the wild population exists. All the remaining animals are available in zoological parks spread throughout the country. So these are all the captive animals, and only the wild population is available at Gir Forest of India. So the problems comes because of this single location, and the remaining animals are captive population. There is always possibility of stochastic events, catastrophic events like inbreeding, depression, etc. So how to minimize this and how to maximize the genetic diversity would be the objective. So the objective is to analyze the genetic and demographic parameters of this captive Asiatic lion population to maximize the genetic diversity and demographic security of the captive population. So here genetic diversity, I mean the heterozygosity, the founders' heterozygosity. Who are the founders? Founders are the animal who don't have any pedigree. We don't know about their pedigree. We don't know about their parent, who is the dam, who is the sire. So they are all wild caught. They are all the founder animals for any population, a viable population. So we have to maximize the heterozygosity of the founders in a captive population to maintain the genetic variability or genetic diversity of that particular population. That would be the objective of this study. So materials methods I used are, I took a stud book data of almost 105 animals of this Asiatic lion, which is spread over the country, almost from painstakingly taken from 1958, from 16 zoological parks of India. And the sample size was 473 animals. 10% was wild caught, remains are captive bred population. The software used for this study was SPARCS, that is single population analysis random kinship software, which is a least square based software. And one more is PM 2000, that is Population Management 2000, to analyze the demographic structure. So you can see the pedigree structure, if it is visible here, it's very, I think, faint. The top ones are the wild animals, that is the founders. They contributed to two animals, Carol and Paul. Then again, they contributed to the next animal, in the year 1997. Like this, the pedigree structure was like so many layers. So these were traced up to the year 1958. That is, up to the founder, these structures was traced and used for the analysis. So the parameters analyzed are founder contribution, founder, who is the founder I told, gene diversity, mean kinship, inbreeding coefficient. These are all the genetic parameters we analyzed. And all the demographic structures like parameters like age structure, generation length, and population growth rate, these were all analyzed. Now we will get into the genetic analysis results. Coming to the genetic analysis result, fortunately, the population retained around 93% of genetic diversity. That means the remaining was lost during the captive breeding because of the inbreeding and other reasons. So the statistics are founders. We found around 25 founders in this entire sample size. I said the founders are the wild caught which don't have any predecessor, ancestor. They are the fresh genome. They have the maximum, they contribute maximum heterozygosity to the population or genetic diversity to the population. So we found 25 founders in this population. But unfortunately, even among that 25, 6 were not at all used for breeding. 25 founders were there, but only 19 were utilized because of the poor planning. 6 animals were not at all bred in the entire population. So we have that advantage. Likewise, the founder genome equivalent. Founder genome equivalent is nothing but the founders who are available to maintain the genetic diversity and to transmit that diversity from one generation to next generation. That is the founder genome equivalent. It was found to be around 8. And gene diversity retained, I told, 93% retained. And last was around 7%. And mean kinship was 0.06 and inbreeding coefficient was 0.07. Here we have to see in a genetically viable population, the inbreeding coefficient should be always lesser than the kinship. Kinship is nothing but the relatives. That is, if an animal is there, how many brothers he is having, co-brothers, he is having sisters, niece, nephew, and all this. These are all kinship. But inbreeding coefficient, you all know, it is the probability that a particular offspring receives both the alleles from same allele from both the parents. If same allele are come from both the parents, it may most probably lead to deleterious effect only. That is called inbreeding coefficient. So here the inbreeding coefficient is more than the kinship, which is not so desirable. Another one is effective population size.
Dr. Ganesh, the time.
Yeah. I think Dr. Martinez can appreciate this effective population size. Effective population size is population, we know the number of animals present in a locality. Effective population is the animals which actually genetically contribute in the population from one generation to next generation. That is the effective population. The NE by N, that is the ideal and non-ideal population ratio should be normally more than 0.5. Here it is 0.20. So coming quickly to demographic analysis, the age structure was not so encouraging because there are more than 90% of animals are less than 2 years old animals. Actually, young animals should be more. But here, 60% of animals are from 6 to 16 years. The population is unstable, but it is has potential because more number of animals are available for future breeding. You can see the pyramid structure. The red one line is the optimal or potential population. It should be wide at the bottom. That is, more young ones should be at the bottom, and it should be narrow in the top. But the structure is little skewed, which is having more adults in the population. A population growth rate. If it is 1, the population is static. If it is less than 1, the population size is declining. If it is more, the population is growing. Here it is 6% for males and 3% for females. The population is generally steadily growing, which is a positive sign. The generation length is nothing but the time or period at which an adult gives birth to a young one. That is the generation length. It should be minimized at any cost. How to minimize it? We have to breed the animal at the young age. For example, Asiatic lion animals, the captive population, the optimal age for breeding is 4 years. But here in the analysis we found it is almost 8 years for both the male and female. So we should reduce the breeding age as well as we should bring the animal to mating immediately after weaning. In both the way we can maximize the genetic generation length.
Dr. Ganesh, almost time. The time is quite over.
Yeah, last one.
Okay.
So the management recommendation we are giving is we have to breed the individual with lowest mean kinship followed by individual with mean kinship who are nearing reproductive senescence. That is, they are going to retire from reproductive life. They have to be brought into. And next one is pair the individual with similar mean kinship to avoid combining rare and common alleles. Ensure the inbreeding coefficient always lesser than the mean kinship. So if we implement these management recommendations, what will be the change in genetic status given the available population? I am not saying that we are going to introduce founders freshly, but within the given available population, the founder genome equivalent will increase to 7.97, from 7.97 to 8.3. Gene diversity will go to 0.94. Mean kinship will reduce to 0.05. For example, in the last top graph, you can see the potential. We have the 6 additional potential founders. If they are introduced in this population and additional 26 founders are brought into the population,
Thank you very much.
Thank
you very much, Dr. Ganesh.
Mean kinship will be 0.01. Mean inbreeding will reduce to 0.01. Thank you all.
Yeah. Thank you very much, Dr. Ganesh. Yeah. Thank you very much for your, thank you for presenters and all organizer and all of you for your contribution to this session. No, sorry. We are wrapping up this session. Thank you very much. And maybe that was a good time to talk with, to hear your presentation. Thank you very much. And
at this outset, I'd like to thank the chair for very good comments and controlling this presentations within time. On behalf of IFGTB and the conference, I would like to give a small memento.
thank you very much.
I would like to thank my colleague Dr. Rekha Warrier also for this session, co-chairing the session. Thank you. Dear audience, we are now breaking for tea and we'll join back at what? What time will we join? At 11.
I once again request all the participants to be seated inside the hall. We are about to begin the session. Good morning all. We now proceed to Theme 11 and Theme 12, Participatory Forest Management and Forensic Timber Identification. The session will be chaired by Dr. Annalyn Codilin, University of Philippines, and co-chaired by Dr. Manisha Taplial, Director ICFRA, Himalayan Forest Research Institute. The moderator of this session is Dr. D. Rajasugunashekhar. to give a brief introduction about Dr. D. Raj Sugneshekar. He is a scientist G and head of the Genetics and Tree Improvement Division of ICFR AFGTB. He has extensive experience in forest tree improvement and genetic resource management with significant contributions to clonal evaluation, seed orchard development, and, and assessment of genetic variation and adaptability in important forestry species. Over to you, sir.
Good morning, everybody. It's indeed an honor and privilege to introduce our chairperson for this session. Dr. Annalyn Codilan is an academician and as well as a researcher from Philippines, specializing in forestry and natural resource management. Dr. Annalyn Codilan had earned her Doctor of Philosophy in Forestry Science from University of Tokyo, Japan. Her doctoral research focused on optimal management plans for multi-species industrial trees plantation in the Philippines, balancing productivity and risk. Her research interest is mostly on valuation of ecosystem services, community-based resource management planning, ecological and environmental studies, and economic analysis of land use and forest product policies. She got a huge recognition and awards, and he has been recognized by her institution for excellence in forestry education and service. She received honors from CNFR, even acknowledging the faculty contributions. And as far as his co-chair is concerned, Dr. Manisha Taplial is currently the director of Himalayan Forest Research Institute. Manisha is a distinguished forestry scientist with more than 27 years of experience in forest research, education, and extension. She is currently serving as the director of Himalayan Forest Research Institute, Shimla, the institute under the Indian Council of Forest Research and Education, MOEF. She holds a doctoral degree from Hemavati Nandan Bogna University, Srinagar, Gadwal. To say she's the first woman director of HFRI. She specializes in forest seed science and technology, seed ecology, conservation of forest genetic resources, and silviculture and sustainable forest management. With this brief introduction, this is just for the benefit of the audience. Now I'm over to the chairperson. Thank you.
Thank you, Dr. D.R. Shekhar, for that very generous introduction, and I'm happy to be serving here with my co-chair, Dr. Manisha. And thank you, everyone, for coming back on time. Our team has a difficult task of keeping all of this in time so that we will not eat up too much of your lunchtime. Okay? So, welcome again to our session on themes 11 and 12 on participatory forest management and forensic timber identification. So for this session, we will begin with a keynote presentation from the Tamil Nadu Forest Department on participatory forest management, and this will be followed by 3 paper presentations showcasing the use of DNA and genomic approaches in forestry. So without further ado, may we call on the podium I. Anwardin of the Tamil Nadu Forest Department for the keynote presentation. Let us all give him a big round of applause.
So the esteemed chair, Dr. Annalyn Codilan, the esteemed co-chair, Dr. Manisha Taplial, my dear friend and moderator, Dr. Shekhar, the director of this institution, the delegates, my colleagues from Tamil Nadu Forest Department, a very good morning to you. I am really happy to be part of this wonderful exercise. I believe that such an international exercise is taking place after a pretty long time. So I am presently serving as the Principal Chief Conservator of Forests for the research and education. in Tamil Nadu, and in the past I have opportunities of working in tiger reserve management and opportunities of working in international projects like JICA assisted projects. So today I am very thankful for this opportunity to have been invited for this. So as a conservation manager who have served about 30+ years, we all agree, I'm very sure that you will agree with me that what appears to be true and correct in conservation today may not be so in the distant future. That I have myself observed in my journey in this wonderful opportunity to try and contribute to conservation in Tamil Nadu, which is a very big challenge. You all know that environmental problems, particularly the conservation problems, are typically classical wicket problems where the reasons for the problems are many, the options are many, and one option may not suit the other stakeholder, and the direction of any action or solution may not, also cannot be controlled and lead to newer problems. So given this background of a complicated conservation scenario, I stand before you today to speak that when dealing with wicked problems, when it comes to complicated conservation problems, one of the most important imperative is involving communities in the exercise of conservation. So that stands central to today's presentation. I understand that the 2 days of deliberation are on population and conservation genetics at the micro level, at the genetic level to the population level. And I will just ease you through the presentation, which has multiple slides, but I have posted some of the slides so that those are the connecting links. I will not be taking time or reading those slides for you. So next, please. So this is the overall structure of my presentation. Conservation genetics, you know. Conservation biology is the answer. There has been so much of scientific output that has come from different institutions in the world, more so outside the country, I would say. Conservation biology in India is mostly limited to Wildlife Institute of India's multiple species-related or ecosystem-related restorations. From there, of course, ecosystem integrity is a dream for every conservation manager. Then the central topic of this speech, the keynote address, is the community inclusion. I believe the term participatory forest management now has been agreed by everyone to be actually termed as community resource management. So CRM, I would mention it as CRM. And I would quickly share out some of the success stories that the Tamil Nadu Forest Department has experienced over the years. We are still learning. I am sharing with you my learnings. Please, next please. So of course, I hope these things are visible because it's not very clear over here. So the conservation biology as such, it is an integrated approach, right? So the species, the genetics, the intraspecies, interspecies, then the ecological framework of the biotic and abiotic components, the processes that happens in between them, the functions of the ecosystems, all these are actually connected to the societal management, societal choice. Is there any difficulty? Oh, I have to navigate myself, right? You're teaching some basic life lessons. Huh? So, yeah, we know that population genetics cannot be thought of in isolation. So, it is very important. It is a core foundation of the biodiversity conservation framework itself, where we all know that minimum viable population is required. Spoke about the effective population size. Then we also know that extinction is irreversible. Even climate change, they say that the effects of climate change, of the many effects of climate change, many things can be reversed, but not the biodiversity loss. And of course, the genetic erosion. Yeah, next please. Oh, yeah. This is the fault of being a senior officer, not doing himself. Yeah, so conservation biology framework, I will not get into the topic of it, just quickly look into this. So again, I spoke about the scientific constructs, that is with reference to conservation biology, the basic ecosystem structure, composition and processes, then the functions and the diversity as such. Then now population metrics, you know, all the quantitative thresholds you are reading as to minimum population that is required to stop inbreeding or extinction. Then now I'm connecting it with the social interface through the landscape context. So the society interacts at a landscape level with the population, and it has a direct effect on the population metrics. So population genetics as such is very important in the sense understanding the characteristics is very important, because if a particular ecosystem has to survive, then it needs to have strong diversity in terms of population. So we all know that one of the biggest insurance against biodiversity loss of ecosystems is the diversity. The more diverse are the ecosystem, the less likely they are to be affected or eroded. So we'll go to the next topic, that which I already spoke about it. So these are the conservation yardsticks, which I will not get into this. Effective population size, they spoke about that in, Dr. Ganesh also spoke about that. So we will not get into that, but it suffices that there are some important yardsticks. One is the fragmentation. So it all boils down to the island biogeography. The bigger a patch size, the bigger chances of conservation. The better the patches are connected, the better the chances of conservation, genetic conservation or biodiversity conservation, whatever you may so. So then that also takes into us the metapopulation. So as such, now most of the conservation biology is focusing on protected area networks. And now, of course, the other effective area conservation measures, OECMs, are also coming up. But it is very important, like what convivial conservation says, that those are all human-modified environments, which they call hybrid environments or novel ecosystems. They also have a great role to play in terms of conservation. See, we are flowing into this discussion that if you want to conserve at the population level, species level, you have to conserve at the ecosystem level. If you have to conserve ecosystems, you have to conserve at population level. And all these things interact with the society through the landscape level. So just for an example, Eastern Ghats in Tamil Nadu or even Andhra Pradesh, from 1920 to till date, 15% of the forest cover is gone. So a rough estimate says that from 1,500 patches, now the patches have increased to 9,500. So that means it has doubled six times. So human activity is modifying the landscapes. So society ecosystem interface, so this is very simple actually. How do they affect, how a land use is decided is societal choice. It has an effect on your ecosystems, the functionality of the ecosystems. Of course, the disturbance regime, fire is a very serious thing. The grazing is another. The governance systems, they are all developing up in a country like India. So now I come to my most important point that I would like to share with you based on my field experience. It is my learning only. I am subject to change. So the community involvement is not ancillary, but it is important for acceleration actually for conservation. So participation is a biological necessity. It is, don't look at that participation is going to help people or it is going to empower people. It is going to increase their capacity or increase to make them come out of their livelihood dependency on natural resources. There are multiple factors. But there is also this reality that participation is a biological necessity. So what happens when a good participation comes? The fragmentation reduces through maintenance of the corridors, both for flora and fauna. There is a reduced habitat degradation, or because people are aware, people are brought into the planning process. Then the people's requirement also met in a sustainable basis. Then the early warning systems like what is coming up in community-based monitoring, which is an important component in the present TBGBCCR project, where like typically Keystone, I believe if anyone is here, so they are coming out with barefoot ecological monitoring with community. We have to develop those systems. So governance and participation, I will get into this participatory monitoring or CRM. So what is CRM as such is in Tamil Nadu, we are looking into a framework where we look into a commitment, reciprocal commitment. The communities take up the conservation, the government organizations take up the requirement of the community, and there is a common commitments to conserve and improve, common commitment to protect the nature or functionality of the ecosystems. Then the other part is that incentivization. That is also one important part, the incentivize conservation, assist regeneration. How do we incentivize the communities so that they take interest? So that is also one thing. Then institutional architecture, right now there are two ways of looking at in Tamil Nadu. A new hybrid has come now. One is that as per the joint forest management guidelines of the country, we have joint forest management committees, which are basically at patch level. One country will look into, say, 300 hectares. So when you connect all these patches together, it becomes landscape level approach. The other side is that these are basically for non-protected areas. So protected area, in protected areas, the concept of these eco development committees have come up. And the eco development committees, there the relationship is mutual. Wherever the communities are living in a zone of influence over the protected area, and the protected area has an influence over the communities, there the eco development committees come up. So these are all people-based organization. The Forest Department, Tamil Nadu Forest is an expert. You ask any of my field functionary to go approach a local community, talk to them about the benefits of a partnership, then you talk to them, create a small community level institution. It is simply done in 5 to 10 days. That is the level of expertise the department has. Now we talk about participation. To what level is the participation? If you classical Einstein's ladder of participation, you will see the final one is totally transferring the governance to the communities. Are we at that level in India or Tamil Nadu? No. We are at the level of mutual understanding, or there are multiple steps in that which I will not get into. There is a theoretical framework. So this barophilic government always. So one important aspect is that the cultural dimensions of conservation. So when we talk about the physical carrying capacity with reference to flora or fauna, we also have to talk about the cultural carrying capacity. To what level of the population size of a particular species, or maybe for example elephants, that communities would like to have in their neighborhood. So these are the concepts that now we have to bring into our forest management and concern. Again, we have to link all these things with conservation of even at the genetic resource level. Very important one new awareness that has come is that now you increase the valuation that a society has over a system. So how do you increase the valuation? Multiple things, incentive. Then the latest development is connecting with the heritage. The more people understand that this has been part of the heritage, the more interest they have in maintaining this. So a lot of newer initiatives are taken up by the forest department in this. So I think I have completed 30 minutes. I will finish it within another 10 minutes or so. So now how do we translate our understanding of dealing with the ecosystems for conservation up to genes, the whole ecosystem as such? Then there are in India these two type of approaches are the macro filter, where you feel that you take up a landscape level or ecosystem level conservation, not specifically fitting into any kind of any particular species. You feel that if you conserve tigers, you have a tiger reserve. There are elephants, you conserve elephant. You feel that we assume that the conservation benefits pass on to the smallest member of the species member of the entire ecosystem, which may be true, which may not be true, but most likely it should be true. There is a positive hope. We don't have evidence that has come out of this, but there are some broad picture that is emerging. So this is Madras hedgehog, which is very unique to Theri landscape, inland desert landscape in Tuticorin and Ramnad. So the macro filter approach is this. Micro filter approach is the smaller species recovery programs. We come out with Tamil Nadu Forest Department has come out with species recovery programs of dugong, species recovery program of 25 endangered species in Tamil Nadu. There are multiple individual like Decalipus hamiltoni or Ariel patera. All these things are simultaneously running. Specific species recovery program, ecosystem recovery programs like restoration programs like grassland ecosystem management is of late given focus by the department. In the morning there was a question on restoration ecology of restoration measures of invasive alien species. Now we have developed almost a framework to repopulate these invaded areas with grasses first. We are not dreaming into making it as a wooded land to begin with, because it was never a wooded land. That is why the invasion came. So then the ecosystem recovery programs, which I have already told you. Some of the important ecosystem recovery programs that are taking place are mangrove restoration. Almost we have doubled the mangrove treatment area in Tamil Nadu from 4,100 hectares to 9,000 hectares. Seagrass recovery, almost we have got about 14 species of the seagrass in Tamil Nadu, in Gulf of Mannar and Park Bay. And we are also looking into this ecosystem recovery program. I am building this, connecting it with the community resource management as I go by. So the coral reef rehabilitation, I just put this so that we say that we look at the micro level, subcomponent level. We even have a slender loris sanctuary. We have a big dugong conservation reserve. So we'll just get into this. Of course, I'm moving towards the success that the Forest Department has seen. So to work with the fishermen of the entire Gulf of Mannar Biosphere Reserve, which is a large, one of the largest coral reef systems in India, we had developed this a separate trust. So that's why for undertaking any CRM measures, there has to be a very strong institution to be created, a dedicated institution, the timeline has to be given, sufficient funds has to be given, and then we have seen that our experience has shown that most likely the success is going to be very high. So that is what we saw in Gulf of Mannar. It is a clear conservation success in terms of interacting with the communities. I have given the numbers which I don't want to read. It is a very huge program where we supported through providing microcredit to the communities to increase their average family income. We made a friendship entry with the community, which then slowly we told them that these are the destructive practices of the coastal areas, that through your livelihood options, whatever you are doing, if you can modify, if you can restrain, if you can understand the relationship between your actions and the productivity of productivity and sustainability of these marine ecosystems, then most likely it will be supporting you forever. So this people have understood. We have multiple stories, good stories. You would have seen that the green sea turtle recoveries, the dugong release even happened some time back, the restoration of grassy beds. Most of these are actually affected by the trawlers at a low depth. So this is clear success story, how landscape level community resource management to look into the entire coastal ecosystems in Gulf of Mannar can perform. Now we are extending these lessons we have learned all over the state. So the next example is the scale of participation, I would say. The community level institutions are almost 2,500 in number. The community level institutions are 2,500 villages are interacting with the department. About 45 ecotourism management committees are there. So ecotourism, we have found out that Tamil Nadu's model is very successful. We find that this is one sustainable incentive practices. For example, the Parlikadu example. Parlikadu is a small place where the local people were actually using coracles to cross the river, and the department joined hands with them and made that as an ecotourism option for people of Coimbatore. And now from a modest beginning of 5 lakhs, it has got a return of about 12 crores over 15 years. So, and the impact over the community is very high, and it is protecting the Malabar pied hornbill habitat, which in turn is connected to the vitality of the tree species in which they live, which in turn is connected to the vitality of the entire ecosystem as such. So, department, Tamil Nadu department everywhere has developing this mechanism as one of the, because we are not that endowed with NTFPs like Kerala or Chhattisgarh, where there is an NTFP based engineering that is taking place. So in Tamil Nadu, interestingly, the participation approach was started in 1998 under the JICA project, where we almost selected all the degraded forest patches along the fringe of the forest. We went to the communities who were living adjacent to these areas. The communities were in the zone of influence. They are affecting the way the forest are going to be protected. Then we spoke about social sensing. We listed out the people who are dependent on the forest for their economy. We came out with alternate livelihood options to them. They accepted it. We dovetailed with the district administration, brought development to their places. And it's a very, very successful project in the entire country in terms of microcredit management. Almost 200 crore of money is available, funds are available with these villages which are all dotted on the sides of the Eastern Ghats and Western Ghats. Then again, this is more a theoretical aspect that I would like to say, which you can appreciate yourself. So there are multiple examples I can keep on telling. As there is in Kalakadu Mundandure Tiger Reserve, we came out with the livelihood systems, where the microcredit system has increased 100 times. The entire ecosystem, eco-development committees in Kalakadu Mundandure resulted in visible change in the entire landscape, even beyond the forest and the people who are living on the buffer, affecting the entire people up to 2 kilometers because the Tamrabarani river flow increase. There is a record by the Water Resources Department of the Tamil Nadu that since the declaration of the Tiger Reserve, since the development of these eco-development committees, if you go there, you will find that not a single cattle goes inside, not a fire is totally rare. Total support is provided for the conservation of this landscape. And mind you, KMTR has the largest wet evergreen forest system in Tamil Nadu. Floristically very high. You know that the Tamil Nadu is #1 in terms of angiosperm diversity in the country, 5,000 plus species. So there is a debate on the exact numbers, but you will find that most of them are in Kalakadu Mundandure Tiger Reserve. And for the purpose of audience, the 1/3 of the population in the entire Western Ghats is endemic actually. So this is what I would like to say. I think I have completed in 23 minutes. Let me conclude by saying that multiple programs are there. As I said that for Tiger Reserve there is a program. We are coming out with for entire Eastern Ghats landscape. We have now developed the Eastern Ghats Landscape Community Resource Management Program, where of 14 hills, we selected about 155 tribal communities, which are having the highest interaction with the Eastern Ghats. And we are taking up multiple measures to protect those patch level forest patches and also the landscapes outside the forest patches. For example, we are taking up soil restoration, soil moisture restoration in all this village, which is not reserved forest. This is the first intervention that the department has realized that you cannot exist without the agriculture mosaics. The forest above has to exist with the sustenance and development of the agriculture mosaics and the community interaction. So Eastern Ghats, there is a program. Western Ghats, we have very strong eco-development program in all the tiger reserves, 5 tiger reserves. On coastal side, we have Gombad, which is very active and live for so many years. So the forest department has realized that the best way of biodiversity conservation is joining hands with the community. And proudly I would say that due to the hard work of my seniors, my field staff, those people who have retired, the Tamil Nadu Forest Department, the tiger population has doubled. The elephant population is stable. The reserve forest is almost about 26,000 square kilometers. It is stable. There is a clear improvement in the topmost category, closed, that is dense forest category. So we have been doing very well only with the great support of people of Tamil Nadu. So I would take your permission to close my discussion by highlighting that conservation cannot be done in isolation of the communities. In highly developed landscapes and societal systems such as Tamil Nadu, where urbanization is almost 75%. So this is the learning that I would like to share with you. Thank you very much. Vanakkam.
Thank you so much for keeping within the time, and thank you for that wonderful presentation and sharing the success stories of participatory forest management in Tamil Nadu. So my co-chair will facilitate that question.
Now the session is open for questions. We can take a few. That was a very comprehensive presentation of various aspects of, participatory forest management. Yeah, please.
For your presentation. Actually, I don't have a question. I think the presentation was very good. I have just a comment. And when I see these conclusions, it's exactly the same we have arrived to in Europe. So we are exactly in the same line of thought that forests are socio-ecosystems. So we need to think in a holistic way. We had to look to the food landscapes, and we had to integrate the local communities. And that's the key for success. And then we can integrate genetics into like a component of this framework. But we need the complete framework. If not, will never work. So thank you so much. I think this is a really great presentation, and this is really great take-home messages that I fully agree with and I fully support. Thank you.
Yeah, thank you so much. Yeah. Thank you, everyone. Thanks to IFGTB for this opportunity.
Any more questions? If not, thank you, Mr. Anwaruddin, for a very comprehensive presentation on various aspects of participatory forest management, be it with respect to species conservation or restoration of marine ecosystems or ecotourism. Thank you very much for a nice time.
Okay, perfect. So now we will move on to our series of paper presentations. May we call on our first presenter on the podium, Dr. Suma Arundev, Principal Scientist from the Forest Genetics and Biotechnology Division of the Kerala Forest Research Institute. She will talk about DNA evidence in combating timber crimes, Kerala's first in India initiative. Dr. Dev.
Yeah, respected Chair, Co-Chair, Moderator, and distinguished delegates. So today I'll be speaking on the potential of DNA tools and DNA evidences in combating timber crimes in the country. So I'll be speaking on the potential of DNA evidences and in the combating the illegal timber offenses in the country. So I'm from Kerala Forest Research Institute. So to begin with, just a brief overview of our institute. Kerala Forest Research Institute is a premier tropical forestry research institute located adjacent to the Pichi Valani Wildlife Sanctuary in the Western Ghats. It's just 2.5 hours, 3 hours drive from the Coimbatore city. And in addition to the main campus in Pichi, we have field research stations across diverse forest types. We also have 2 sub-centers. And we have a multidisciplinary team of scientists working across 7 research divisions. And we have many live collections. The live collections include the Arboretum, which has all the tropical forestry species collection. We have a Bambusitum comprising of 75 species. We have a Palmatum. We have the across global collection of palms. And we have different collections like Orchidarium, Fernarium. And this is one of the main attractions, the butterfly garden. And then we have medicinal plant garden. We have different museums, soil museum, wildlife museum, and we have the unique Teak Museum in the sub-center Nilambur. So in 2023, we established in the institute the Center for DNA Barcoding and Timber Forensics. So this provides the DNA evidences to the timber crimes to the judiciary. Whenever there is a timber criminal offense is happening, we submit the DNA evidence to the judiciary. So timber forensics uses advanced scientific tools in two instances, one for species identification when a timber theft offense is happening. If you have to accurately confirm the species identity, we will submit the DNA barcode evidence, and when illegal extractions are happening, we can have the log to stump comparison by doing the DNA profiling. So these two are the main tools that is being practiced in the Center for DNA Barcoding and Timber Forensics. And why we need such a strong precision evidence DNA tool or DNA-based evidence for the timber theft offense is as we know, whenever the timber crimes are happening and whenever the trials are happening in the court. Recently, there was a Supreme Court ruling in one of the sandalwood smuggling case in the country. The court was unable to prosecute the illegal traders because of the lack of the scientific evidence. So this is the relevance why we should develop such a tool so that we will be having a very precise tool before the court to prove the crime and to convict the illegal traders involved. This, in a way, it can combat the illegal felling that is happening in the country. So these are some of the newspaper reports where you can see in almost all the highly priced timber tree species, illegal felling is happening. It's quite rampant in the country. And we have, when it comes to the legal, the enforcement mechanisms, unlike other countries, it is very weak in our country. And as a result, we have many cross-border smuggling is also happening. So whenever there is, especially from Myanmar, we know there is an extraction ban in Myanmar. So the Burmese teak is very globally recognized. So because we have a very weak enforcement system in our country, this Burmese teak is coming to India through the cross-border. We are bordering, we are sharing a border with Myanmar in the state of Manipur. So the Burmese teak is coming to the country, and from India it is being exported as the Indian teak. So that much weak is our enforcement system, and India has also the third best destination for illegally sourced countries, because we are heavily depending on import. So it is India is the fourth largest importer of tropical wood. So India is one of the favorite destinations for the illegally sourced country because of the very weak enforcement system in the country. So coming to I will just quickly touch upon the major drivers of the illegal felling or illegal timber harvest that is happening in the country. So we know we have been witnessing an increase in the forest cover every year through the State of India Forest Report. But still the country faces a deficiency when it comes to the timber production, the domestic use. We have a huge domestic consumption. So when it comes to the demand and supply, always there is an imbalance. So because of this, and then this is because of the heightened focus of conservation. We have many Forest Conservation Act, we have many policies in place, and we have a green felling extraction ban, a green felling ban by the Supreme Court of India in 1991. So because of this, there is a heightened focus of conservation. Almost all our forests are conserved, and we have a huge domestic demand. So definitely there will be a chance the violation of these policies. And when it comes to that, especially in a country where we have very limited means to implement these laws, also the way by which we are identifying the species whenever there is a crime is happening, we are using a very traditional means of identification by using wood anatomy, which is not foolproof, where we can identify only up to the genus level. So our legal enforcement system is very weak, and our tools of identification right now, what we are practicing are traditional tools. There should be some level of policy interventions by which we have to strengthen our tools for identifying the timber, tools for tracing the timber, or tools to confirm the falsified origin of the timber. So all these have accelerated the illegal timber theft or timber offenses in the country. So the situation demands a integrated approach. In fact, along with genomic tool, we have very high precision tools like isotope, stable isotope. So all these together in integrated framework we can develop so that we will be in a better position to combat illegal logging and the decline of timber resources in the country. So almost all the global agencies, including UNODC, ITO, and GTTN, is already accepted DNA tools as a precision tool for combating illegal felling. So coming to the tools that is being used for timber forensic applications, like in the Center for DNA Barcoding and Timber Forensics, we mainly uses two tools, as all of you must have heard about the DNA barcode tool, just like our universal UPC barcode in supermarket items, where we have a unique black and white code. We can develop a biological barcode that is being attached to a species. So DNA barcode is a species-specific tool. So DNA barcode is very much specific. Each species will be having only one barcode. So if you are in a position to develop DNA barcode for each species, and if you can generate a DNA barcode reference database for different highly traded timber tree species, we can identify, or whenever the timber theft offense is happening, if there is a false claim that this is not the highly priced timber, this is some other alternative species, then we can prove before the court, we can provide the DNA evidence to prove the species identity of the timber log that is involved in the crime. So that is one tool, DNA barcode tool. Next tool is just like our paternity testing, where we used STR profiling. Same STR profiling we can use in timber tree species, but the problem is we have to have the species-wise genome-wide STRs for each of the species. So this tool can be used whenever there is an illegal extraction is happening in the country, and if you have to compare the log to stump. So if you have the seized logs, and if there is an original stump that is located in the crime location, we can have the log to stump comparison by using STR profiling. So these two are the main tools that is being practiced in the facility, and we also experimented some of the machine learning algorithms. Say Vika is one of the very strong machine learning algorithms. There are different modules, and we have the SMO module, and this often we can practice whenever there are massive felling instances where you cannot visually go for confirmation of the species. So Vika, and for the assignment or individualization test, when we do the log to stump comparison, when massive felling is happening in highly traded timber tree species, you can use AssignePop or MicroRiza. All these platforms are tested in our lab, and it is experimentally validated. So, and these are some of the novel tools which we are now integrating into our genomic tools. These are called stable isotope ratio analysis. So, in addition to these timber theft offenses, this is one of the very strong tools that we can use for the timber traceability. When it comes to forest certification, is only gearing up in the country. So, when it comes to the timber traceability, if you have to trace the timber from the point of extraction to the point of sale, the chain of custody validation, this is a very strong tool, stable isotope ratio analysis. And along with that, we have the spectral tools like FTIR spectra, Dart Auformis spectra. So Dart Auformis spectra will give you the spectral profile of the species. It will just give you a profile of the low molecular weight, which is less than 1000 Daltons. So both these spectra will supplement your DNA barcode tool. In case if you have some very extremely shredded or decayed sample from which you were not able to get very good quality DNA, then these two tools can supplement your species. So together it can form an integrated approach where you can ensure the species authenticity, and you can trace back the timber back to its origin, and you can even confirm the false claims of the timber origin. So when it comes to KFRA, over the last 15 years of research in different phases of research that is supported by Kerala Forest Department and many other funding agencies, we were fortunate enough to develop a DNA barcode reference database for 41 highly traded timber tree species. So we have a strong reference database with us, and with this database only now that we are in a better position to confirm the species authenticity whenever there is a timber theft offense that is happening in the state. So these are the reference database. So whenever you would like to take up any of these activities, similar kind of activity in your state, the first and the most important thing is that you have to have your own database with you, before you start off the actual timber forensics activity. So these are the major stakeholders, we are catering to Forest Department, Police Department, and majorly to the judiciary. So since the establishment of the center in 2023, we had attended to 49 timber theft offenses. In 34 offenses, we submitted the evidence, and of the timber theft offenses, 87% was for species confirmation, and 99% of this was sandalwood smuggling, and 13% was related to illegal timber extraction, application where we actually did the log-to stump comparison. And this is the way by which we are generating the DNA barcode sequence, the same genomic DNA extraction, even if it is from wood. Now that we have modified very standard protocol, because that is been standardized by using trial and error method over years, using different types of specimens. Now that we are in a better position to get good quality DNA even from an extremely dried and shredded specimen, and we will generate the DNA barcode sequence. We will either use our DNA reference database, or you can depend on using blast similarity search, you can depend on the GenBank database for identification, and whenever there is 100% similarity only, we will be submitting the DNA evidence for species confirmation to the judiciary. So next, and this is the paper that has come out by using the artificial intelligence platform by which we had validated and using blind test in the lab. And this is one of the first reports that is in 2014 for identifying DNA market adulteration standard using DNA barcoding. And coming to the DNA profiling, so one of the prominent tools for species identification is DNA barcoding. Next tool is DNA profiling. Just like our STR profiling in parentage analysis, the same STR analysis like short tender repeats or simple sequence repeats profiling is used for log-to stump comparison in illegal extraction cases. So, and here also the most important thing is like we have to use genome-wide SSRs, STRs, or SNPs, which are developed from the genome of that particular species itself. So it is very highly polymorphic, and we can go for log-to stump comparison. And here also the most important thing is like you have to have a database. So across population, across distribution zone, you have to go for multiple individuals, and you have to develop your own database. And based on the database, suppose if you have a genome-wide SSRs, you will be sampling across the population, and you will be developing your database. And from that database, you have to identify the SSR which has got very good forensic potential. So there is an analysis called STR analysis for forensic application. So if you have to perform STR analysis for forensic application, you have to have a database. So database creation, of course, it will take years, maybe in one or two couple of projects only, we may be able to develop a database in any of the highly traded timber tree species. So this is the analysis trap, and we have to do the validation using blind test also for using it further for forensic application. So this is again with Dr. Yashoda ma'am and with IFGTB and KFRA, we together developed the draft genome in 2019 for TEAG for the first time, and we developed the population genetic parameters, gynecological zonation. I think ma'am must have presented this in the first day. And this particular database which we developed as part of the project, which comprises 425 individuals and with using 23 genome-wide SSR markers, which is developed from the TEAG genome itself. This particular database now we subjected to analysis, and we identified 10 best performing markers with all the good forensic parameters, and this is the work that is reported. So this is again we developed in Sandalwood X part of ACRP3. This is again the reference database, and we did the stress analysis. We identified forensic parameters, and similarly we have a huge amount of illegal trade that is happening in Indian rosewood. It has the same amount of at par with the illegal wildlife crime. As per the UNODC report, whale crime report, it is highly endangered. The Indian rosewood species is highly endangered, but and we have an extraction ban, but there is illegal trade happening. So right now we have a program that for the creation of reference database, we have a program from DST, and we have a program from ITTO and World Forest ID for creation of reference database in various tree species. And to conclude, definitely we cannot do anything in isolation as servers managing. So there should be a concerted effort, there should be a collaborative effort across researchers with policy makers and across various stakeholders, so that we can have a global reference database. The importance of global reference database in highly traded timber tree species is not to regulate or combat the trade which is happening within the country, across borders. Many of the illegally logged woods are getting smuggled. So there is a need for a global reference database. There is a need for harmonizing the forensic standards, and there should be, our legal enforcement system should be strengthened by introducing, there should be a policy level intervention by which all these high precision tools has to be integrated into the surveillance system, and we have to, and this will also help in the forest certification process. You can, for the timber traceability, you can have a chain of custody validation. You can trace the timber from the point of harvest to the point of sale. So collectively we can make out a system has to happen so that we can combat illegal felling, and we can protect all the forest genetic resources from the decline, from the resource limitation. Thank you all. So I would like to acknowledge our director, my colleagues, Dr. Donald, he is here, he is helping me with genomic tools. My colleague, Dr. Sandeep, he is helping me with chemical tools. And my former and my current PhD students, two of us are here. And all the funding agencies. Thank you.
Thank you, Dr. Suma, for a very interesting presentation on timber forensics. We just have time to take one question on the presentation. Yeah, please.
So that was a very nice presentation, thanks. In my lab, we are using similar approaches as you do. What I would like to ask you is the legal system in India accepting this kind of results by law or by case, or how does that work?
Sir, right now what we are doing is like we are restricting our activity within the state of Kerala. KFRI is located in Kerala, and so whatever the research and whatever the database so far we had developed is with the sponsorship of Kerala Forest Department. Now that the state has approved this as a nodal center under the Forest Department, so right now whatever the cases we are attending, the judiciary is very well accepting the scientific evidence, and all the samples are coming directly from the judiciary to our institute, and we are providing. But we are not in a position to take up activities outside the state because that many samples are coming to our lab. So this is a nodal center, approved center for the state of Kerala, but we are not intending to go for any national nodal agency because we are not in a position to handle the large number of samples that are coming into our lab for analysis. Thank you. Thank you, sir.
All right. Can we? Okay, please.
Ma'am.
Yes.
Is there any region-based DNA barcoding system developed by KFRM to identify the origin of the timber?
Sir, for identifying the origin of the timber, you cannot use DNA barcode. DNA barcode is a species identification tool. But of course, you can have DNA profiling, like SSRs. But genome-wide SSRs, you can have region-specific because we developed in teak and we have 3 gynecological zones and we have the database with us for the teak based on the genome-wide SSR. DNA barcode is just for confirming the species identity whenever a timber theft is happening and if you want to confirm if it is sandalwood or some other species because always there will be false claims in the court. So you can prove by using DNA barcode. But we have the SSR database for region-specific SSR database for highly traded timber tree species, say teak, for sandalwood, for rosewood, now that we have already developed for the state of Kerala. So that is the...
Thank
you. All right. Thank you, Dr. Suma, for your nice presentation and for taking up the questions. Thank you very much.
Okay, thank you so much. So after hearing the use of timber forensics in India, we will hear a similar paper, but now in Malaysia. So let's call on our next speaker, Ms. Alias Nur Nabila, Research Officer from the Forest Research Institute, Malaysia. We would like to remind our speakers to keep the presentation under 15 minutes, so we will have more time for questions. Thank you. Okay, thank you so much. So if you are interested in discussing this with Sir Anwardin, so you can approach him during lunch. Okay, Ms. Nabila, please.
Assalamualaikum and very good afternoon to all. So I am Nur Nabila binti Alias from Forest Research Institute of Malaysia, FRIM. So basically I'll be talking about the timber tracking and also the DNA barcoding. So this is from Malaysia. So this is my my title is the genomic approaches for forensic timber tracking, enhancing the Dithrocarpus species identification and DNA profiling for sustainable forest management. So for the background, various international regulations have been implemented to prohibit the global trade and importation of illegally logged timber products. As a result, due diligence diligence has become mandatory to ensure legality and supply chain traceability. So for example, is EUTR, EU Timber Regulation. So what is a due diligence? Due diligence is actually involves submitting information on timber species and source of origin, along with the documentation, proving the legal harvest of timber products. So DNA technology become one of the reliable tools in order to trace back the wood or any related species. So here, is it possible for us to extract dry wood or even the wood product to get the high quality of DNA? Is it possible? So in our country, yes, we have developed the optimized DNA extraction to get the high quality of DNA. So for DNA barcoding, we use for species identification, whereas for the timber traceability, we use the phylogeography analysis and DNA profiling, which is the STR profiling. So for the timber traceability, we can actually identify back the population and also the original stump, where the log comes from its two original stump. So for the DNA barcoding, we actually developed around 699 species of timber, and actually we have more than that. So the example is like cengal, balong, kiam, keruing, and for the DNA profiling, we have developed around 13 species. So only for these 13 species, we can actually trace back into its population or the original stump. And for the DNA barcoding, we can use for the species identification. So in our case, today we present you about the Dithrocarpus species. Dithrocarpus species, local name is keruing, or the timber trade name because it's keruing. So why is keruing? Keruing is actually it's a very commercial important genus from the family of Dithropacaceae. And in Malaysia, we have 51 species of Dithrocarpus. So we have a developed database for these 55 species of Dithrocarpus. So what is important? Because this species is listed among the 20 most well-known export species in Malaysia. Because they have many uses, as stated in front. So under the IUCN Red List threatened species, 36 out of 51 species of Dithrocarpus is actually categorized as endangered and critically endangered. So there is a very high justification in order for us to develop the DNA barcode for this genus. So here are the lists for the 55 species of Dithrocarpus. So the standard DNA barcode, a diagnostic technique of species identification of or authentication using short standard of DNA region. Sorry. So why we use this DNA barcode? Because of it's very accurate, cost-effective, and fast. However, the standard DNA barcode has limitation because it cannot discriminate all the species. In this case, from the 29 Dithrocarpus species, we can only differentiate 55% of these species because of due to the heteroplasmy and heterogeneity variation. Standard, if you know, standard DNA barcoding, we use the RBCL, ITS2, tRNA, PSBA. So this kind of DNA barcodes have a limitation. So this is the comparison between the standard DNA barcode and the new technique that we have ventured recently, which is the genomic-derived DNA barcodes. So for the genome-derived DNA barcodes, we actually resequencing the DNA for each species of Dictrocarpus. So we start to sample collection throughout the Malaysia, including the Borneo, Sabah, and Sarawak. And then these are the list of the Dictrocarpus species. And this is our DNA bank. And also we have prepared the voucher preparation. So yeah, the genomic-derived DNA barcodes, as I mentioned earlier, because it is using whole genome resequencing approach. We actually developed the new specific DNA barcodes from the resequencing approach. So these are the results. We actually managed to get 4 new DNA barcodes region specific to this Dictrocarpus. So these are the technical things. Yeah, we also developed DNA database for the genus. This database is actually open to public. It's our own database. So beside the DNA barcoding, we have the STR profiling. So the recent STR profiling we developed for Kerwin Gombang, which is the Dictrocarpus cornutus. And then this is the STR profile. And then as I mentioned earlier, the timber traceability using the STR profiling, we can actually trace back the population and also the original stump. Okay, this is the example of the DNA barcode species identification. This is more to application. If we say that we have, if we have one lot or one product, wood product, so we want to know whether this is from Dictrocarpus or not, what species is. So we can use the DNA barcode identification by, of course we need to develop the reference database. So finally we have the similarity plus and NG3, and we know that what species it is. Some others application, which is for wood industry, we identify wood species to export to EU market. We managed to extract this product, and we know that it's actually the Dictrocarpus body, which is 100% match. Also, another application is for forest department. So we can know which stump the lot come from, even the population also. Okay, for the conclusion is accurate identification of Dictrocarpus species and reliable timber tracking using DNA technologies are critical for supporting the timber industry and strengthening forest governance. So the acknowledgement with Forestry Department and Forestry of Sabah and Sarawak. That's all for me. Thank you.
Thank you, Ms. Alias Noor Nabila, for an interesting presentation on forensic timber tracking, especially with respect to Dictrocarps.
Yeah.
So now I open the session for questions, please. Any questions?
Thank you for your presentation. I'd like to know about the identification using genomic DNA. Genomic DNA is much less than chloroplast DNA. Is it possible to extract high quality, enough quality to, to be analyzed for identification, even in the timber used for several years?
Thank you, Dr. Abe, for the question. Yes, it's possible to extract the good quality from the wood. We have used the CTEC method, the conventional method, and yeah, finally we managed to get the good quality of DNA so that we can sequence the sequence for species. Then how we create the DNA barcodes, the specific, the new DNA barcodes for using the genomic. Basically, it involved the bioinformatic analysis, and we found that each species actually have unique region. It's a very short compared to the standard region. For RBCL, it has around 605 base pair, but our new region, we have the small region which is around 150 base pair or 200 base pair. So it's very unique and it's very specific to each species.
Well, thank you very much.
Thank you.
Any more questions, please? If not, okay.
Do you have problems with identifying hybrids? Because I assume that for some of these species, there must be a lot of natural hybridization. So can you separate hybrids from pure species with?
Okay, thank you, Dr. Santiago, for this question. In the case for Dithrocarpus, we haven't found a hybrid for this species, for this genus. But for other species, we have the hybrid and even the clone. For the standard region or standard DNA barcode, RBCL cannot differentiate the hybrid. But the ITS2 or tRNA-PSBA, it can actually discriminate between the hybrid or the different clone.
All right. Thank you, Ms. Nabila. Thank you very much. And we now move on to the next speaker.
Yeah. Thank you so much. So now let's welcome our next speaker who will also be talking about molecular tools for timber identification, but this time for Indian sandalwood. So let's welcome Reshma Bhasker from KFRI, PG India. Please, on the podium.
Hello, everyone. So I'll be, you might have heard about the species identification, barcoding, profiling, all those in the previous two sessions. So here we would be specifically talking about what's happening in sandalwood. So I'm here to talk on tracing the source, molecular tools for timber identification in Indian sandalwood. So yeah, why trace the source of sandalwood? Basically, we all know it fights illegal logging for law enforcement, conserve threatened species, ensure sustainability, provide legal evidence, support fair trade, and all those. But additionally, every wood has a backstory, and that backstory really matters. So the previous sessions, like yesterday and day before, we had talked about different the reasons for genetic variation, how it is assessed, what are the causes of variation and all that. So the same historical processes that drive population differentiation determine the feasibility and resolution of genetic timber tracking also. Genetic variation shaped by different demographically important forces like population divergence, gene flow, etc., forms the biological basis for timber provenance identification. So basically, Sandalam album is a commercially important, economically and commercially important species in India for its timber and essential oil, and has been facing several conservation issues related to infestations, illegal extractions, and overexploitations, and currently under the IUCN Red List of Threatened Species, which is categorized as vulnerable. So even though the government has increasing land area as plantations, and it is rising the cultivation, there are a lot of offenses also reporting, and these are from the data is from the Kerala Forest Department, where a number of offenses are being reported, sandalwood illegal offenses are being reported in the states. So all the trade statistics says that India is going to be a major net producer of Indian sandalwood by 2040, and this will increase the rate of our exploitation and illegal extraction of the natural resource. And the major problem it is that all these resources are going to the illegal traded or unauthorized sources. So you might have heard about the famous sandalwood smuggler Veerappan, and this is one of the operations that the armed forces has done to catch him. So what is the problems with sandalwood authentication is that first it doesn't have any legalized supply chain, and a lack of certified timber source is also one of the major issues. So any management related to the illegal logging or any management practice to control such thefts definitely need the conviction of culprits. So for that, for the judiciary, evidence need to be provided, and all the regulatory agencies like UNODC, GTTN, etc. Always supports DNA evidences to confirm species identity as well as verify the origin of the sourced timber. So for the study, currently we have surveyed the natural populations of Indian sandalwood across 4 major biogeographic regions like the Western Ghats, Eastern Ghats, Central Indian Highlands, and the semi-arid regions of Western India. So basically, the genome, as previously said, we require specific genome-based markers for each species. So in Indian sandalwood, we have the whole genome developed by Dasgupta et al. And the transcriptome developed by Ishwaran et al. In 2025. And those data were used to mine genome-wide SSRs and EST SSRs. And the particular polymorphism was identified, 42 polymorphic SSRs and 12 polymorphic EST SSR were mined out from the whole genome and transcriptome for the study. So basically, the initial process in any forensic tool development would be the generation of a reference database. So here we have developed a reference database of 236 individuals from 18 populations across the country using 25 microsatellite loci. And initially the genotype data was developed, and the quality was tested, like the unique genotypes were identified, potential null alleles were detected, and Hardy-Weinberg deviations and all the linkage disequilibrium, etc. were tested to verify whether the data is appropriate. So then genetic diversity parameters like the number of different alleles, effective alleles, allelic richness, number of private alleles, etc. were determined. So basically, we could see that the Western Ghats populations, basically the southern Western Ghats population, including that of the Nilgiri part as well as the Marayur, Palani belt of southern Western Ghats, as well as the populations including Jawad in the Eastern Ghats, have higher allelic richness and higher number of private alleles. So the analysis of molecular variation reveals that 12% of the genetic variation was partitioned among the population, and 88% was attributed to within population variations. So population structure was determined using Vanno method. Delta K indicated a minimum of 2 genetic clusters, and also few populations have shown extensive admixture and substructuring also. So basically, two clusters were formed. One was in the populations in the southern Western Ghats, including the Kerala, Tamil Nadu region, and also clustering the southern Western Ghats population with the semi-arid regions of Western India, and a second cluster of the northern Western Ghats and central India, the central Indian highlands formed a separate cluster. So at a higher value of k is equal to 5, we could see that the central Indian highland populations, including the Seoni from Madhya Pradesh and those from Chhattisgarh, formed a distinct cluster. Even in the discriminant analysis, we could see that the Seoni populations showed a well separated cluster, which indicates a strong differentiation of those populations. So based on that, we had like few hypotheses to be tested. Like the Indian sandalwood populations are structured into two major genetic clusters, two separate lineages in two different biogeographic regions, one probably from the southern Western Ghats and one from the central Indian highlands. So the second hypothesis was that fine scale substructuring exists within major genetic groups like that of Seoni, which is partially isolated. And the existence of such a fine subscale structuring was to be tested. Then the Eastern Ghats population represented a historically differentiated gene pool, but recent gene flow events have occurred, so that hypothesis had to be tested. And another one was that southern Western Ghats harbor more diversity and unique alleles compared to the northern Western Ghats. So that may be attributed to the higher environmental and topographic heterogeneity. So basically, to test these hypotheses, we carried out different analysis, like the genetic network analysis, which reveals the connectedness between populations. So we could see that there were certain populations like that of Hosur in Tamil Nadu and few populations in Gujarat, which acted as connecting populations. And this is also evidential from the structure where admixturing of populations were seen in these. You can also see that the Seoni population had long thin edges in the network analysis, which means that it is greatly differentiated from the rest of the populations. Similarly, relative directional gene flow estimates were found out, and it was used, the parameter was used, allele frequency asymmetry, like the NM value was used to make out a relative migration network. We could see that all Rajasthan populations and the Seoni from Madhya Pradesh had received migrants but contribute less because they seems to be isolated or fragmented. In Madhya Pradesh, Seoni, we could find strong isolation signals. So these evidences, and there was a structured gene flow where there was a sourcing dynamics between the populations. So these supports the hypothesis that southern and central Western Ghats populations represent long-term refugia that can act as sources of the gene flow, whereas the northern and peripheral populations functions primarily as the sink populations. Landscape isolation models were also tested. We could get weak pattern, a pattern of isolation by distance. The preliminary analysis reveals that gene flow is primarily constrained by landscape features and environmental heterogeneity rather than geographic distance. So connectivity analysis, the circuit theory based connectivity analysis was also carried out, which shows that high cumulative current density was concentrated in the southern Western Ghats. Hence that can be an evidence that the southern Western Ghats populations are a major connectivity hotspot and a long-term genetic refugium. Northern populations exhibited reduced current density. And bottleneck tests also revealed few inferences. So the major key findings were that southern Western Ghats reveals a genetic diversity hotspot with higher allelic richness and private alleles. But few prominent populations like that of the Nilgiri and the Marayur-Palani belt faced recent localized declines which are evident from the bottleneck assumptions. And there are a few populations in Rajasthan which have experienced demographic decline, but the historical connectivity and long-term isolation has prevented it from endangerment. And there are populations in Sioni, like all the analysis shows that they are genetically isolated with limited gene flow and connectivity. And finally, the genetic differentiation primarily shaped by landscape resistance and environmental heterogeneity. So these were the conclusions from the genetic diversity aspect. So for forensic evaluation of the DNA tool, we have a reference database, and the polymorphic loci identified was subjected to Straff analysis. Markers with high PIC was chosen and validation was done. So basically, yeah, so these are the forensic parameters analyzed for all the 25 loci, and 10 loci with highest power of discrimination, highest PIC, highest power of exclusion, and a low PM was selected for downstream forensic analysis. So individualization test was conducted using a blind test framework. So these two cases were done. One was related, where 6 stumps and 12 confiscated logs. The data was generated, and a combination of 5 polymorphic SSRs could completely cross-match the stump and the log. But when we reduced the number of primers, the number of markers could not completely distinguish. So I think minimum, so from our study, a minimum of 5 markers is reliable for cross-matching both. So assignment test, basically the population FST estimates says that the genetic differentiation is moderate. So we prefer, frequency or likelihood based assignment is preferred. So frequency based genetic assignment of all populations was done to understand the discriminatory power of the data set. So although the posterior probability values for individual assignment were generally less, this is a assignment likelihood plot which shows that the genetic a clear genetic structuring is present across the populations, and the allelic frequency patterns can identify or trace back the population origin. And cross-validation assignment accuracy was tested using DAPC. Cross-validated assignment accuracy was highest for Karnataka and Rajasthan clusters, and intermediate for Eastern Ghats and lower for Western Ghats because it's like admixed population. So unknown source plantation samples were taken, and we did Bayesian-based clustering assignment along with the reference data. So we could see some level of admixtures, but it was like the some level of admixtures could be seen, but it was able to trace back the origin. But we need like more different methods of assignments, improved reference database, and integrated approach for better accuracy. So conclusively, we have developed a DNA reference database to trace the geographic origin of illegally sourced timber. So different forest revenue departments, policymakers, judiciary, farmers, etc. Can be beneficiaries of these studies. Yeah, thank you.
Okay, thank you so much, Reshma Bashkar. Unfortunately, we have no more time for question and answer. So if you have any questions, please approach Reshma Bashkar during lunchtime. Thank you so much. Okay, so now we'll move on to the technical session on CAMPA-funded projects. We'll have a first presentation by Dr. Geeta Joshi on ICFRE, Fostering Forestry Research in India. So Dr. Geeta is Assistant Director General for Media Extension of ICFRE. Dr. Geeta. Thank you.
Can I get hand mic? It's a bit inconvenient here to look there and talk, so I thought. Yeah, good afternoon to all of you once again. And now in morning I gave a presentation about what we have been doing in genetic improvement aspects and the extension part of it, how we are taking it to people. Now I would be talking about what we are doing research in Indian Council of Forestry Research and Education. I would like to give a bird's eye view because we are doing work in 9 institutes, so it's a big work what we are doing. But here I would give a very brief, maybe the tip of iceberg, I would say, that for you to know that what kind of research we are doing. This is basically because many dignitaries are there from international agencies. We can have further collaborations once it is known that what kind of research we are doing into. So, vision, as I told about ICFR in morning, we are already into it. We are into ecological stability, sustainability, economic security, conservation, and scientific management of our ecosystem. As we saw in morning, we have 9 institutes spread throughout India. We will not repeat it. Now, the key thrust areas and themes what we are working into, there are 4 thrust areas related to research, 1 into education and 1 into extension. And into the themes of our research are managing forest and forest productivity for livelihood support and economic growth, biodiversity conservation and ecological security, forest and climate change and forest genetic resource management and tree improvement. And under these 4 themes, thrust areas, we have 33 themes under which we work on. And the other, we work on forestry education and policy research, and forestry extension for taking our research to the people. These are the major areas we are working into. Tree improvement and genetics I have already told in morning. I will not repeat it here, what we are doing into it. I will just tell you that we are into large-scale production of quality planting material through different methods, genetic diversity studies, DNA barcoding, and genomic database development, and also genetic transformation. These are the things we are into apart from what I talked in morning. This is already have told that we are into all the gist of it is that we have released 74 clones and varieties already into it, and most of it is into now we are into indigenous species. Now, next aspect we are working on is livelihood support, and that is agroforestry. There we benefit the farmers. Are directly benefited apart from our forest department are always our primary stakeholders. Other than forest departments, here we are tribals, forest range villagers and farmers are into it. And here we do identification of promising species, development of agroforestry models for different agroclimatic zones, and as for all the themes what we are doing into capacity building and livelihood enhancement. We are working on number of indigenous tree species. We are developing the models for not only the clones and varieties that we developed, but for other species also, which are fast growing timber growing species. We work into the different models of it, or economically important species we work on, and then share it with our stakeholders. We also work on fodder species. Then another aspect of livelihood support is non-timber forest products. We are number of forest dwellers dependent on forest products, and apart from that also pharmaceutical industries and many food-based industries are dependent on forest-based products. So we are into a standardization of propagation collecting practices, non-destructive and sustainable harvesting, nursery techniques and cultivation of medicinal plants, value addition and capacity buildings. We are into it. We have developed a number of forest-based products. We are sharing it with industries so that they can take up, and not only with industry but also with the self-help groups, smaller groups, those who are dependent on forest communities. We are working with them. So that, and we share the technologies, the package of practices, the products that we prepare with them, so that their livelihoods can be improved. Then another aspect we work on is forest genetic resources. Dr. Annalakshmi had talked about it day before yesterday. She has talked about it on documentation. We are working on documentation, assessment of population, structure of FGR, distribution and mapping, development of standard operating procedures, then conservation of FGRs, and we are also into rediscovery of extinct or declared so-called extinct species, or identifying new species. We are also in that area. Then in managing forests, we work on diverse aspects. Those aspects relate to forest soil health card, forest fire monitoring and evaluation of plantation, forest mensuration, preparation of working plan. There we help the forest departments. And then forest hydrology. We also work on invasive species. And we are also into the kind of studies we are doing into like evaluation of forests for abatement of pollution. These kind of studies are also we are involved into. This is one of the slides showing that forest health card. We have like soil health cards were there for related to agriculture, but for the first time ICFRA has prepared the forest soil health cards for 848 divisions of forest divisions of our country, and we have provided the capacity building trainings to our state forest departments. Then we are into forest fire. Everywhere in the world, we often see with the change of, usually in the summer season, most of the countries are facing the problem of forest fire. We also are in the same boat. And we have developed SOPs, standard operating procedures, for combatment of these forest fires and shared with forest departments. We have also tried to develop the different tools which can help in controlling the fires. Then for public outreach, we have prepared jingles and booklets and publications we have come up with and circulated to our stakeholders. Then on forest biodiversity and climate change, we work on assessment of forest biodiversity, eco-geographical mapping, climate-driven effect on biodiversity, net ecosystem carbon dioxide exchange, and also we work on RED+. In RED+, we strengthen our state forest departments on developing RED+ system and also providing training to them. Then we also are involved in national and international carbon measurements, those kind of studies, and biodiversity related, we give our inputs. Then we are also into eco-restoration. We work on degraded land and deserts, rehabilitation, reclamation and rehabilitation of mined out areas, environmental impact assessment, and on environmental audit performance evaluation. So we are in large scale into rehabilitation and of our degraded areas. Then we also provide on these aspects, we also provide consultancy on carrying capacity, environmental audit, biodiversity, reclamation and rehabilitation of landscapes. And we have developed 14 detailed project reports for rejuvenation of our rivers. And for that, only for one river, Narmada in central India, it has also already been implemented by a forest department. Then on wood science aspect, 2 of our institutes are working. All the other work, what I have shown, all the 9 institutes are doing, but wood science are 2 institutes, FRI and Institute of Wood Science Technology, Bangalore, are working into it. We work on the aspects of wood anatomy, properties and usage, drying, preservation, weathering, composites like wood polymer composites and different kind of wood composites. We are making modified woods and wood adhesives, and nanotechnology, use of nanotechnology into various aspects of utilization, preservation and coating we are doing. Here also I would say that we are also into this testing services, where we give our expertise for identification of CITES species. We are giving our inputs for forensic and police department, and also to the judiciary. The cases come to us, we do the sampling, and that reports are accepted by the agencies. Then we also do consultancy for our wooden heritage structures. We have been regularly doing that. Then we are into wood protection. There we do for protection taxonomy and damage potential of pest, population dynamics of insect pests, and integrated pest management, we are into it. And then we are in a large way into development of biopesticides and biofertilizer. There will be one presentation on that also today. Then assessment of health status of avenue trees. Urban area trees, everywhere the problems are being faced, so we are giving our inputs for and consultancies for that also. And technical advice of maintenance of heritage trees like Mahabodhi trees. They are like that, trees are there in heritage sites. For that, for maintenance of those, we are giving the our inputs. Then we have one center on sustainable land management. We have a center, and we are doing international national trainings into it. Then we are giving reporting for G20, CREX, then National Action Plan for combating desertification. These things are we are implementing project in that sense, different projects for related to land management on that center, and is regularly coming out with number of publication on, like recently we have come with drought resilience, land restoration, combating desertification, we are into it. Then the new initiative has come up in India with our ministry has implemented, and ICFR is the administrators for that. That is Green Credit Program. That is green. It aims at promoting voluntary environmentally sustainable actions through market-based incentives. Right now, state forest departments and public sector units are involved into it, and already restoration work has started in 218 land parcels. Then we are also providing technology training on green credit to concerns. Then we are into transfer of technologies. We have incubation center in one of our institute of wood science and technology. We transfer of technology to in industries, a regular practice that we do. We go for patents. Then we provide our services to advisory services to for forest departments. Then whatever work we do, we regularly come out with publications as popular articles, papers, research papers, books and chapters. That is a regular phenomena that it comes at a headquarter level also and an institute level also we are doing. Then extension activities I already discussed in morning. We have Van Vigyan Kendras, then demo villages, training, tree growers, Mela, documentaries, exhibitions, like that we are into it. There's one Prakriti, one student scientist connect program is there, where we try to sensitize students about environment and forestry at school level. This is, we are regularly into it. Tree growers, Mela we have been doing. So these are it. And then we are into education also. We have FRI deemed university. In FRI Dehradun, we have a deemed university. There we are doing PhD courses as well as 4 MSc courses are there. That is into forestry, wood science, cellulose, paper technology, environmental and environment management. And we are into accrediting forestry colleges, universities and institutes. So far 19 have been accredited. Then we have number of repositories in our all different institutes. In FRI, we have 5 such museums on entomology, NWFP, forest pathology, silviculture and timber. IFGTB, which we could not see yesterday, one of the oldest forestry museums, the gas museum is there. It is there. Then museum interpretation centers are there in almost all the institutes. Then we have insect repositories, arcanarium, xylarium. Xylarium is there in FRI and IWST. We have samples from different part of world, wood samples are there. Then fungarium is also there, and we have a very big library, national level, international level library in FRI Dehradun. We regularly sign MOUs with national and international agencies. Yesterday also from NIFOS, they were telling that we can go for collaboration. We are trying to do for collaboration with signing MOU with FRIM. So any institute interested in doing collaborative work, we can go ahead further, formalize it as MOUs and go ahead further. Then after doing all that work, what you have seen, we have been regularly doing that work. Our Ministry of Environment, Forest and Climate Change has given one scheme to us. It was given to us to work on strengthening forestry research for ecological sustainability and productivity enhancement. It was funded by our CAMPA authorities in MOFCC. It was for 6 years from 2020 to 26. It will be finishing in March now. And the project partners are not only ICFRE, 9 institutes of ICFRE, but 17 non-ICFRE institutes, outsiders were also involved in this project with us. And in this scheme, we had 6 components. The first component was on All India coordinated projects. There were 31 such projects over there, which aimed at undertaking research aimed at efficient and sustainable resource use through value addition development for appropriate technologies. So that was the aim of it. Then the second part was, as I discussed earlier also, for forest genetic resources we have worked on. Then policy studies has been done. RED Plus we have worked on, human resource development and forestry extension. These are the aspects we worked on. Today we would be discussing about the first, the All India Coordinated Project and FGR. Presentations will be there on that. Out of 31 All India Coordinated Project, 13 were species based. And on that work has been done on tree improvement and genetics aspects has been done. So we would be discussing those projects and FGR subsequently. No? Thank you. Thank you.
Thank you so much, Dr. Gita. So it seems that ICFRE staff has a very extremely busy schedule to have all of those outputs. Thank you so much for your dedication to all the ICFRE staff. Thank you. So are there any questions for the presentation of Dr. Gita? Any clarifications or any interested parties to collaborate with ICFRE? Yeah, okay, maybe you can have a discussion during lunch or after lunch. Thank you so much, Dr. Gita. Thank you. All right, so I think that concludes our session. And we can all agree that we have heard from very good presentations today. And personally, I learned a lot. And we saw how participatory forest management and forest genetics can be used to improve forest conservation, legality, and sustainable forest management. So I hope that all of us could continue to do what we are doing, continue to improve on our work and our research, and continue to contribute to the improvement of forestry sector globally. So thank you so much, and thank you to my co-chair and moderator for this session. Yeah. Okay, do you want to say a few words, Dr. Manisha? Okay.
I'd like to thank all the presenters who made their very comprehensive presentations, and on participatory forest management, timber forensics, and of course a very detailed presentation by Dr. Gita on different research activities, extension programs of ICFRE, AICRPs and all. So I thank all, and with this I hand over the mic to the moderator.
I would like to thank the chair, co-chair, and the moderator for hosting the program. Now I request the moderator to kindly felicitate the chair with a memento. Now I request sir to hand over the memento to our co-chair, Dr. Manisha Taplial. Thank you, sir.
Before we break for lunch, I have a couple of announcements. The first thing is the speakers, all the 3 days. We are intending to, put up the PDF of, copies of the presentations because there's a lot of demand. But I would request if any presenter has any reservation, they can just let us know so that particular PPT would not be put up in the online portal. So what I would do is probably after the conference is over, I will mail to all of them. So, or if you want to put up a curated presentation also is fine. So you can just let us know, and then what we are actually planning is all the conference is actually going live now. I think Phil was suggesting it would be in the UFRO also, but in the website it is going live. And all the photographs and extension materials or about the institute, everything will be available in the digital format. So we will be putting it up in the website. We'll be giving a time duration for all to download. So I will mail to all of you, and you could please let us know if we could share it as a PDF, the presentations. That's one. And the recommendations, I think I've got till yesterday. I would request Dr. Anantalakshmi and Dr. Shekhar to kindly hand over the recommendations within an hour or so. I know it's a short time. A few points so that that would be presented to the during the concluding session. So we join back for lunch after lunch at 2:15. So first we will be having, as Dr. Gita had said, that we will be talking on the All India coordinated projects which we have implemented, but we are going to mostly talk on those, the national project coordinators who are available in person. So it would be a 5-minute comprehensive presentation as what we have done, just to give a bird's eye view of the progress we have made in different species and themes. That would be followed by a presentation by the exhibitors. We have, I think, 2 of them are available today. So they would be making a short presentation about their innovations and technological, breakthroughs they have done. And that would be followed by the concluding session. So I request all to kindly join for lunch now. Thank you. And we'll be back at 2:15.
Coordinators. For this, I request Dr. Madhumita Das Gupta to kindly deliver her presentation on AICRP 3 on sandalwood. Over to you, ma'am.
Yeah, thanks, Naushad. Before I actually go into this, I would like to say why the conference was selected as a platform for, showcasing the advancements which was done under the ICRP, because ICFRE felt that, with a plethora of experts from both other countries and from India, they need to know the work what we have done in the past 5 years with the CAMPA funding, and that's how we had actually designed that those NPCs who are present here can talk for 5 minutes about the significant achievements made in the last 5 years. So I think Dr. Geeta had already given a outline of the AICRP projects. So I'll be talking on this project, that is Conservation, Improvement, Management and Promotion of Sandalwood Cultivation in India. This project rather had 8 institutes. We had 4 institutes from the ICFRE umbrella and 4 from the non-ICFRE institutes. And it had 3 objectives. The first objective was to establish a base population, because in sandalwood we do not presently have a population to start the breeding program. To do pan-India surveys and establish a base population with known genetic diversity, which will capture the complete spectrum of genotypic diversity in it. And then the second objective was essentially on agroforestry, that is sandalwood-based agroforestry, which is becoming very popular in the country now, because it is considered as a money-spinning parasite, sandalwood. So a lot of farmers and industries are into agroforestry using sandalwood as one of the species, and this being a parasite, it's very amenable because you can intercrop it with different species which can act as its host. And the third was to document the phenotypic variability in terms of the stable carbon isotopes, oil yield and oil content, and also we had a very targeted focus was to develop a non-destructive method for estimating the hardwood content in sandalwood. That was one of the major objectives in the project. So coming on to what we have done in the past 5 years, we have done pan-India surveys, identified 70 natural populations, 1,000 odd candidate plus trees. 5 base population trials have been established in 3 states, and we have the first sandalwood breeding program has already been developed under this program. Genomic resources, we have vast array of genomic resources. I think there was a booklet available in our institute, in ICFRE Pavilion. We also have a sandalwood GDB, an online interactive portal. Anyone can visit, it is free to access. So you have the genomic and transcriptomic resources of Santalum album in that. Other than that, we have 16 sandalwood-based agroforestry trials which was established in 6 states of India. Now these had known seed sources. So we were seeing how the seed source can affect agroforestry model. Other than that, we had also done a lot of surveys of the existing agroforestry systems in the country to document how the farmers have different silvicultural or agronomic practices and what are the prevalent species they use, the spacings they use, the irrigation patterns they use across different states. So that also around, I think, 236 surveys were done for that. And then we had two components on disease and pest. One of the major pests of sandalwood in plantations, in cultivated condition, is the stem borer. So management practice was developed for this, that is a light trap and entomopathogenic fungicides are very useful in controlling this pest. In terms of disease, the major disease today which is becoming a concern for sandalwood genetic resource is the sandal spike disease, which is caused by your phytoplasma. Till date, we do not have any management practices for it. What the forest department does is they just uproot it and they burn it off. So it's a fatal disease which is spreading across the country. So there was a query that does this phytoplasma move through seeds? It was not known. So this project established that yes, it moves through seeds and seedlings. So care has to be taken when we collect seeds and when we deploy seeds to different parts of the country, because seed is the only mode of propagation in case of sandalwood. So this was a very important finding, which says that we need to be very careful when we when we collect seeds from sandalwood trees. Next is as I said, we had to develop a non-invasive, that is electrical resistivity tomography system. This is a technology which is used for, I think, several applications. In this case, this was used by IWST, Dr. Divakara, I think he has already presented it. Here in a standing tree, it is just, you can nail it and it is like a CT scan. So it gives a very, very clear picture of the heartwood content, which is actually the economic trait in case of sandalwood. So this was established, it was validated extensively, it was extended to farmers and forest departments also. Other than that, as I said, we have also documented in about more than 200 samples the carbon isotope ratio. Now the aim of this was, we know that stable isotopes can be used to used as signatures for location-specific sourcing of material. Usually stable isotopes are used, and carbon isotopes are also used for that. But in case of sandalwood, we could not see that pattern, meaning to say that the carbon isotope ratios could not give a location-specific identity to the wood. So not a very good, probably carbon isotopes are not a good signatures for using in case of sandalwood. So that also we identified. Then coming on to another thing is, the oil yield in sandalwood. Again, an economic trait. We had developed a non-destructive model for standing tree, heartwood determination in standing tree. But for oil yield, we usually require a core sample. And most of the departments or the farmers or the forest departments would not want to give us a core sample because, you may lose the tree, or there could be infections because of extracting the core sample. So we came out again at IWST, we came out with a small protocol where 100 milligram of a wood tissue could be used and the oil yield could be just estimated with UV spectrophotometer. So this was actually correlated with about more than 200 samples with the core wood sample. So we got, I think, about 80 or 82% correlation with the classical method of estimating oil yield and the UV spectrophotometric method, meaning to say that this also can be considered as very minimal invasive method for yield estimation. Other than that, we had also a component of tissue culture, where we know sandalwood tissue culture is difficult because it's a parasite. So when we go for hardening into the primary and secondary hardening, lot of mortality occurs because of its host dependency. So about 3 genotypes were selected and they were successfully, the in vitro protocols were established, and today they are in a demo trial to show that it is possible to have a tissue culture raised sandalwood also. So the trials are I think it's about 6 months old, so we need to see how they perform. So other than that, we had extensive extension activities done. Like I said, the main activities in extension were the popularization of the ERT technology, the disease and pest prevalence in sandalwood, that was very important. That also has been extensively taken forward to the sandalwood growers. And then the agroforestry systems. I mean, what are the agronomic requirements? What are the considerations when we consider a host in sandalwood agroforestry systems? So that also was extended to the stakeholders like forest departments, industries, farmers, and so on, across about 5 institutes in the country. So we have also done about 11 peer-reviewed publications. We have many booklets and pamphlets which has been generated through this project. So now, with this project in place, we are now positioned to take forward, or we are positioned to accelerate the sandalwood breeding program using the concept of genomic selection. So that is what we have proposed in the second phase of sandalwood, to accelerate it using a GS model. Thank you.
Thank you, madam. Thank you for briefly explaining the findings on ACRP3. Now I would request Dr. A. Rajshekaran, National Project Coordinator of ACRP7, to present his findings.
Thank you, Naushad. As part of my team, as National Project Coordinator, I am presenting the significant findings of the ASRP7, Assessment and Monitoring of Invasive Alien Plant Species in India, and formulation of strategies for management of these key invasive alien species in different regions of the country. So in this project also, it's a multi-institutional project. In this ASRP, we've got 8 ICFR institutes are involved, and apart from ICFR institutes, 3 institutions outside ICFR also participated in this 5-year project. So coming to this project, since it is difficult to work on all the species, so we have prioritized 4 invasive species, Lantana cambra, Prosopis juliflora, Mycania micrantha, and Acacia mensi wattle. So all these 4 species, they are widespread, and they are causing a lot of impacts to the native system. So that's why these 4 species were prioritized, and with the objectives, the main objectives of the project include to assess the spatial spread of these invasive alien species in different parts of the country. So for that, we have used satellite mapping as well as modeling software like Maxent were used to predict what are the areas which are vulnerable for these invasive species, and to see the impacts on what impacts these invasive alien species cause to the native plants as well as to the soil physical-chemical properties were assessed. And we also modeled the future distribution of these invasive species in different climate change scenario. So what are the areas which are likely to be vulnerable for invasion for these species? And then we try to see is there any bioprospecting potential is available on these invasive species. And then we also tried the various control methods, both physical, chemical, even biological methods were tried. And lastly, we tried that, so can we have some restoration model for these affected invasive species affected areas. And then based on all our findings, and we have prepared the management practices for managing these invasive species. Coming to the key findings, as I said that we assess the spatial spread of these invasive species using satellite mapping as well as modeling. And our results predict that in the case of Prosopis juliflora, it is widely spread in southern, western, and northern India. In terms of climate change also, Prosopis juliflora is expected to increase its spatial spread in different future climate change scenarios. So that's why we are emphasizing that management of these Prosopis juliflora should account for these climatically vulnerable zones. And in the case of Lantana cambra, we found out that eastern India and parts of Western Ghats are highly vulnerable for this Lantana cambra. And in the case of Mycania micrantha, eastern India and southern India are highly vulnerable. Unfortunately, all these three invasive species are expected to increase their spread in future scenarios. Whereas in the case of wattle, it is the future climatic suitability is expected to decrease. That's what our modeling result shows that. So for managing these invasive species, we need to prioritize areas because since all these species are already highly established, we may not be able to completely eradicate these species. We can only manage these species. So for managing these species, these are the zones which are we need to give more priority. And then we tried different control methods for managing these invasive species. We tried various physical methods, chemical methods, as well as biological control methods for various invasive species. And one breakthrough we got is we have identified one fungus called Lesidioploidea theobroma. It's a fungus and which was isolated from the natural population of this Mycania micrantha found growing in Assam. And then we have isolated and made pure culture, and we have done pathogenicity and host specificity test and all. And then we also observed the disease incident in the treated Mycania micrantha. So this is one of the breakthrough in biological control of this Mycania micrantha. And apart from that, various methods were shortlisted for managing these invasive species. At the same time, we tried various restoration model for these invasive species. If you see that when we go for restoration, we need to account for improving the soil moisture as well as introducing grasses, and at the same time introducing tall saplings as well as application of seed balls and application of biofertilizers are very effective in restoring the invaded areas. And we also try to see what is the bioprospecting potential of this invasive species, and we could, with the help of our team from Institute of Wood Science and Technology, we have standardized the technique for preparing high energy briquettes from Prosophilus juliflora, both from the wood, sawdust, as well as from the twigs. And both these parts has high energy efficient fuel wood capacity, high calorific value products, and which can meet the thermal power plant standards. Actually, there is a policy for mixing this agriculture and waste to the fuel products. So that we have tried, and we also standardize the extraction of natural dye from Acacia munsey bark. So, and based on all our results, we have prepared a standard operating procedure that will be that are being disseminated, as well as we have made 10 publications in the projects which are widely distributed, and 10 impact articles in impact journals were published. And we hope to disseminate our findings to various stakeholders through various extension media, and will be conducting capacity building and training programs for the field staff of State Forest Department. Thank you.
Thank you, sir. Thank you for sharing the findings of ACRP 7. Now I would like to request Dr. R. Yashoda, National Project Coordinator of ACRP 9 on teak to deliver her presentation.
Good afternoon to everyone. This project is on quality plantings, cloning for, quality teak production for capitalizing on cloning. This is mostly, we worked with the different ICFR Institute as partners. this project I can say is an example of transfer of biotechnology to field. Like we were interested in popularizing the clonal plantations of teak, and for teak clonal production is possible, I can say only through tissue culture, because the normal cutting propagation, the probable yield is quite less. So if we want to convert the selected trees as clones, tissue culture is the only option. So we have adopted the technology, and the institute had already developed technology. So under this project, we could easily popularize this technology. And world over, of course, short rotation teak is cultivated. And India, being a native to teak, we were mostly comfortable with the seed-raised plantations. And seeing the variability in the productivity, we shifted to the clonal plantations. And under the project, we have established model plantations mostly in the farmer lands, about 150 hectare area in 11 different states we have established, so that we encourage the farmers to go for clonal plantations of teak. And interim results, like 6-year-old clonal plantation, we could see the height of 10 to 12.8 meter height and girth of 50 to 58 centimeter. We are currently popularizing 5 different clones. These clones actually originated from the tree improvement program of teak conducted by the institute. So we had some background there, so we could easily clone these individuals and go for field testing. And apart from that, in the country, state forest departments are also interested in utilizing the tissue culture technology for large-scale propagation. So that way, through this project, we could help different forest departments to establish their laboratories and standardize the protocol for different species of trees. And we have signed a license agreement with the commercial tissue culture laboratories, because we cannot meet the demand of the entire teak propagules. So there are n number of commercial laboratories are available in the country. They are mostly concentrating on banana cultivation, banana propagation as a cottage industry. So spare space available in those laboratories, there are like half a dozen laboratories are interested in transforming their spare space for these kind of important tree species. So with them, we initially transfer the cultures, we test their capabilities to mass multiply. Once they qualify that, then we license out the clonal material, so that for the institute, they will multiply and supply to the users. And we also disseminate the package of practices, because we know that teak demands a lot of silvicultural interventions. So we also disseminate the practices to the farmers in the local language, as well as the videos, all those things. These are the work carried out under this project. Thank you.
Thank you, madam. Thank you for giving a brief presentation on teak. Now I'd like to request Dr. Manisha Taplial to deliver her presentation on ACRP 10.
Good afternoon, everyone. This AICRP 10 on developing seed testing and seed storage protocols of selected forestry species from diverse forest types was a theme-based AICRP that was in forestry seed science and technology. Under this project, we had 7 out of the 9 ICFR Institute as partners in this project, and the lead institute was Forest Research Institute, Dehradun. And in this, we worked on 78 species from different forest types, ranging from the temperate forests, subtropical forests, to arid zone, to rainforests. and dry deciduous, tropical dry deciduous forests to the Western Ghats area. So there was a diverse forest types which we worked on, the species growing in these areas. So there were many, these 78 species, most of the species, majority of the species, in fact, all the species were native species, and majority of them were there on which hardly any work had been done, especially with respect to seed science and technology. And we knew that the species are distributed there, but then some of the trees, especially in the Western Ghats and the northeast region of the country, were very tall trees, and it was, there were some problems in even collecting the seeds and all, and observing the phenology, like as to when the trees are coming into flowering, and when the fruit set takes place, and then when do they mature, and also these kind of basic information was also not there in many of the species. I mean, little scattered information was there, but proper systematic studies had not been conducted on these species. And the species ranged from, if I talk of the Himalayan region, these were the betulas, there were some of the quercus species were there. Then coming down to the central zone and all, or arid areas, there were Capparis decidua, certain albizzias were there, acacias were there, and then certain species in, some of them were of medicinal value, some of them were of timber, their timber was important, some of them were tree-borne oil seeds and and all of them were trees actually. So we did, the teams did a proper survey in the forest areas for these species, for locating the populations of these species, and then observing the phenology as to when the flowering is taking place, when the fruits are appearing, when they are maturing, and then the fruits were collected, and then the seed collection, like the optimal time of maturity of the fruits and all, that was observed, and that is when the fruits were collected. The seed collection, extraction, and processing was standardized for these out of 78, 75 species, and there were few species like some of the salix species that we couldn't get really much seeds, and all sterile seeds were there, so we couldn't go ahead with any work on the seeds of such species. And then we developed the seed germination protocols. Now, as we know that many of the forestry seeds, they have different kind of dormancy, different levels of dormancy. So that was worked out, whether it was physical dormancy, it was morphological, morphophysiological dormancy and all, and what were the pre-treatments to be given to the seeds to overcome those dormancy, different kinds of dormancy. Those were standardized. So we developed the seed germination protocol for these 75 species, and these protocols will definitely be used for the quality evaluation of the seed lots that will be collected by different stakeholders for raising planting stock in the nursery. Then we also carried out studies on the seed storage physiology of these species. So like whether they were orthodox seeds, like they were not sensitive to desiccation and chilling. They're easy to handle and easy to store also for different periods of time. And whether and some of them were recalcitrant, like they're very sensitive to desiccation and of course to chilling also, that they wouldn't be stored for very long periods. Now again, the recalcitrant seeds were categorized into tropical and temperate recalcitrant seeds. Like temperate recalcitrant seeds, some of the oak species, they were the acorns were sensitive to desiccation, but not to chilling, because that is the cold environments, they grow in the cold environment, so they were tolerant of chilling and all, but not desiccation. So we could not desiccate them and had to be stored as such at ambient room temperatures and all. So when we did this study, so we found out that seeds of 44 species were orthodox, and those of 29 species were recalcitrant, and 2 species were categorized as the seeds were intermediate. Then we also did systematic seed storage studies for different on all the seeds of all the species, and we could see that the seeds of seeds which were orthodox of 15 species, we could store them successfully for over 2 years, and after 2 years also the seed retained above 50% germinability. Whereas there were some species which the seeds did not retain viability for even a month or so, so there were recalcitrant seeds. So this kind of reports are also were done for the first time for some of the species. Then there was also a model was developed for correlating seed traits and habitat with desiccation sensitivity for the seeds of different species. These were mostly for the species hailing from Western Ghat area. Then we also developed the nursery techniques for few species like Anogesis latifolia, Boswellia serrata, Prunus cerosoides, Betula utilis, Salvadora persica, Capparis decidu, and Sorbus lanata, which will be, of course, these were during the capacity building programs. These nursery techniques were extended to different stakeholders, especially the forest department, who would utilize these for raising the quality planting stock of the species in the nursery. Then, as we know that whatever research work we do, the findings have to go to the stakeholders, that they have to have the applied aspect, and for that we conducted about 20 training and awareness programs on seed and nursery technology, quality seed production, even seed ball preparation, and conservation of rare, endangered, and threatened species for the frontline staff of different forest department, many states in India like northern, central, southern, and northeast regions of the country also, and also to the officer trainees of Central Academy for State Forest Service, and farmers also, self-help groups, and students also. 80 brochures and leaflets were published in bilingual format, like one was in English and of course other was in the regional language on forestry species, and they were distributed to different stakeholders to get the benefit from all the work that had been done. And training manuals were prepared, forest seed identification guide, lab manuals, and we also published 9 papers in peer-reviewed journals and presented the findings in conferences as well. So these are some photographs. Thank you.
Thank you, Madam, for the presentation. Now I request Dr. C. Bhuvaneshwaran, National Project Coordinator, ASRP 18, to present his findings.
Very good afternoon to all. This is the project falling in the theme silviculture. Here in this project, 4 ICFR Institute and 1 non-ICFR Institute, that is Forest College and Research Institute, were involved. So basically the objectives were to assess the productivity and carbon sequestration potential of important species, particularly in farmland condition. Around 526 plantations were surveyed across 5 states. And then we gathered the data, and the basic objective of benchmarking of productivity and carbon sequestration potential has been done under this project. And I'm presenting here a very interesting result before you. One is that regarding teak cultivation. So the normal suggested practice is that teak has to be cultivated on closer spaces as it is a light demanding species. Later, it is suggested to go for thinning, 2 mechanical thinning, and that way the final spacing will be 4 into 4 meter. But now, the present scenario, we have moved from seedling based teak to clonal based teak. So tissue culture teak now we are promoting. So in this context, we wanted to avoid deploying the tissue culture teak, which is a costly thing. So the short rotation crop, casuarina, and nitrogen fixing, and another advantage it has. So we wanted to test the mixing of teak and casuarina so that the thinning is avoided. Two mechanical thinning is avoided, and planting stock requirement is very much reduced. So only 25% of teak is sufficient, planting stock is sufficient for cultivation of teak. So our initial results were very, very encouraging. So we can recommend that this method, this model can be adopted. And more importantly, what we observed in the third year, the growth, besides the greater growth obtained, there is a more number of bigger sized teak stems we have recorded. So that indicates that there is a complementing impact of cultivating casuarina with teak. And then we benchmark the productivity, and very interesting observation is that teak requires very specialized site condition, very specific site conditions. That's what we got from the data. We analyzed the productivity across different agroclimatic zones. Very glaringly it was reflected both in block planting as well as in boundary planting. So the specific site conditions required. So we have to look for a suitable site condition for tea cultivation. And not only the growth pattern, and heartwood formation also got influenced by site conditions. That's another very, very important points here. So the heartwood formation, both in block plantation and in boundary plantation, the same results obtained. That means to say that the site conditions highly influencing the heartwood formation in teak. And carbon sequestration potential we assessed, and what we understand is that the carbon sequestration is very, very minimal in range. That means to say that the cost per carbon unit, what is in the market, will not be that much economically viable. So we recommended that there can be a reconsideration on the cost part of this carbon credit. And the way forward from these studies are that as all the plantations are now seedling based, and there's a difficulty in arriving at relating the silvicultural inputs and the species performance. And now we are in the time of deploying clones. So clone-specific silviculture has to be the future program. And this advantage of mixing nitrogen fixing trees with non-nitrogen fixing trees. There are a number of reports, literatures available from Western countries like eucalyptus and acacia, sandal and dalbergia in China. So a number of in-depth studies are available in other countries, but in Indian condition, this particular aspect of mixed plantation of nitrogen and non-nitrogen fixing trees can be explored, and the models can be developed. And heartwood formation, there are more clarity, and which silvicultural intervention will be favoring heartwood formation. That's a lot of studies required. With that, I conclude my presentation. Thank you very much.
Thank you, sir. Thank you for that brief presentation. Now I request Dr. N. Senthil Kumar to present his findings on ACRP 20.
So very good afternoon. This is Senthil Kumar from Institute of Forest Genetics at Tree Breeding, an entomologist. This is yet another ASRP project based on themes, development of biopesticides. IFGTB is pioneering in developing marketable biopesticides with the collaboration of industrial partner funded by BioRac DBT. The learning experience is in order to develop marketable biopesticides, we got to register in the Central Insecticide Board and Registration Committee. There are 3 different criteria has to be followed in order to register in the Central Insecticide Board and Registration Committee. The first one is chemistry part of it. Whether it is a chemical insecticide or a biological origin, we need to have a chemistry part of it. That is, what are the major active ingredients of insecticidal potential, followed by we have to have a multi-locational trial with multi-crops, multi-insects, and multi-regional evaluation. The third one is toxicological part. So that is why we get into this project to fulfill the first 2 criteria in this project. So there are about 7 institutes involved in this project. And then why we get into this tree-borne oil-based, seed oil-based biopesticide in the sense. So if you go to Central Insecticide Board and Registration Committee website, there are about 950 insecticides are registered, of which 448 are biopesticides. And most of them are microbial-based biopesticide because it is very host-specific. There is about single-digit number is available for tree-borne seed oil or oil-based biopesticides. That is why we are getting into tree-borne oil-based biopesticides. So we have already developed 2 biopesticides, and we have gone into multi-locational trial with the different institutes across the country, and it has found very good efficacy. And then, as I told, that these are the 10 different tree species and their insect part that we have selected and evaluated over it. And then these are the 10 different tree-borne seed oil. It is non-edible tree-borne seed oil for developing into a biopesticides that we have. So as I told that as a chemistry part of it, we have identified number of active ingredients which are having the insecticidal potential, made it into a formulation thereof. And then we have gone into multi-locational trial with the different insects and different crops and different regions of the country. And we have also arrived at the high efficacy of these 10 different tree-borne seed oil, and then we have shortlisted to 3 formulations. And the 3 formulations we have evaluated across the country, so it needs to go for toxicological studies. That is why we have gone into follow-up studies, a follow-up project of ASRP 20. And we have also gone into toxicological, little bit preliminary studies on these parasitoids as well as the honeybee. So it found that the 3 formulations is good enough to go for further toxicological studies to make it into market available biopesticides and registered with the Central Insecticide Board. That is what we are going into follow-up projects. With this one, I close this, and thanks for the opportunity. Thank you very much.
Thank you, sir. Thank you for sharing the findings of the project. Now I'd like to request Dr. A. Karthikeyan to deliver his presentation on ASRP 21.
Good afternoon to everyone. This project is under CAMPA funded project. The title is Development of Superior Biofertilizers for Enhanced Plant Productivity. Including IFGTB, 6 institutes took part in this project. The objectives are very simple. One is development of superior biofertilizer and their efficacy to test. Second one is whether the biofertilizers are controlling the pathogens or not. So To control the pathogens, the biofertilizers are used in the second objectives. And the third one is the outplant survival. The biofertilizers, inoculated plants, how it is performing. That is the third objective. And fourth is the training cum demonstration of biofertilizers. According to these objectives, 5 biofertilizers were developed. One is arbuscular mycorrhizal fungi, and which is transporting the phosphorus from soil to plants. Another one is nitrogen fixing bacteria, Azospirillum, and another one is another nitrogen fixing bacteria, Azotobacter. Then phosphate solubilizing bacteria that solubilize the phosphorus from the soil. And another one is potassium mobilizing bacteria, that is Fractoria aurantia, that is one bacteria that mobilizing the potassium in soil for easily taken up by the plants. These biofertilizers were developed, and accordingly these biofertilizers were inoculated in the nursery in the selected species like Santalum album, Melina arborea, Dalbergia shushu, and Melia dubia. And the performance also assessed in the nursery level. Later, it was transplanted to field conditions, particularly farmer's field. The fields are unfit for cultivation. In that field only we have selected and tried to establish these seedlings along with biofertilizers. The performance very wonderful in the field conditions, and we, across the country, the biofertilizers were used in these selected tree species, like 10 hectare area of plantation was done in this project. And we come to the conclude that the AM fungi, PSB, KSB in the combined consortium gives better results. So that consortia was formulated as a product. It's a product and named as bioreclamair, and it is released under this project, and we are supplying to the farmers and the stakeholders like State Forest Department with minimal cost. So this is the main achievement, and accordingly 8 trainings were conducted for the stakeholders about the biofertilizers, and 8 publications including 3 international journals were published, and we have successfully completed this project with the support of IC for IFGTB and IC for Dehradun. Thank you very much.
Thank you, sir, for the presentation. Now I would like to request Dr. Fatima Shirin to present her findings on AACRP 23.
Thank you, Naushad. Good afternoon, everyone. The All India Coordinated Research Project, which I am going to present, is Genetic Improvement and Value Addition of Madhuca longifolia. It is one of the species-based AICRPs, and it was conducted in 6 institutes of ICFRE throughout the distribution range of this species. Madhuca longifolia, which is commonly known as mahua in India, it is a very important non-timber species with medicinal values, and it is also a tree-borne oil seed. And the importance of the species is because it is very useful, and it is the livelihood of the tribal communities and the local communities depend on this species, most especially in central India and eastern part of India. And The other parts are also very useful, like the seed oil is extracted from the seeds, and this oil is also edible and also used for various purposes. So it's a very important tree in all. This species was taken up for doing work on a systematic manner. Earlier works on genetic improvement of this species has not been attempted in a very organized and systematic manner. Sporadic works here and there are there. So, because the reason being that it is a very long gestation species and also very slow to grow, that's why this genetic improvement work has not been attempted earlier. So, in this All India coordinated project, genetic improvement of this species was attempted in a planned manner, and as it is a species of local importance, production of some products was also attempted in this species. This project was carried out under 12 states in the country, and there were around 7 objectives of this project, starting from the selection of survey and selection of candidate plus trees and their morphological, biochemical, and molecular characterization, then studies on their natural regeneration and production of some quality planting material, and then production of some value-added products, and then establishment of vegetative multiplication gardens, seedling seed orchards, and the last objective was to document the indigenous traditional knowledge related to this species. So with these 7 objectives, the project was carried out. So in the poster, is it on? Okay. The poster, you can see the achievements. A total of 1,152 candidate plus trees were selected in the 12 states. And when we have done the biochemical characterization, we found a lot of variation in the individual trees as well as in the population, with a range of 23 to 94% in sugar. And similarly, in oils also, we had a lot of variation. And we have recorded natural regeneration in 22 locations of the country. And we found that seedlings and saplings were very few. Only the mature trees were present in the population. Because the natural regeneration is problematic in this species, when the flowers collection takes place, the people burn the floor of the forest, therefore the regeneration doesn't take place. And also because the seed oil is edible, the people collect the seeds from the tree itself, so regeneration is affected. Then we have produced 4 value-added products in this project: Mahua chocolates, Mahua hand wash, antibacterial hand wash, and wound healing cream, and a tooth powder. And 2 products have been filed for patent: Mahua chocolates and the tooth powder under this project. And this is the map which shows the areas where survey was done, carried out, and CPDs were selected. And now coming to the genetic diversity studies, SSR primers were used for genetic diversity studies. And one very important achievement in this project was the standardization of the cleft grafting techniques, so that we are able to produce vegetative propagules and put them into the field as VMGs, vegetative multiplication gardens. The advantage of the vegetative multiplication garden being that we are expecting flowering and fruiting in around 4 to 5 years instead of 10 to 12 years in the natural populations. So this cleft grafting was standardized in this project, and we were able to establish 7 vegetative multiplication gardens throughout the institutes and throughout the country by the different institutes. And in addition to that, we have established 3 seedling seed orchards and 1 seed source trial also in different locations. And 2 fungus has been genetic sequences of 2 fungus have also been deposited in the NCBI database. And as it is a very important species for the tribal communities, we went to them and we have documented the indigenous knowledge which they have about this species in the form of a technical booklet. And in addition to that, other extension activities also we have carried out in the form of trainings and peer-reviewed papers, 11 peer-reviewed papers, and around 12 booklets and 4 leaflets. So these were the findings of this project. Thank you very much.
Thank you, Madam. Now I would like to request Dr. B. Nagarajan to present his findings on ACRP 15.
Good afternoon, everyone. Tamarind is an exceptional species, particularly in an era of climate change, because it shows enormous adaptation across different countries completely. It can be traced up from Africa through to Southeast Asia. So one of the most interesting things what the future lies is we have an hypothesis to say that those trees with compound leaves and higher drought resistance, they are the trees which could be of much use in changing agronomic conditions. For instance, this experiment was very interesting. We started selections from 8-degree northern latitude, and we moved in up to 26-degree latitudes. And most importantly, tamarind is a very, very important component of food to not only India, Southeast Asia as well. In India, the peninsula states have it in its dietary requirements. So the results are like this. We started the work somewhere in 1996, and well, a few interesting things is that despite it is being a leguminous tree, it doesn't fix nitrogen. So it is one of the very few trees, despite not having nitrogen fixation ability, it grows in very, very tough conditions. So it's a future climate resilient, perhaps a mitigatory tool in very tough sites, which we need to think about. So about 20 selections, we have pushed it across multi-location trials. And the answers are, we have got some 4 varieties, which we consider pan-Indian, and it can be used. So in the process, we did understand some genetics part of it. For instance, in any given selection, the onset of flowering is genetics, but the extension of flowering is ecological. So based on this, we have all these 4 clones which have been selected have very long flowering duration. For instance, normally flowering in tamarind is restricted to 30 to 40 days. And here we have found a phenomenon that these selections, what we have made, extends up to 60 days. And this is a self-incompatible tree crop. We made a small mistake in tamarind improvement 20 years back, which we had an option to correct here in this particular project. For instance, some of the wolf trees, which showed very heavy productivity, were selected, and they were cloned and put it in the same plot. Eventually, what happened was it was all monoclonal planting, and it ended up in self-incompatibility. No fruiting was found at all. So here we have made it a point that these are multi-clonal selections from different families we have developed it, and there is no mismatch of reproduction. So based on that, there are 3 phenotypes which we recognize among these selection. One is a red phenotype, which is very rare, rich in anthocyanin. The second thing is a sour phenotype, which is used in the food industry. And the third thing is a sweet phenotype, which is largely found in Southeast Asia. For the first time, we are reporting in and around India. So which means to say that that phenotype, which was normally thought process. Now, yet another interesting term in terms of forest genetics is that this is not a species where you can find defined provenances. It is a naturalized species. During the paleo migration of Africa, this moved into India. So very interestingly, one of the most important genetic diversity hotspot is Gujarat, which was the last bit of portion which got separated from Africa. So in that way, we find that there is a peninsular population, and then there is a northwestern population. So these are the 2, perhaps not provenances exactly. So end of it, we find farmers going for it. Very recently, the United States, there has been a variety which has been developed, which is called Diana. And in Florida, they have started it. And I'm very glad to say that this is largely concerned with tribal lives. Till date, India happens to be one of the largest, India is the largest tamarind exporting country, as well as consuming country as well as. So we have encouraged a set of young engineering and biological graduates from tribal communities, and we have got a company registration, a company which will be exporting tamarind from this diversity. A part of the diversity will be moving across the world, particularly to the Indian diaspora. That's the thought process. So the startup is to start. So end of it, a species which is known to show an onset of flowering as a seedling at the end of 10 years, we are able to get flowering as early as 2.5 to 3 years. In short, until recently, the country didn't have selected varieties of tamarind with the pedigree, and currently we have a pedigree established, and there are 4 varieties, prospective varieties we have shortlisted. Thank you very much.
Thank you, sir. So we have ended the session on presentations on AACRPs. So the floor is open for questions. If anybody have some questions, we can discuss briefly.
My question is to Bhuvaneshwaran. Bhuvan, one small doubt, clarification. See, my experience with mixing species, Castorina and other species is, Castorina has got a definite allelopathic effect. The leaflets that are shed really prevent even small grass from growing. So you have tested 1:1. Would it be nice to test 3:1? 3 teak is to 1 Casuarina? Because I didn't find any advantage in growth compared to the pure slight advantage. What you see now may not show up later also. Teak is a... Yeah. Yeah. No, 3 teak is to 1 Casuarina. Did you try that?
The harvest period goes beyond 3 years.
Oh.
Now, what we are proposing is that plot we have harvested Casuarinas. So that way.
Another thing is when you plant Castorina, Castorina takes off faster, no? Growth
Yes.
is much faster, no?
Yes, sir. Yes, sir.
So how does that come affect
A
it?
very interesting question, sir. We have tested the clonal effects. Sir. Suppose if we plant CH5 and teak together, that Casuarina will dominate teak. I will not, for time constraint, I will not present it in details. And our Maracanaum clone, that is what we observed in the field. That sir also may be knowing,
Yeah,
sir.
yeah, I know.
It is a initially slow grower.
Okay.
Maracanaum
Oh,
clone.
that you tried, is it?
Yes, sir. Yes, sir. That
But
is
then,
a very
yeah.
interesting observation, sir. It is
Yeah.
not that clone specific. So, very clone specific silviculture. Now we are moving towards that. So, what is the clone of Casuarina? What is the clonal material? That way we have to now further proceed.
Yeah. Another small doubt is,
Sir.
did you test the root system of tissue culture teak versus
Dr. Yashodhan.
Actually, in our study, we have not tested the root system, but there are literature available from different countries. They showed the root system of clonal teak as well as a seedling teak. Rather, in the clonal teak, the secondary and tertiary root system is well developed. Good anchorage is there. Should not be a problem.
So any more questions? So,
I have a question about the Sandal Good project. I don't know if Mohimita is in the room. Well, it's okay, she's not here, I can ask to her later. So. Or maybe someone else in the room will know. So, it's okay. Okay. So, I wanted to ask if there is other. I found the genomic project on Sandalwood very comprehensive and very complete and excellent. So, but I was wondering if there is other countries working with other species of sandalwood, and if you are connected with them and if they are doing similar things. So, but I don't know if someone. Okay, yeah, we'll do my question later. It's not a problem. Eh? No, there is nobody. Okay, thank you so much.
any other questions? So we'll wrap up the session. Now we have exhibitor presentation. So for that, I now request Shri Trideep Chakravarti, General Manager, Chromosome Labs, to deliver his presentation.
So, very good evening, everyone. So, myself, Trideep Chakravarti, General Manager Sales, Chromosome Labs. We are a lab in Mumbai, and we are doing NGS, Next Gen Sequencing Services provider, using Oxford Nanopore, Illumina, San Diego-based companies which have their sequencers, and we have those sequencers installed in our labs, and we are NABL, ISO, DSIR certified labs. So let me give you a brief introduction. So our lab has been found around 6 years back in Mumbai, Jogeshwari, and we have, it's the ARTOF facility where we have NovaSeq, NextSeq, MySeq, and Oxford Nanopore. So we have different read lengths of sequencing, like from 2 into 150 base pairs, 2 into 50, 2 into 300, as per your requirements and different services which will be you guys are using in your research work. We facilitate the researchers to those services at our end. So going forward, so we are a group of medical coders. We are a group of researchers. Our scientific team is from MIT, and Dr. Gautam Dasgupta and Dr. Nandini Dasgupta. Those people are there. They have been giving inputs in this industry from last 15-20 years. They did their PhDs from ISC Bangalore, then they moved to MIT for the postdoc, and they came back in India and they started this company. So we are located, as I told you, in Mumbai. So we are a one-stop testing point for all the things. We have genomic research, we have clinical genomics, we have pathology labs, around 10 pathology labs across Mumbai. We have molecular diagnostic, histopathology, microbiology, and radiology upcoming. We are about to set up a radiology lab also. We have NABL accreditations, ISO certified lab, and 8 labs high throughput sequencing facility is there in Mumbai, and 2 collection centers.
So we do, as I told you, we do all kind of sequencing services, right from antibacterial resistance, drug, pharmacogenomics, metatranscriptomics, whole genome sequencing. As your area of interest will be SNP genotyping, SNPs, copy number variations, whole genome sequencing, or low-pass genome sequencing of a novel genome, plant genome in which you are working. So we have a facility for long reads and short reads both. So I will go through one by one what all we are doing. So this is a basic NGS workflow where we are taking the genomic DNA or RNA from our clients, and we are preparing the NGS library. After preparing the NGS library, we are sequencing on the NGS sequencing platform. Then the raw data goes to the bioinformatician. So bioinformatician takes out the meaningful information from that raw data, and they give you all the analytic reports. So this is the design structure, how it comes. First, we discuss with the customer and we understand the ethical and regulatory compliance required for handling their samples. Some customers like biopharma accounts or seed companies which we work, they sign an NDA with us for non-disclosure things so that we should not disclose their research with other people. So then after submitting that, they let us know what are the sample types, what are the controls and test groups which they are working on. Then different sample types, right from bacteria, viruses, algae, cultures, then plant tissues, root tissues, soil, water. We are working with human samples also, blood samples, FFP blocks for cancer oncology samples. All samples we are processing in our lab. Right from any living organism, name it, we are working with them, and we are giving all different kind of services across pan-India and internationally. So this is the workflow, how it works. Like first the pre-sales, like the sales team, or we generate the leads. And then after generating that, we does a client meeting, and after that a technical team arrange a Google meeting with you guys, scientists, and understand the scope, what is your project is about, what is the feasibility and the commercial. Then the quotation goes. Once the quotation goes, you send us the purchase order, and according to the purchase order, we start preparing the samples and start initiating. Entire logistic work from your lab till our lab in Mumbai is taken care by us. We ship the samples in dry ice using our logistic team and gel packs for the DNA. And for RNAs, we use for dry ice pickups. So not only doing the work, what we also support the customers is that we support them till the end of publication. Even the reviewers, whatever questions they arise, these points you need to work it out more. So we work on those reviewers' point, and we do the reanalysis and give you the appropriate results so that you can publish it in your journals, and we keep your data for 1 year at least. After that, we take a permission from you and we discard, because we need to free our storage. Okay. So this is our lab, how it looks like. Nova 66000 Illumina based. This has a capacity to generate around 3 terabyte of the data in one run. Then we have NextSeq 2000, where we can generate 540 GB of data in one run. Then you can see Oxford Nanopore, that is long read sequencing platform, which can generate 100 GB data of a complete one human whole genome or plant genome, and bacterial around 100 samples we can process in one go. So now coming to the different service as based of the nucleic acids, like RNA-based projects, what we are delivering is that RNA-based whole transcriptome, where we are sequencing mRNA plus long non-coding RNA, LNC RNA. So people who are interested in seeing the mRNA plus long non-coding RNA, we do whole transcriptome sequencing, where we ribodeplete the rRNA of your sample, and we find out all the coding and non-coding RNAs. Then we are doing a de novo RNA-seq when the species is not known, its database, its genome is not there, or transcriptome is not there in NCBI database. So what we do is that we go forward with a de novo approach for a RNA-seq.
So this is the thing where people are working for something new, novel thing. So for that, de novo approach is there, and different pipelines, bioinformatic pipelines are there for de novo RNA-seq. Then comes reference-based RNA-seq. Reference-based, as you are aware, reference is there, genome is well-defined, genome is there, we have a transcriptome, we do the sequencing, and then we map it to the transcriptome of that, and we find out all the gene regulations, differential gene expressions, and we give you all those findings from your samples in a report. Then coming circular RNA we are also doing smaller RNAs like microRNA, miRNAs, novel RNAs, tRNAs, all smaller RNAs also we are working on. Then long non-coding, as I told you, we are also working long non-coding RNA. Now coming DNA-based. DNA-based services like viral whole genome sequencing, reference-based genome assembly of all the bacteria, virus, fungi, plant, human, etc., anything. You give us the DNA samples, or you even given us the tissue or culture sample, we'll isolate the DNA and we will do this service for you. Then we are also doing plasmid whole genome sequencing, complete length plasmid, whole de novo genome sequencing, GBS, genotyping by sequencing. This is the area of interest where you guys will be interested upon to see the parent lines and the progeny, how they behave and what all the SNPs are there, SNPs are there. So genotyping by sequencing, we are doing using NGS. We also have a facility for SNP genotyping using Thermo Fisher Gene Titan, where we have certain arrays which are well-defined array, 10K, 50K, cotton, rice, different crop-based arrays are there. If you want to find some, your trait-based analysis you want to do, you can give those orders also to us. We can discuss over it. And then we have PCR-based product sequencing, where people are doing some targeted analysis, certain genes they want to target, and they want to understand what is their role. So we are doing a PCR product-based sequencing also, mitochondrial DNA sequencing, and hybrid genome sequencing using ONT plus Illumina. Hybrid means we are sequencing the genome using ONT, Oxford Nanopore, and Illumina. Then the bioinformatician sits with both the data, long read and short read, and they try to join the short reads with the long reads. So long reads you will be getting as around 5 to 7 to 10 to 21 KB bigger reads. Then these short reads of Illumina join the genome, and you can get a complete picture of the genome of a new novel species. Now, coming to the metagenomics segment, where we are doing shotgun metagenomics, where we are doing any environmental sample, right from water, sludge, you can give a stool or whatever it is, we are trying to get the DNA out of it, and we are going to find what all microbial diversity present in that sample, right from bacteria, viruses, archaebacteria, everything will be find it out and listed out, and their abundance with the class, phylum, everything will be there. Now coming to next segment, that is 16S V3 V4, which is Illumina's profile, where this is the targeted sequencing for V3 and V4 region, where maximum bacterial diversities are present. So we find it out. We find out what all bacteria are present. So these kind of projects we are doing for human, for like stool samples, saliva sample, gut samples. Then also we are doing for dental samples. Some dental colleges also, they give us the project for finding the microbial diversity. So like this way we have been doing this from last 6 years. We have a consortium of around 5,000 to 6,000 samples of human gut. So we have the database where later of this project, PPT, I will show you, we have a product into the market for human gut assessment. So then we are into like fungal ITS region screening, then 16S V1 to V9 using ONT, entire bacterial variable region. So then epigenetics also we are doing like human whole genome, whole bisulfite sequencing, MIDI chip sequencing, cut and run, DAM-seq, RIP-seq. Then in computation also we have a good computation facility. So if you have a raw data which you have sequenced earlier, you can give us the raw data. We can do a analysis and give you the report. Then coming to long reads, we have Oxford Nanopore Prometheon, which has a capability to run at least 100 GB in one single run, where we are doing tRNA sequencing, mRNA sequencing, whole genome, plasmid, phase, amplicon sequencing, 16S and shotgun. Now, these are the bioinformatic projects which we have delivered till date. So customers are coming up with new requirements. We have developed the pipelines. So if whatever you are seeing here is the pipelines which we have set pipelines which are developed by our coders. We have certain paid software, some of the open resources which we have using Linux softwares and using Python coding. So we do a taxonomy profiling, hybrid genome assembly, de novo assembly, then reference-based genome assembly, metagenomics, comparative genomics between two different strains or two different species, then population genomics of human or mouse. Then we are doing epigenetics data analysis, de novo transcriptomics, reference-based transcriptomics, splice variants. People who are working on cancer, oncology, they are interested in splice variants, long non-coding RNAs, small RNAs, and RNA modification analysis, modified RNAs. Okay, then genotyping by synthesis, and as I told you, SNP genotyping. Now, these are the clinical genomics. This is another vertical of our company where we are working with human reporting, clinical reporting, where we are doing a human whole exome sequencing, where people who are suffering from genetic disorders or certain things. So we do a whole exome sequencing, and we let you know what all variants are there in your body, which all genes are mutated. Accordingly, the treatment can be given. So we are linked with many of the hospitals across India, and we are delivering this service, whole exome sequencing. Then HPV and STI detections, HLA typing for people who are going for a organ transplant. They want to do a HLA typing profiling, so that facility is also there. Then lung cancer NGS panels are there. And upcoming, we are planning a comprehensive genome profiling. We are working on our R&D to launch a profile for comprehensive genome, where we will have a set of around 100 cancer-specific genes which are responsible, and they are there. So we are making a lab preparation. We are making a kit. So that we will be launching very soon. Now, this is a thing which I was talking about, our other company, Gut Microbiome. So there's a product inside the market which is available. You can go to the website and see Microbiome Research, where what we do is that we collect the stool sample of a healthy human being or a human being who is suffering from bowel disorders or mood swings, anything. We have seen that Alzheimer patients also can be, well treated if they do a proper gut microbiome analysis and change their lifestyle. So what we do is that we collect the stool sample from the patient, and then we take the samples to our lab, and we isolate the DNA, and then we do a metagenomics, 16S, V3, V4 using Illumina platform, and we find out the different bacteria which are there inside your body. Like most abundant bacteria who are favorable bacteria are there, who are pathogenic bacteria is there, opportunistic bacteria or probiotic bacteria. You will get a ratio, how much percentage of these bacteria are present in your body, and if your probiotic bacteria are less, how to increase those bacteria. So what food chain, what diet chain, or what you should eat or what you should not eat, that our dietitian our nutritionist will let you know during the counseling session. So like this way, we are doing a proper counseling session with the patient for diet and nutrition, and then we are proposing them the probiotics, what they should take, and what all the essential things. Sometimes you will get surprised to know that sometimes some people think curd is very good for everybody gut, but once I analyze your gut, you will see that your gut doesn't respond positively to curd or certain eggs or something. You will get to know which things your body is not liking it. They are saying do not eat me, but we cannot in a day-to-day life, we will not able to understand. So analyzing your gut is very important, and understanding how to improve your gut. So these are like samples which we have received around last year. We received around 3,000 samples plus. Now this year till January 19, we have received around 6,700 samples we have processed across pan-India. For any inquiry, you can touch base me. My mail ID is there. I will keep my cards here, so you can contact me anytime for any kind of your requirements. And Y Chromosome Labs, we deliver high quality data. using our Illumina and Oxford Nanopore technology, and we are economic prices. Our pricings are very economic. Whatever we give, pricings are from the market. Other service provider is much competitive, and we have the in-house facility, which I told you. I showed you the facility we have. This is ISO, NABL, DSIR certified lab, and we maintain a faster TAT. Any project which you give us, we finish it in 4 weeks' time. That is 1 month. We deliver to you with reports. So this is one of the best TAT which you can get in this industry. And for anything, you can please contact me. And thanks a lot for giving me opportunity to talk here. Thanks a lot, Dr. Madhumita, for giving me opportunity here. Thanks a lot. Bye. Good.
Thank you, sir. Thank you for the nice presentation. Now I would like to request Shri Manikya Tapagaundar, founder chairman of MAC India Limited, to kindly deliver his address.
Good evening, Aishullah. Ladies and gentlemen, I've been advised by Colonel Joy Gupta to talk only relevant to this subject because international delegates are there. Thank you for reminding me. I will say I am here to sell myself. Of course, my body only belongs to me. I am belonging to somebody else and upstairs. But I'm going to sell my ideas and my research findings. I'm just 76-year-old. I think God has been guiding me all the time to help people to help themselves. And after my retirement at the age of 65, I started thinking about using my rest of my life for the betterment of living beings, be it human beings or birds or anything. And I did a lot of research. One of the findings is the forest. When I was in Memphis, Tennessee, adjacent county was DeSoto and city of Olive Branch. My manager there took me to a plot, 11-acre plot, and he showed me a tree, 1,100 plus years old tree. Mac, we are going to cut this tree. DeSoto County is going to cut this tree because they are developing residential complex. You must save it. I said, drive me to the mayor. And we went there. What do you want to stop this? I want $50,000. I wrote a check right there. I bought that property, still there. And I put a board, it's 1,100 year old. Still, I don't know the actual proof, but local county says 1,100 year old. And I measured the diameter is about 16 feet and 60 feet trunk. All branches above that. And after that, it came in the newspaper, local newspaper and all. And people from colleges, researchers came to me and they said they want to take samples and do research. Because even the tree name is not known. And any fruits given, no, some flowers drop in. And I don't know what is you can, whatever the dropping is there, you take it. I will not allow you even to cut a trunk. If you do that, I'll cut your leg and cut your hand. I put a board there, it is still there. This is the kind of built-in qualities I have saved, I have created, I have planted a lot of trees. I don't boast myself, it doesn't come in the newspaper, but this is what I have been doing. And the research I did, why God has given me that kind of an instinct. is because forest gives everything for the living being. If you have good forest, you get rain. Rain is everything for us, water. Whether it be groundwater or river water or lake water, all those are useful for drinking or irrigation. Drinking for not only for our human life, but every living being. Then the forest produces all kinds of, you already know. But I thought I should do some meaningful research. I went into the forest. When you go to Ooty, some 3-4 decades back, they brought in eucalyptus trees from Australia and planted there. It's all tall, high, big trees. What is the usefulness, excepting the timber value? It's good for making paper. It has got no other use, I found out. And no living being, its own, the sapling cannot grow under the tree. What a big mistakes we did years before. I want the forest department to create trees and plants that are very, very useful for all the living beings, produce fruits, produce pest control products, organic pest control products, like your neem tree and so on. There are several trees like that. They are very, very good for organic farming. Then you have the leaves. They are very good for, as a basil, adiuram we call. It's fertilizer, very good fertilizer. Imagine, that is the potential we have from the forest. And I read about the rules in the forest, in Indian constitution. They are all vested with huge powers. They can shoot anybody down inside the forest. Nobody will ask, but nobody does it. But that is the type of protection we have given in the constitution, and it's not being enforced. People are freely going inside and cutting the trees, taking it away, selling it, all that is happening there. And I estimated over 40% of the forest area is under threat, or it's already, all the trees have been cut, it's empty. Let me not talk the statistics which you already know, but what I want to do is grow forest everywhere. So I thought of bringing in a rule by the forest department. I did discuss with madam yesterday, DG madam yesterday, and I briefed her about the possibility of making a rule. Every empty land, let it belong to forest department or to railway or to highway or to individuals or local government, any barren land must be grown with trees or do agriculture, no problem, or pay rupees 20,000, which is about $200 per acre per year. That's a penalty they have to pay. I believe if it is implemented, and why I don't see any reason why this rule can't be enforced, because considering the benefit for the humanity, it should be implemented. This is what I have advocated. And how that is possible, practically possible also, I written rules. In India, there are several unemployment opportunities, particularly for the graduates. And the graduates, everybody wants to become a software engineer today. And now slowly they are realizing everybody will not get desktop jobs. So people will have to go out and work and make their livelihood. I estimated about 100 lakhs or 10 million unemployed youth could be employed in just allocating some 100 acres, 200 acres land and making them to plant and protect it at least 5-6 years, so that the cattle going around will not eat that. And supposing if an individual owning 300 acres feels, I can't pay the fine, but I'm ready to plant trees, but I don't have time. Okay, give 5,000 rupees per acre, allow this unemployed youth to plant trees for you, grow for 10 years. Every year you pay 5,000 per acre, which they can afford. All those people who invested in it's a kind of a real estate market. If they are not able to afford, you pay 5,000 rupees. I have talked to several people, they agreed. So they will give money, and this unemployed youth can be employed under the control of the forest authority. Existing rule, I don't know forest authority what they can do, but if required, they can form an authority and make sure all the empty land. And I talked to our friends in railway, they are ready. There are several thousands and thousands of acres empty land available across the railway track area. Everything should be populated. If that is done in another 10 years, we can't stop the rain. That will be the, and we can get enough revenue, $1 trillion. What kind of a revenue? I mean, you can imagine, I don't have to, because you're all associated with Forest Department. What kind of revenue? If you want, you can ask me, I'll send you my working. Trillions. After 10 years, you will easily earn $1 trillion out of the income from that forest resources. So, having said all this, I have also worked and found solutions for all pollution. You name it, sewage water, zero sewage water, right at the when it's coming out of the toilet, we have our bacteria living there in a container, it eats. It's not a magic. We implemented, DRDO and myself implemented this technology in DRDO Bhavan, Delhi. More than 32 years, still 34 years, still the tank has not been evacuated. This is the technology we have. Why we have not taken all across the country, all across the globe? I think God has been waiting for me to come in and implement. I'm doing it already. We are doing it. And one day, all these youngsters will see one day, maybe another 10 years' time, there won't be any sewage water all across the country in India and all across the globe. I'm ready to give the technology free of cost. So you can take it all across the globe. That is one thing. The second major pollution is our solid waste. And I have seen in Delhi, I have seen in Texas, Dallas, huge dump yards and producing methane gases, not able to track methane gases. Huge problem all across the globe. And we have solution. We have found solution, patented, implemented. Very, very simple technology. And, taking PhD, I found we are reinventing certain things and we are taking PhD. What has been done years and years and years back. And we have PhDs already, plenty of them. We know how to manage, but who is going to implement? Who is going to build the cat? That's a promising proverb. Who is going to build the cat? I have solution for that also. The next major pollution is, textile dyeing. The root cause for the dyeing pollution is salt, common salt. So we developed, with a local research organization, we developed salt-free dyeing. So no more pollution from textiles. USA can import the technology and have their dyeing unit there itself, because they export it, they outsource, because they don't want to create pollution there. Canada also may be same. So you can put up your own dyeing unit in your country. The next one is next major pollutant is our leather pollution, tanning. Tanning also we have made it salt-free. Salt is the major pollutant. There are other chemicals. All other chemicals can be eaten by bacteria, and we have developed those bacteria. Only salt can't be eaten by any bacteria. It's God's creation. If that happens, then the entire sea water will not have salt. Luckily, salt cannot be destroyed. So we can be converted into different form, but Salt acids cannot be destroyed. So we have found a solution. It's implemented already, approved by CLRI. It's a CSIR subsidy, CLRI in Chennai. And you name any pollution, we have solution. What other pollution? All I have covered? Atmospheric pollution. Air pollution. Air pollution, you know, you might have heard in Delhi, even today, it was around 490 PM air quality index. The air quality index, major cause is the PM particulate matter, 2.5 and 10. And we have solutions. Already this is known. This technology is not an issue. It's an implementation problem. Who is to implement this? Not only in India, all over the world. This is a problem. So I suggested, you know, all other, supposing Delhi you take, one is the PM matter, the other one is, there are five pollution issues. Carbon dioxide, the vehicle emission is there, carbon dioxide and carbon monoxide. Don't allow any vehicle which is not certified for Tier 6 engine. Don't allow inside. The Tier 6 engine is almost breathable. I have done that already. In USA we implemented and we can breathe it. But or you go for CNG engine, output is water, or you go for battery vehicles. But if a fellow from Rajasthan or UP, oh, I only have Tier 4 engine, how can I take my merchandise inside the city? No issue. Put a 30 kilometers away, put a stop and translocate the merchandise into a compatible vehicle, take it inside. So that pollution is gone. The next is road pollution. That is also emanating a lot of PM matters. Metalize, concretize all the roads and spray water, like your sewage water, treat it, we get drinkable water. I think some of you already seen, probably, we have the technology, we already implemented. Take that water, put a pipeline all across the highway and put a sprinkler, it will spray, keep it wet, very clean, no particulate matter after the transport. So next is the sewage water emission of H2S and all kinds of nuisance. So never allow the sewage water to generate. Treat it there itself and discharge. Make the rain drains are used as sewage drains. Stop that. That is not natural. Treat the water, treat the sewage water, put it on the drain and take it across and put it in a lake or somewhere. And that's a very useful water. Although we don't recommend for psychological reason for drinking, put it in a lake. After 1 kilometer away, pump it and put it back. You know, Mother Earth is a great filter. Put it back for drinking. It's allowed. Every lab will allow that. Test it. Test all the qualities. Already done. So you avoid sewage drain, sewage pollution. You avoid solid waste pollution. Solid waste also then and there. Within 24 hours you eliminate. Already implemented. We have the technology. Within 24 hours, solid waste is converted into useful products. So take this technology, implement across the globe, improve your living conditions for the betterment of our life. All the life, not only humanity. Anything, I am available. Dr. Madhumita knows, or a lot of colleagues knows. You just send a message to them. They will get connected. And I will be very useful for you people. Thank you.
Thank you, sir. Thank you for the motivating speech. It was truly inspiring. Now we shall break for the tea. We'll resume the concluding session by 4:15. Yeah, tea. 4:15. 1:5. I request the participants to kindly reassemble inside the hall for the concluding ceremony. I request all the participants and delegates to kindly occupy the front seats so that the seats in the front are not left vacant. Good evening, everyone. Respected Director, Institute of Forest Genetics. Good evening, everyone. Respected Director, Institute of Forest Genetics and Tree Breeding, Sri T. Ravikumar IFS. Respected guest, Professor Shanti Sri Dulipadi Pandit, Vice Chancellor, Jawaharlal Nehru University, New Delhi. Respected Organizing Secretary, Dr. Madhumita Dasgupta. Respected Co-Organizing Secretary, Dr. Rekha Arwarier. A warm welcome to one and all to the concluding session of the International Conference on Population and Conservation Genetics. I now kindly request Director IFGTB, Sri T. Ravikumar IFS, to deliver his welcome address.
A very good afternoon to you all. Professor Santishri Dulpudi Pandit, Vice Chancellor, Jawaharlal Nehru University, New Delhi. Respected delegates, colleagues, ladies and gentlemen. I once again welcome you all to the concluding session of the International Conference on Population and Conservation Genetics. The 3 days have passed quite quickly, thanks to the excellent quality of presentations and the deliberations, and I must say your accommodating nature. The insightful presentations enriched our understanding on the subject. The discussions on the sidelines, I am sure, will help us to collaborate in future. The knowledge and the perspective shared during the last 3 days will undoubtedly be valuable for the future research endeavors. The connections that we established during these 3 days, the ideas that emerged during the deliberations hold significant potential for joint research, knowledge exchange, and continued professional engagement in future. So I thank you all for making it possible to participate in this conference and actively participating in the deliberations. So I take this opportunity to thank you all, and I wish you a safe journey back home. Thank you.
Thank you, sir, for the kind words. Now I request Dr. Madhumita Dasgupta to deliver her address.
The presentation, please. The presentation. Yeah, thank you. So I'll just give a glimpse of what had happened for the last 3 days, followed by the recommendations which the chair, co-chair, and the moderators have given for each of these sessions. So as my colleague Naushad has already said, we had this conference for the past 3 days from 20th to 30th of January. And the organizers essentially of this conference is the UFRO Unit 2.04.01, which is on population, ecological, and conservation genetics. And we have been co-partnered by Asia Pacific Forest Genetic Resources Program, that is AFFORGEN. So the number of delegates who have as registered participants were 67, and we have delegates from 11 countries, including India. The next, please. Yeah, these are some of the photographs. The inaugural session was graced by the Director General of ICFRE, Srimati Kanchan Devi ma'am. She inaugurated this conference. And we had felicitations of the sponsors. And then the delegates were from not only research institutes, they were from academia, they were from state forest departments, paper and pulp industries, and we had also NGOs who have attended this conference. The next, please. Now, the different sessions, we had 3 sessions, 3 sessions. The first was on strategies to assess and trace adaptability of tree populations, where we had 3 invited speakers, Professor Om Rajora, Dr. Santiago Gonzalez Martinez, and Professor Philippos Aravinopoulos from Greece. We had 9
oral presentations, and we had 17 short oral and poster presentations. The session 2 was on conservation of tree genetic resources, where we had invited speaker Dr. H.S. Suresh from IISc Bengaluru, India. We had Dr. Manisha Thapliyal from HFRI Shimla. We had 8 presentations and 3 short orals. In the session 3, which was held today, which is on building collaborative networks and scale conservation efforts, we had the invited speaker, Dr. Jarko Koskila from FAO, and we had Sri I. Anvaruddin from the Tamil Nadu Forest Department, which also, this session also had 6 oral presentations and 6 short orals. So these are some of the glimpses of the presentations which were held in the last 3 days. The next, please. So other than that, we also have the short oral and poster. We have some awards given to motivate the speakers, which we will be requesting ma'am to do the honors. So then one of the highlight of this conference was to organize a youth forum, because we had a partner in SSVM, a group of institutions with us. So the aim was, this youth forum was held yesterday afternoon. The main aim was to foster youth participation in combating the key climate change issues, create a diverse and multidisciplinary network of climate-focused youth, and develop future climate champions. So there was an overwhelming representation from the schools, which we ourselves, it was very heartening to see so many children here talking on climate change, talking on environment, that idea on genetics. We were really surprised with the in-depth knowledge the children today have on genetics, population, environment, and climate change. So we had 12 schools, which included 3 government schools. We had 104 students from 12 schools, and 12 posters were presented. So this forum was graced by Dr. Manimekalai Mohan, the founder and managing trustee of SSVM Institutions, Coimbatore. Next, please. So these are some of the glimpses, the students, their presentations, and also the awards were handed over by our DG, Kanchan Devi ma'am. Next, please. Yeah, so this is all about the conference. So let us go to the recommendations, which is actually an important part of this conference. Because the conference was co-organized by 2 international bodies, IUFRO and AFORGEN. So the idea was to have these recommendations tabled in the forthcoming COP17 at Armenia. So that is why a lot of stress and significance was given, the next please, to these recommendations. I'll be reading out these recommendations as given by the chair, co-chair, and the moderator. Any corrections, they can always be, you can always mail me if you need any additions or corrections, which we will be compiling before sending it to COP17. So the first session was on strategies to assess and trace adaptability of tree populations. We had 2 themes under that club together. One was landscape and ecological genomics, and the other was genetics and genomic tools for tracing adaptive variability. We had the chair, Dr. Jarko from FAO. We had Dr. H.S. Suresh from TDU, and Dr. Nicodemus was the moderator. So the recommendations essentially was that the results which come from this genetic and genomic studies, currently the contemporary tools of omics, they need to be applied in conservation and management of genetic resources. Essentially, very, very few studies today are available where cutting-edge technology in terms of genetic and genomic tools are used. So a lot of emphasis was laid on that. Then the results of the studies on the distribution and structure of genetic diversity should be used more widely. Meaning to say that when we do genetic diversity assessments using markers, they need to be used in conservation priorities, in identifying seed sources, and also in reforestation and restoration programs. So we are essentially telling that we need to have a convergent conservation priorities when it comes to population studies in case of trees. The next is there are a lot of studies, I think, which were presented, which showed that there was tree populations were fragmented due to habitat loss or due to anthropogenic pressure. The idea was that most of the time we forget these isolated populations. We look more into those populations which are in continuum. So what emerged during these discussions was that these isolated populations may be harboring rare and useful alleles, and they also need to be conserved. Development of forest management practices, of course, we know that we need to have that in a more full-fledged way when it comes to tree genetic resources. The next, please. Coming on to the third theme, which was on genetic basis of local adaptation, where the chair was Professor Aravinapoulos, co-chaired by Dr. R. Yashoda, and moderator was Dr. V. Shivakumar. The recommendations like, we know that trees have a very strong selection pressure because their progenies are in millennium. You know, a lot of progenies come from tree populations, so they are naturally, they face a selection pressure. So local adaptation is a very strong, I think, driving force in case of tree species. So studies on local adaptation should be a key point when it comes to restoration programs. We need to look into these adaptive populations and then go for restoration instead of unscientific way of just taking some seeds and doing a restoration program. The next was using the concept of genomic forecasting. Genetic and genomic monitoring is today becoming a very important aspect of conservation genetics, where long-term monitoring is done to see how the gene flow is occurring. Is there a loss of gene, or is there an accumulation of deleterious gene alleles, and so on. So this forecasting needs to be continuously done in the field, which will help us to conserve the rare resources. Coming on to the omics approaches, I've said this earlier also, we have many omics approaches today. You have heard a company like Chromosome Lab, which says that they offer every services available. So today, the cost of sequencing analysis have come down drastically. So integrating omics into the, any of these population studies would be very advantageous. So that's what emerged as one of the recommendations. And similarly, using genetic markers to assess how the diversity is across populations, conserve populations which have very high diversity. If the diversity is low, we could do something like genetic rescue. We could bring in populations looking into their adaptability, and we could sort of rescue populations which may dwindle or which may become extinct over years. The next, please. Coming on to theme 4 and 5, which is on emerging technologies for developing climate-resilient trees and genetic monitoring and management. We had the chair, Dr. Santiago, co-chair, Dr. Nagarajan, and moderator, Dr. Matish. Here again, the stress was on using, advanced tools. So using cost-effective genetic monitoring approaches need to be developed, especially for the tropical species where we lag behind. And then using the population genetics using computational statistics and to understand inbreeding, genetic differentiation. That is, again, these are all overlapping recommendations, I would say, but since the chairs have recommended it, so I'm just showing it. So that should be the way of, an effective genetic conservation strategy. Then we need to, there was also a stress that we need to integrate the technologies. It should not be like we only do, genetic, we don't do phenotypic, we don't do functional traits. So integration of, because adaptation is a polygenic trait, so one technology may not answer all questions. So understanding local adaptations using multi-locus approaches, multi-omic approaches, combining them with functional traits, they're very important. Ecological traits are absolutely important because that's what we see in the field. So combining them together would probably give better insights. Then the new strategies of pan-genome, as I said again, the cost has come down, so we can have integration of next generation sequencing. Then the concept of accelerated breeding through genomic selections, again, these are new technologies which has come up. Our own institute works on teak. We are proposing to work on sandal. We have experts here from other countries who have implemented in other conifers and so on. So this is one thing we probably will need to start working upon to accelerate adaptive breeding. And then understanding adaptation, again, through other biotechnological means, whether it is transformation, whether it is CRISPR-Cas. So these technologies also need to be integrated into the programs. The next, please. Coming on to session 2, which is on conservation of tree genetic resources, we had theme 6 and 7 merged, which was on strategies for ecological and economic sustainability tree genetic resources, and we had habitat landscape restoration. We had, the session was chaired by Professor Rajora, co-chaired by Dr. Hannes, and moderator was Dr. Bhuvaneshwaran. So essentially here they tell that we need to have a PPP model. Today that is the mantra we say, you work with public-private partnership is very important today. We cannot work in isolation anymore. So PPP model needs to be brought into even for the conservation aspects, monitoring. When we work as institutes, academia, forest department, we can also bring in other players, other NGOs who are interested in conservation aspect together to understand and support the restoration and breeding programs. Then deployment of full SIP seed lots over, the monoclonal plantations, what we say, they could be sometimes very dangerous. They can be washed out by any pest and diseases. So advocating, use of diverse materials in clonal forestry. Site-specific management, yes, that's very important, that we need to know the place where we are going to restore or we are going to, bring in our material, the seed or the clones. So site-specific management is very important. Then in vitro priming strategies, there are some very, I would say, cost-effective strategies in enhancing the climate resilience of seeds, of tree seeds. The next, please. Then the theme 8 and 9 was on tree seed system, a very important part of forestry, and also on conservation and restoring of genetic diversity in Asia-Pacific region. We had the chair, Dr. Pin, co-chair Dr. Geeta, and moderator was Dr. A. Rajshekaran. Here, the stress was on a certification system, very essential in case of trees that we need to have a seed certification system at national level. Then followed by use of modeling tools for strengthening the seed source management, and then of course the capacity building of all stakeholders who deal with the seeds, the nurseries, the forest departments. They have to have a hand-holding from academic and research institutes to strengthen the tree seed system. The next, please. Coming on to the last session which we had today, building collaborative networks to scale conservation efforts. The theme was on strengthening collaboration to conserve tree genetic resources, chaired by Dr. Hisashi, co-chaired by Dr. Rekha, and moderator was Dr. Anandalakshmi. Here the recommendations were, again, since we were looking at networking, strengthening extension activities. That is considered to be one of the most important activity in tree genetic resource conservation. Participatory programs to accelerate deployment of improved germplasm across ecological regions. The use of improved varieties, whether seeds or clones, instead of uncertified material, so that you have better promise in the coming years from these materials which are planted. And a strong backup of extension is essential. The next, please. This was, I think, the last session on theme 11 and 12, participatory forest management and forensic timber identification, chaired by Dr. Annaline, co-chaired by Dr. Manisha, and moderator was Dr. Rajasuguneshekar. Here they were talking about, again, involvement of communities in all ecotourism restoration programs. Use of DNA barcodes, this is an upcoming field, timber forensics. Very soon, it will, all countries would have to sort of comply with the supply chain using markers where we need to prove that the material which is entering the trade is sustainably harvested. It is not illegally illegally taken. So we will be, because we have such supply chain regulations in EU, in US, so we will be very soon getting, they will get implemented in every country when we try to export. So this is one area we consider that India should be working on. And collaborating with other countries who are also working, so that we have what we call as a global reference library, which we can use for timber forensics. And then again, coming on to collaborations with national and international forestry institutes, because ICFRE is the premier institute in forestry education, forestry research and education. So probably we should have more collaborations, is what the delegates felt. And then integrating participatory approaches, again, as I said earlier also, we cannot work in silos, we need to work in group. So this is an era of networking, so that is what has been emphasized by every sessions. The next, please. Yeah, these are the recommendations I've just summed up. So if you feel there are any more recommendations you would like us to add before it goes as a recommendation of the conference to UFRO, Afforgen, and finally to COP17, I request that you all have the conference email ID with you. So please mail us your thoughts and we will integrate it. Thank you so much.
Thank you, madam. Thank you for summarizing the conference and giving a detailed account of the recommendations made by the delegates. Now we have the prize distribution ceremony for best short oral presentations. Now I kindly request Professor Shantishree Dulipati Pandit, Vice Chancellor, Jawaharlal Nehru University, to kindly distribute the prizes for the best oral presentations. So under the theme 1, 2, and 3, first prize goes to Muthulakshmi Ishwaran. Second prize goes to Elias Noor Nabila from FIRM, Malaysia. Now on to theme 4 and 5, the first prize goes to Manoj Kumar R from ICFR AFGTB. Second prize goes to Prachi Upadhyay from IIFM Bhopal. Now under themes 6, 7, 10, and 11, the first prize goes to Komal Ari from ICFR AFGTB. Second prize goes to Dr. Mayavel A from ICFR AFGTB. Now I kindly request Director IFGTB to felicitate Srimati Shantishree Dulipati Pandit with a memento. Now I kindly request our guest, Professor Shantishree Dulipati Pandit, Vice Chancellor, Jawaharlal Nehru University, to deliver a special address. Okay. I request Dr. Madhumita Dasgupta to introduce Madam.
Thanks, Naushad. Before, I think ma'am doesn't require, introduction, but still we have delegates from other countries also. So a very short introduction to Professor Shantishree Dulipati Pandit. Ma'am is a Vice Chancellor from Jawaharlal Nehru University, New Delhi from 2022. She holds a PhD in International Politics from the School of International Studies, JNU, post-doctorate in Peace and Conflict Studies from Uppsala University, Sweden. Ma'am has a distinction for being the first woman and alumnus Vice Chancellor of India's most prestigious university, which is internationally recognized. Yeah, I need to underline that, an internationally recognized university. Her expertise includes international relations, Asian studies, culture and foreign policy, conflict, violence, and gender. Ma'am has a teaching and research experience which spans for more than 35 years. And she has published 4 books, edited 2 of them, and I think there are several peer-reviewed publications which I don't have the time to narrate now. She is presently, a member of, I think, many committees. What I discussed yesterday were in BIRAC, CSIR, ANRF, biotechnology. So I think the list must be very huge. And another very unique distinction she has is she knows 6 languages. So multilingual person, I should say. And ma'am is also a recipient of several awards. I'll just name a few before handing over the floor to you, ma'am. She is the recipient of the Youth Forum for Gandhian Studies, Chennai Women Educator Award, Vice Tex Award for Women Leaders in the Next Decade, Veer Savarkar Award for Education, Srimati Sushma Swaraj 3 Shakti Award, Universal Peace Federation Ambassador of Peace Award, and Deen Dayal Upadhyay Award. I think the list is big. I've just concised it, ma'am. So I now request Professor Pandit to kindly take on the floor.
Good afternoon to all of you. Ellarkum vanakkam. And let me, respected Director, ICFRE, Coimbatore, Shri T. Ravi Kumar, Dr. Madhumita Dasgupta, the Organizing Secretary, and we have the Dr. Rekha Warrior, the Co-Organizing Secretary, delegates, friends, ladies and gentlemen. Let me tell you, I think I'm the most ill-qualified. I'm not an expert on environment, but maybe the reason of calling me, maybe I'm Jawaharlal Nehru University's Vice Chancellor, which has certain good things about environment. We are some of the better practitioners in India's university system. Minimum forest area that is required by any university is 15%. JNU has 42%. And we have 2,000 acres of land today in the center of Delhi. So I can also say I'm one of the richest landlords of Delhi. And the other is we have a very good School of Environmental Studies, which works very closely with the Government of India, as well as the Special Center for Disaster Management and the School of Life Sciences, which also does a lot of work on plant genetics and as well as tree genetics and others. And recently, we got the Prime Minister's research fund of 500 crores. We are the only university competing with Indian Institute of Science, the IITs and NITs, for a project which was on environment. I, as a Vice Chancellor, I'm the first non-science Vice Chancellor of JNU, because in India, scientists dominate the posts and men. And I think I'm the first non-science VC to support science research. So maybe these were the issues that, and also recently, let me tell you, Jawaharlal Nehru University with IIM Mumbai and the World Intellectual Forum gave the world the first ranking. You know, the West has always ranked all of us. For the first time, we gave the Responsible Nation Index, where we made one of the things environmental responsibility and climate change towards as one of the things which 53 indicators. We also had a university, the University of Paris from France, which authenticated many of ours with the United Nations and UNDP and others. So Jawaharlal Nehru University worked on this for about 3 years. So let me tell you, maybe that was the other, and we also are the university that builds the narrative for the narrative architecture for the government of India. Let me at the outset congratulate all of you who have participated in this conference of International Conference on Population and Conservation Genetics. And I'm very happy you've involved young people, madam, because that's very, very important. And community involvement, which is even more important because universities can no longer be ivory towers or institutes like yours. We have to reach to the larger community. And let me here only try to tell you the little I have always been reading. One is, what I want to give you is What has been the Indian ecological narrative? Do we have an alternative narrative to the West? I would say yes. And here I would bring in the ideas brought in by Jagdish Chandra Bose, who very early, many, many years ago, told us plants and trees also are sensitive. They also, if you talk to them nicely, be nice to them, they grow better. At the time we thought it was funny, but today it has been proved. And I think that is extraordinarily important because this brings in our concept of the environment. We are not human-centric civilization, we are a nature-centric civilization. Unlike the West, we don't say you have to conquer nature. We say we have to live in harmony and balance with nature, especially the cosmos, because human beings are just a small element with all other elements, species, plants, animals, bacteria, everything in the cosmos. So with this, I would rather say we also have concepts where trees are given divine symbolism. The concept of Devarai, where, we have the mother goddess placed outside the forest. You're expected not to cut certain trees, and maybe that is how they taught us conservation, because anything that is divine then does get, so we always mixed science with spirituality. And I think that is very important. Being just materialistic does not work. And I think that is the alternative framework we have given, what I would call the theories of the Panchabhutas, that is balance and harmony. As most of you would have done, you're all genetologists, but finally you are telling us that as human beings, we cannot destroy the nature that has been given to us. Otherwise, we may not be able to sustain the planet for several years coming. And population also, I don't know, I think new theories have come in, though I think today we are the most populous country in the world. But at the same time, population is both seen as an advantage and a disadvantage. At least when I was growing up, my grandmother told me for every one human being, there should be 20 trees. I don't know whether we have that number, but I think that is what takes us to the balance. And here I've been seeing through the topics and other things, I think most of you must have done it very well. I invite you all to Jawaharlal Nehru University and even with your institute, sir. I think let us not work in silos, but I think we should share knowledge. Sharing knowledge is extraordinarily important. I think we have a lot to learn from the West as well as from ourselves. And it's a two-way process. It's no longer a one-way process. And here I would say there has to be a balance between the Panchabhutas, that is water. Today, there is a substantial scientific evidence to show that water has tremendous memory. If you just generate, a thought looking at the water, the molecular structure of the water will change. If you touch it, it will change. So how you approach water is extraordinarily important. The other is earth. And I should congratulate our Manikam sir, who's a great industrialist who has been allowing us for reusage of especially wastewater. And that is why I came here to meet. And the next is earth. For us, earth is treated as mother goddess. Let me tell our international friends, we are a feminist civilization, sir. You would ask me why. Even among our gods, the most powerful portfolios are with women, not with the men. Right? Goddess of wealth, all of us want money. Right? I always say, you can't be a strict economist who doesn't understand power. Then you're a perfect idiot. Or a politician who doesn't understand the value of money. And I think recently with Trumpism, Europe must have realized just economy is not enough. Charles de Gaulle comes back. He warned the whole of Europe not to outsource security. So finally, security, so a primacy of the political. And political economy go together. Hence, power and money are twins. So I would rather say this is extraordinarily important for us today. And I'm very happy that thanks to Trump, European Union and India have come together. So it's a benefit we have got, though in a period of tremendous uncertainties, anxieties, and others. So I think earth is important because earth element is an element that there is a basis that all other elements are built on this physicality. Though people tell us that we can capture Mars, you can capture moon and go everywhere, but as of now, we just have the earth to live on. And it is extremely important that earth as a physical matter around us too, and it is important the way we perceive it. And I think your forests, your conservation, and we as human beings also come within this. Think, why is JNU acting as though it's running the government? So we are really running the government. We have 3 ministers in the present cabinet, 6 opposition leaders, and 30% of the bureaucracy belongs to JNU. So we have quite a lot of power when it comes to that. And hence, we think that we can use this at least for Delhi, which is indeed a gas chamber. Only JNU is saved thanks to the trees, sir. Our AQ level is nearly 100 points less, and our temperatures are much lower. Because please visit our campus. We have got nilgais, peacocks, even wild animals live with us very peacefully. Let me tell you, snakes, as well as jackals, none of them have bit our students because our students are too radical. Maybe they are also very scared of them. So, we are left of the center. We have a lot of very strong. The other is fire. In Indian system, fire gets a very important value, and it has. And finally, Akasha or ether, which is the cosmos, because we believe that energy and the cosmos, it's not just the earth, it is also the cosmos that we influence. And I think the conferences like this, sir, you should do it in all parts of the country, because it's extremely important that environment, the importance of Mother Nature is extraordinarily important. And I thank you all for inviting me, and I would rather say that we are more the implementers, and maybe whatever good ideas, madam, you have, do even percolate to other institutions, and maybe all the central universities and IITs. We are neighbors to IIT Delhi, Indian Institute of Technology, and we also have a common master's programs and other programs. And I will surely take this thing to my School of Environmental Sciences, and we hope that young people like you will be greater scientists. Whenever I was just telling sir, but he corrected me, the scientist I knew best was M.S. Swaminathan, when he talked about genetics and rice varieties being done in Philippines, and hence these are issues that I would like to highlight. And thank you for inviting me here. Though I was totally unprepared, it was just yesterday that I was asked that I should be here. Once again, I thank you all, and would like to remind you that there is a lot of knowledge with communities which we have to enthrall. One last idea I want to give you. When we had the tsunami, let me tell you, all the animals ran away in Andaman Nicobar. It was only educated Indians like me who refused to run away. Even the tribals ran away because they understand when there are danger signals. And it is educated people like us who died or who faced fatality, not the animals or even the tribals. So I think there is a lot of value. University education or degree doesn't mean real education. Let us be very humble and learn from these practices from all over the world. And once again, let me thank you for inviting me to talk to you, and thank you once again. Jai Hind, Vande Mataram.
Thank you very much, ma'am. So it was an extempore talk with a tinge of humor, with lot of motivation, lot of insight, and ma'am has invited us to JNU. We'll surely go there. I'm here to request, where is Professor Phil? You wanted to say something about the conference. So.
Madam Chancellor, dear colleagues, and now I will say dear friends, because we are at the last day of the conference and we have gone through 3 wonderful days. It comes this time that the conference is about to end, and in some cases, everything falls into place. We all know what each other is doing. The questions we ask are more interesting to each other. The discussions in the break are more interesting. And what does that show? It shows that the conference was very successful. And I would like to thank the local organizing committee for the extremely nice work done, and for the fact that through our UFRO unit on population conservation and ecological genetics, together with Up4Gen, and through this wonderful institute, we were able to have this extremely nice conference. And then the next question is, why is this happening? Why at the time that we are so becoming close together, so developing some collaboration, so thinking what to do next, why do we have to go away? Well, the answer is very simple, because the next conference will be coming up. And our next units conference is a place that I would like to invite everybody, all of you, please do show up in these wonderful meetings. Let us continue our collaboration. Let us work together. Let's advance science. And let us take something of this inspiring speech the Vice Chancellor just gave to us. Thank you very much to all. Thank you.
Thank you, sir, for the nice comments. Now I invite Dr. Rekha R. Warrier, Co-Organizing Secretary, to deliver formal vote of thanks.
A very good evening to all of you. It is my privilege to propose the vote of thanks at the conclusion of this enriching and successful conference on population and conservation genetics. At the outset, I express my sincere gratitude to Professors Shanti Shri Dulipodi Pandit, Honorable Vice Chancellor, Jawaharlal Nehru University, Delhi, for gracing the valedictory session and for her insightful address, giving us lots of thoughts to ponder upon, and which has added great value to this conference and inspired us all, not just in terms of the research, but also in terms of our everyday lives. I express my sincere thanks to the Director General, Indian Council of Forestry Research and Education, Srimati Kanchan Devi ma'am, who inaugurated this conference. Madam could not be a part of this valedictory session, but she conveyed her good wishes here and wished that we collaborate better for the betterment of forestry of the country. She is away due to some other pending engagements. I place on record our deep appreciation to Shri T. Rabi Kumar, the Director of Institute of Forest Genetics and Tree Breeding, for his constant guidance and unwavering support in ensuring the smooth and seamless conduct of this conference. I extend my special thanks to all the international and national delegates, keynote speakers, chairs, co-chairs, moderators, presenters, and participants for their active involvement, stimulating discussions, and valuable contributions which have made this conference a truly meaningful and impactful one. We extend our sincere thanks to the Youth Forum participants for their enthusiastic participation, insightful presentations, and forward-looking perspectives for a meaningful dimension to the conference and for a greener future. Our heartfelt thanks are due to the collaborating and partner organizations, including the UFRO, APAFRI, Up4Gen, IIFM, and other national and international institutions whose association has enriched this conference. We sincerely thank our sponsors, partners, exhibitors, and funding agencies for their generous financial and logistic support, without which this conference would not have been possible. Their commitment to advancing forestry research and sustainable development is deeply appreciated. Here, I would like to mention specifically the untiring efforts of Dr. Madhumita Das Gupta, the organizing secretary, towards the successful conduct of this conference, and her contribution deserves a special mention. Her indomitable spirit, her unwavering resolve, and steadfast determination, even in the face of personal tragedy, continues to inspire us all. I also acknowledge with gratitude the support of the press and the media for covering the event and disseminating the conference outcomes to a wider audience. We sincerely thank the hotel management and the staff for their excellent hospitality and the professional support, which has contributed significantly to the smooth conduct of the conference. Finally, I wish to place on record my appreciation for the support and dedicated efforts of the IFGTB family, the officers, staff, volunteers, and students whose hard work behind the scenes ensured the success of this event. I am confident that the deliberations and the recommendations emerging from this conference will translate into meaningful actions for the conservation and sustainable management of our forest resources. Thank you once again for all your support and participation. Jai Hind.
Now I kindly request all the participants to rise for the national anthem.
I request all to kindly join me for the national anthem. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल वंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाए तब जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. Thank you. So I think you can come and announce. Yeah, photo sessions with you. That's the on popular demand.
Please come forward.
We'll have
two set of photographs, one with our in-house people and with the international delegate, please.
Please, delegates, please.
Matis, please. Yeah, please.